All right. So kicking off our scientific component of the program, we're fortunate to have Alicia Gamboa here from Barry Stripp's lab and the PhD program, and she's going to be sharing with us some of the work that she's been doing in single cell transcriptomic profiling of lung cells. So Alicia, why don't you come on up and we'll get your talk queued up.
Can everyone hear me okay? Okay, great. Okay, so the title of my talk today is, is that better? Okay, so the title of my talk today is Lipocalin-2 Regulation of Alveolar Type 2 Cell Regeneration Following Influenza-Induced Acute Lung Injury. And I'm just going to provide some background on the project and how this led us to choosing single-cell RNA sequencing as the technology to answer the questions we're interested in. So I'm in the lab of Dr. Barry Stripp, and we're really interested in the role of lung progenitor cells in maintaining lung function. And here is a cartoon diagram of mouse and human epithelial cells that make up the lung. And this can be divided into two zones. The first, the conducting airway zone, which is the trachea and the bronchi, which consists of cells such as serous, ciliated goblet that are important for producing mucus and mucociliary clearance, with the basal cells being the progenitor cells of the upper airway. And then as we go deeper down to the bronchioles, to the distal part of the airway, or this respiratory zone, there are two major cell types in both human and mouse. These are our alveolar type 1 cells, which are critical for gas exchange, and our alveolar type 2 cells, which produce surfactant and provide stability and prevent collapse of the alveolar type 1 cells. These also serve as facultative progenitors of the distal lung with the ability to self-renew and differentiate into our alveolar type 1 cells. And in the normal lung, there's very little turnover of these cells. They are not very active. However, in situations of disease, if these progenitor cells become too active, they can cause things such as cancer, as in over self-renewal. And if they lose their function and become senescent in extreme cases, this can lead to idiopathic pulmonary fibrosis. This is why we really want to understand what regulates the self-renewal and differentiation of these type 2 progenitor cells, both under homeostasis and in response to injury. So as I mentioned, these are facultative progenitors, meaning they require some external stimulus to assume an activated state. So this is thought to be through the integrated stress response, or ISR. So some external signal, whether it be unfolded protein, ER stress, iron deprivation, amino acid deprivation, or viral DNA, this will activate one of these 4 protein kinases to phosphorylate EIF2 alpha, which leads to a global reduction in translation and specific transcriptional regulation that will determine the cell fate to either undergo apoptosis, to renew, or differentiate. And under homeostatic conditions, there's an adaptive ISR response where these type 2 cells can become this transitionary cell that can then differentiate and repair alveolar type 1 cells. However, under pathologic conditions, the ISR becomes dysregulated and these cells lose their ability to regenerate into these type 1 cells. So what we want to know is what regulates the activation and the differentiation of these activated type 2 progenitor cells. And the model that we like to use is respiratory viral infection. So zoonotic viruses such as H1N1 have the ability to penetrate into the distal lung and damage the alveolar region, which can be seen in these H&Es over here. So within the normal lung, you can see these thin alveolar structures with lots of airspace. However, after injury, these cells thicken and we lose that airspace and lung loses its function. And another characteristic of H1 influenza injury is the expansion of these basal cell keratin 5 positive cells into the distal airway of the lung. And we want to understand what mechanisms drive alveolar repair and remodeling in response to viral infection, because this does damage the lung, but it also, these cells are able to repair and resolve the injury in most cases. And it's nice because we're able to model this in mice. We can give mice influenza virus, and you can see a normal lung, mouse lung here on the left, with our type 1 cells being stained in red. And then our injury-induced lung on the right after PR8 or H1N1 influenza infection. You can see the damaged regions by loss of the type 1 staining. And also similar to what we see in the humans are the expansion of these keratin 5 basal pods. So it's phenotypically similar to what we see in human patients. And the reason we like this model is because it's a known ISR activator. So we know that these mature cells are getting activated by an ISR response. It results in damage of our alveolar type 1 cells. And we know that these type 2 cells are able to differentiate and repair our type 1 cells and also self-renew. So a former PhD student in the lab, Andrew Beppu, did a comprehensive single-cell RNA sequencing analysis of various time points post-PRA infection. And what he found was this protein, the pulcalin-2 or LCN2, was predominantly expressed within the epithelium as opposed to the immune cells, and peaked at early stages post-infection and then went down at later stages. And this peak was typically around 7 days and started to go down after about 14 days. And when we see what cells are expressing LCN2 within the epithelium, it's predominantly our alveolar type 2 cells and specifically a subset of these cells. And furthermore, if we look where are these cells located, we typically find our type 2 cells colored in green tend to be near the border of the injured regions expressing high levels of AT2, suggesting that they're playing some type of role in repair of the alveolar epithelium. So what do we know about LCN2? It's produced in many cells across various types of organs. High levels of it are associated with fibrosis and injury in many of these organ systems. Its most well-known function is bacterial defense and iron homeostasis with the ability to bind hydrophores and limit iron uptake. It's also can bind metalloproteases and responsible, therefore, for cell migration, and can bind hydrophobic molecules such as steroids and hormones for increased distance of transport. And also known for activation of cell states, which has been shown in astrocytes. LCN2 is produced in their activation state. And we believe it also plays a role in the activation of our alveolar type 2 cells. So this led us to our question, what is the role of LCN2 in the activation and what aspect of this pathway is it potentially regulating? So this led us to do single cell RNA sequencing. We had 3 genotypes, so our LCN2 wild type, LCN2 heterozygous, and LCN2 knockout, and 3 time points, day 0, an early time point post PRA at day 7, and a late time point at day 14 for each of these genotypes. Harvest the tissue, we performed FACS enrichment for epithelial cells, and then we used the parse kit, which was great because you have the fixation. I was able to process like 4 samples at a time, store them, and then do all of the library prep and barcoding of the samples simultaneously. And because it's a well plate format, it was nice because I was able to maintain the biological replicates for all of our samples. Then we performed sequencing, and we were really happy with the quality of the data. And now we'll get into our analysis. So we were able to characterize all of the major cell types you can find in the lung. So we have our epithelial cells, which can be divided into airway here in purple, alveolar in blue. We have endothelial cells in orange, immune cells in red, and our mesenchymal cells in green. And then we looked at where is LCN2 being expressed across all of these cell types. And again, similar to what we saw in Andrew's data set, it's specifically highly expressed within the alveolar epithelium, and particularly within a subset of these alveolar cells, as you can see from this density plot and the heat map below. So we then reclustered this alveolar cell population, and we identified 7 different subsets of these alveolar cells. So we have our type 1 cells shown in this dark purple, the activated cells in blue, BAS or bronchial alveolar stem cells in green, a cycling population in orange, infected population in red, this mature in purple, and then our transitional cells in this light blue. And then we looked at LCN2 expression across the subtypes. And again, we see that LCN2 is highest within this activated type 2 population. And if we look over the time course, we see at day 0, LCN2 is low. It gets increased at day 7 after the onset of this viral injury, and then goes down at day 14, suggesting it's playing some type of role in the repair of the lung. So this led us to look at the changes in alveolar composition over our time course after PR8. And we can see at our day 0 time point in wild type, knockout, and heterozygous mice in our stacked bar is mostly composed of our mature type 2 cells, which is what we would expect. And then after PR8 infection at day 7, we see the increase of this activated type 2 population in all of 3 genotypes. At day 14, we see the disappearance of this population, along with the increase in transitional and cycling type 2 cells, with a significant increase in our LCN2 knockout of these transitional and cycling cell types relative to both the wild type and het, with the het phenocopying the wild type. So then this led us to think these activated type 2 cells might be giving rise to these transitional and cycling cell states. So we performed an RNA velocity analysis. And if you look at the trajectory of the arrows, we can see that the activated type 2 cells seem to be giving rise to our cycling and transitional cells. And so we wonder, does LCN2 regulate the differentiation of activated type 2 cells in the cycling and transitional cell states? So we have a nice organoid model. So I isolated lineage label type 2 cells from both wild type and LCN2 knockout at day 0 and 10 days post PR8 infection, and quantified the colony forming efficiency of these cultures. And you can see after PR8, there is an increase in both wild type and LCN2 knockout colonies. And this is likely because after PR8, we now have these activated type 2 cells within the culture. But specifically within the LCN2 knockout, we see even higher number of colonies relative to the wild type, suggesting that LCN2 is regulating the clonogenic potential of these type 2 cells. And furthermore, we not only see more colonies, but also a larger size of these colonies as well. So then this led us to wonder what mechanism might be driving these differences in these activated type 2 cells. So we performed a gene set enrichment analysis. And I know this is hard to read, but these are the top gene ontology terms. And I'm going to show that most of these are related in some way to the ISR pathway. So we have pathways related to translational regulation, pathways related to autophagy, pathways related to ubiquitination and the unfolded protein response, and pathways related to phosphorylation. So then we wanted to look more into ISR genes specifically. So this is a volcano plot of differentially expressed genes in our activated type 2 LCN2 knockout cells, and specifically looking at genes within the ISR pathway. And we see one of the top genes is EIF2AK2, which encodes for PKR, which is the kinase necessary for activation of the response, and EIF2S1, which becomes phosphorylated and is required for the downstream signaling of the ISR. And if we look at expression of EIF2AK2, it is very specific to this activated type 2 cell population. And that the expression of EIF2AK2 and EIF2S1 at the transcript level is significantly higher in our LCN2 knockout, as well as at the protein level. So you can see in the Western blot, knockout on the left, wild type on the right, and relative expression to beta actin is quantified below. So in summary, we have these mature type 2 cells that under some stress response, in this case a viral response, leads to activation of these cells in this activated type 2 state. These cells produce high levels of LCN2, as shown in the density plot. And they have the ability to inhibit the ISR pathway, likely through activation of this PKR, since we see lower expression of PKR in the wild type. So there seems to be a negative feedback loop. Our trajectory analysis suggests that these are the cells giving rise to the cycling and transitional cell populations. And from the bar plot in our organoid cultures, we see this difference in the number of cycling cells as well as these transitional cells. And with that, I would just like to thank everyone for listening to my talk. Thank you to my mentor, Dr. Barry Strip, and everyone in the Strip Lab, particularly Changfu and Gianni for teaching me how to do the single cell analysis, Catherine for all of her help with the mouse work, previous Strip Lab members, Andrew and Juan for getting the project started, our collaborators, AP and Promoda, my dissertation committee, and then Parse. Brian was really great throughout my whole process of from the fixation all the way to the barcoding, and then Sarah for inviting me to give the talk, and thank you.
Everybody, can everyone hear me okay? Yes? No? Awesome. Well, hi, thank you all so much for coming today. My name is Sarah, and I'm the technical sales manager for Parse Biosciences here. And first off, I want to give a big thank you to Alicia for coming to share some of her Parse work. And I'm just going to give a quick little synopsis and overview of Parse technology if you aren't aware. So, yeah. So I like to show this slide first because it kind of gives a nice idea of where the single cell field is headed. And so it's basically a graph summarizing where the publication trends are over the past basically few years for single cell studies. And as you can see, as we've kind of come into more recent years, there's more and more papers that are using single cell, and those papers consistently have more and more cells. Hence the need for more scalable single cell technologies, right? And that's pretty much where Parse fits in. And so if you're unfamiliar with how Parse Biosciences breaks up, there's basically multiple different flavors of kits for any size of single cell project. You can do anywhere from like 10,000 cells all the way up to like 5 million, even 10 million cells in a single experiment. We specialize in whole transcriptome profiling, but there's also TCR and BCR profiling, CRISPR screening, gene panels. So there really is a flavor for kind of most project needs. And just to give you a quick overview, so I've already kind of harped on the scalability, but what's really nice is that there is no instrument that you need. So there's no startup costs, really. Everything you need is pretty much on your lab bench, most likely. So like a multi-channel pipette, a thermocycler, a centrifuge, very standard stuff. We have really great data quality, which I think Alicia definitely mentioned in her speak. But we also are powered by the fixation step, which if you're doing time course studies, asynchronous samples, maybe you don't want to harvest all of your mice in one day. It's a nice way to break up your workflow. And just to give you an overview, it's very similar to most single cell technologies. So the first step is fixation. You can fix all of your samples down. You have 6 months of stable storage. And then from there, once you have all of your samples ready, you can do barcoding and library prep all in one go. As you can imagine, this helps to eliminate batch effects, right? You're doing everything all together. You can sequence on really any platform, and then we have a user-friendly analysis pipeline that is free to use for any academic user. And just to give you a quick overview, so basically we're built on fixation, and part of that reason is because we're doing everything in situ, and fixation really allows us to do so because you're fixing and then adding a perm step, which basically allows you to pump in the barcodes inside of your cells themselves. And this step itself is what allows us to bypass the need for any sort of instruments, right? Because everything is occurring in situ. Barcoding is relatively straightforward, right? It all takes place on your lab bench. You're doing everything on 96-volt plates. It's called combinatorial barcoding. So the idea is you disperse your fixed cells across 4 different rounds of barcoding, and every single round, a cell is going to pick up its own unique 4-barcode combination, or a unique barcode, so such that by the end of it, every cell has a unique 4-barcode combination. And that's what you can use to differentiate cell type, sample origin, all of that good stuff. All right, and I will leave you with a quick overview of our Trailmaker platform, which is great for really any single cell user. It's agnostic, not just to parse data, but if you are using any kind of other single cell platform, you can actually upload your matrix files on there. It's no coding involved, so if you're new to single cell, it's a really great way to get your hands dirty with the BI work. But basically, it'll take you from FASTQ files all the way through the analysis to generate publication-ready figures. Very user-friendly, as I mentioned, and it's free, which is great. I think we're out of time here, so I will just leave you with a thank you, and we're right outside on the tables if you're curious to learn more or want to talk about any single cell plans.
Hello. Hey, Thank you all for having us here. It's a pleasure to be introducing Xenium and talking about it before Joe presents how it was used in his research. 2 years ago, I was here in the same room. It was raining in LA. Two people showed up, both were competitors. So it's uh, it's nice to have a full room. At 10x, I used to work for Akoya, by the way, 2 years ago. I just joined 10x 9 months ago. Um, Anyways, at 10x, we do want to empower researchers to identify biomarkers, and we're making a big push towards that. And now we have a whole team around biopharma as well. So a lot of biopharmaceutical companies are using our products. And what I'm going to be focusing us on Xenium. You all know about 10x's leadership in single cell, and I think the Vision platform was launched a few years ago. The latest platform is Xenium, which is what Joe's talk is going to be about. The Xenium platform is able to do single cell imaging at high resolution and up to 5,000 genes. And I'll talk about that a little bit more. But before that, the platform itself is fully automated and high-plex resolution spatial imager. I think these attributes that you see on the slide focus on all the problems that 10x is keen to solve, right? You have an automated platform, you have multimodal segmentation, you have a simple workflow, no optimization. It's compatible with downstream IF and H&E workflows, and also the panels are customizable, and the analysis is easy. This is really important when you're a postdoc or a technician or a PI in the lab and you want reproducible data. Your techs are going through your lab, your postdocs are rotating. You want to make sure that any technology or instrument you use is really simple to use so you can get reproducible data, not only within your lab, but within your collaborators' labs as well. And 10x has tried to address these problems with these attributes. The other thing I'd like to highlight is 8 independent studies have benchmarked Xenium against other spatial instruments. We know outside there's multiple spatial instruments, spatial transcriptomics. Customers are typically very confused as to if I have to spend a lot of money, what technology I should spend on, and they should do their evaluations, their studies. They should be very careful about that because these are expensive purchases. But what I like to say is these 8 benchmarking studies had a takeaway that said Xenium detected more transcripts. It could spatially resolve and discriminate different cell types and showed greater specificity, and also exhibited greater consistency with single cell RNA-seq. And the biggest reason for that is our robust chemistry, which is essentially a padlock chemistry. You have a padlock, 2 sides of your base pairs have to attach. Only if they attach, then they have to ligate. And only if they ligate will the rolling circle amplification work. So we have a lot of mechanisms to avoid false detection. And that's why we have high sensitivity, high specificity. And this also allows us to do high homology transcripts. And you can detect isoforms and single nucleotide, SNVs and SNPs. So there's a lot of advantages to this chemistry, and especially for short degraded FFPE transcripts. Again, this is also tunable chemistry. So if you have low expressors, you can increase the number of probe sets. And if you have high expressors, you can decrease the number of probe sets. The other thing I wanted to highlight is customizing panels. We have off-the-shelf panels. You can customize panels. And we have 5,000 gene panels for human and mice. And you can fully customize your own species panels. We've worked with fish, plants, and other kind of tissues. So it's very easy to do that using our online tools. And on the right side, you'll see all the sample types that we can work with. It's a broad sample type. If you have a tissue that's good quality, we'll be able to work with that. Last year, we introduced multimodal segmentation. We included a cocktail of 7 different components that can segment your cells accurately. And we also have an algorithm that does that pretty well. So we have a boundary stain, we have interior RNA stains, we have a nucleus stain, and then the interior protein stain. Overall, the feedback from customers using this has been pretty remarkable. Coming to the workflow, it is a simple workflow. Again, I can't highlight this enough. Most posters, if you see the poster outside, the advantage is easy workflow. So you spend around 3 to 6 hours of hands-on time. Once you get to your probe hybridization and ligation, you then get on to staining for your cell segmentation. And after that, it all is walk-off automation on the instrument. At the end, you get all your data on the Xenium analyzer. You can do a 5K prime, which is a 5,000 gene panel, or your customized panels or smaller panels. And you can add on genes to these panels as well. The other advantage is that the imageable area is fairly large, so you can put in multiple tissues, and you can also create TMAs if you want. And overall, you can then analyze your data in third-party software or on the analyzer. And one thing I'll leave you with is that this workflow is non-destructive. You can follow this up with H&E staining or aqueous staining if you want, or a lunaphore. Anything you want afterwards, the tissue holds. And if you want to do a Visium after a Xenium, you can do that as well. Now, coming to what we're going to launch later, I think in a few weeks, is protein plus RNA. And that's coming up. It is inline protein. We have 27 proteins focusing on human FFPE. And these would be one single shot, one cocktail. It's optimized to work with a variety of tissue types, FFPE, and it works with a smaller, up to 480 genes RNA panels. So if you're interested in that, talk to us outside. I can give you a list of proteins. This is the list of 27 proteins. And the next thing on the roadmap is customizing these. So this is all I have, Joe, and thanks for presenting. And I wanted to focus on the technology so Joe can focus on the science. And thanks again for speaking.
All right, I'd like to start by thanking 10X and also organizers for recommending me to speak today about some of the work that I've been. Today I'm going to talk about EGFR mutated non-small cell. All right, so I have no disclosures for this project. So I'm going to talk a little bit about how we can think about molecular diagnostics in non-small cell lung cancer and how we can use some of these newer spatial technologies to kind of reinvent how we think about biomarker discovery. So I'm going to start with talking about early non-small cell lung cancer molecular diagnostics and then move into where we are now. And then how do we start to implement some of these assays into biomarker discovery? So EGFR has been known for almost 3 decades now, to be involved in non-small cell lung cancer. And it was found to be overexpressed in 1993. It was really the first study. And this led to the development of EGFR specific inhibitor, TKI inhibitors. So this was gefitinib was one of the early ones. And it did not do very well in trials. And if you look, there's really no difference between non-small cell lung cancer patients treated with gefitinib and not treated with gefitinib. So this kind of failed initially upfront, but then it was pretty shortly after when they started to look at some of the molecular characteristics of these patients that they started to see some very important differences. What we can see in A here is patients with a high EGFR copy number, so they have overexpression of EGFR, and they have an EGFR mutation, do better with the addition of gefitinib, while patients that have high EGFR expression by copy number, but have a non-mutated EGFR, do not do well with gefitinib and actually do worse. So it really suggests that it was a mutation-specific event that was allowing gefitinib to have its benefit in these patients. And this led to gefitinib and then additional TKIs to be approved for first-line therapy in non-small cell lung cancer patients that were EGFR mutants. So current molecular methods that are used for non-small cell lung cancer. So EGFR copy number was initially used quite a bit, but this has really fallen out of favor now that we're really focused on what the actual mutation is. And this moved to Sanger sequencing before we had next generation sequencing being mainstay. But now pretty much every lung cancer patient is going to have next generation sequencing, specifically looking for that EGFR mutation as well as a few others. And then over the last decade or so, there have been a lot of studies trying to predict who's going to do well to specific therapies in non-small cell lung cancer and who's not. And a lot of this is really focused on immune checkpoint inhibitor responses and not as much with who's going to respond to EGFR TKIs. So there have been some expression-based approaches. So this was back when we were using microarrays rather than sequencing-based approaches. But what they found is they did a broad gene expression panel and found that they were able to predict pretty well who's going to actually respond or who's going to have a good outcome versus bad outcome. But this was completely agnostic to treatment and not specific for any specific mutation in lung cancer. people have started to use sequencing-based methods for predicting who's going to do well versus who's not going to do well. So in this one, the mutant TP53 patients are actually doing a little bit better. And then also what has really come up in the last decade has been who's going to respond to immune checkpoint inhibitors in non-small cell lung cancer. And this is really focused on the expression of PD-L1, which is a companion diagnostic marker for the use of checkpoint inhibitors in lung cancer. And what they found is high levels of PD-L1 led to responses for these patients, but also having a high level of immune infiltration led to responses in these patients. And when you combine these two features, you can really try to start to predict who's going to respond to therapy and who's not. But again, this is with immune checkpoint inhibitor treated patients, so this is not EGFR mutant. So we next want to look at, people have also done this with gene expression data in ICI, so T cell gene signature expressions are upregulated in patients who respond versus who don't respond. So we wanted to look at this in TKI-treated non-small cell lung cancer patients, since really this has not been explored to the same depth as immune checkpoint inhibitor-treated patients. So we developed a cohort of 34 patients that were treated with frontline osimertinib, which is a third-generation tyrosine kinase inhibitor used for EGFR mutant lung cancer. And we had 12 patients that had early recurrence, and we had 22 patients that had a late progression. We constructed TMAs from these just for cost savings to be able to do all of these methods. We performed xenium on these with a 280 gene lung panel. We performed imaging mass cytometry on these same samples, and we performed bulk RNA sequencing on a subset of them as well. And with our cohort, we had a median progression-free survival of about 20 months, which is pretty consistent with the literature. So we performed spatial transcriptomics with this 289 gene lung panel. And as you can see, we were able to very clearly differentiate different cell types, such as different immune cell subtypes, as well as epithelial types as well. And because this is a spatial methodology, we're able to actually put these cells back into spatial context rather than just single cell sequencing, where these cells, we have no spatial context of what's interacting with what. It's all a solution. This is actually allowing us to have spatial interaction, be able to look at how the spatial networks are really. So being a spatial conference, I'm not going to dive too deep into what niching is, but we perform neighborhood analysis using a modified cell charter approach. So we use the batch corrected latent space to actually do niching based on the latent space vectors for all of the gene expression data. And what we found is we had 7 different neighborhoods that came up in this. And as you can see in the top here, we have niche 3. And niche 3 was really dominated by epithelial cells, and we call this essentially a cold tumor region, versus other regions which like 0 and 6, which were more stromal rich, and niche 2, which was a little bit more dominated by immune cells, more of like a TLS-like. And just doing a base analysis, when we bin our patients into early and late progression, we found that early progressors actually had higher amounts of cells that were in the niche 3, which is our cold epithelial niche. When we look at that just based on high and low, we found that patients that had high amounts of niche 3, so that very epithelial rich cold tumor, did significantly worse than patients with lower amounts of that specific niche. We want to see what's actually different in this niche that might be driving some of these effects. And what we found is we first performed cell communication analysis using cell chat. And we found that patients with early relapse actually had increased epithelial to epithelial interaction, which is not surprising given the fact that these had increased amounts of a niche that was predominantly epithelial. And then late had increased amounts of immune to interaction. When we looked at specific genes that were pulled up by the cell chat method, we found that enriched in the early group was actually communication between a protein called amphoregulin, which is an EGFR substrate, between epithelial cells and basically an autocrine loop between epithelial cells, where they are producing amphoregulin, and it's actually interacting with the receptor EGFR on those cells. So it's really suggesting that there's this tumor intrinsic mechanism that might be allowing these tumor cells to persist through TKI treatments. And when we just looked at bulk expression, we found that amphoregulin was actually increased in epithelial cells in patients with early recurrence. And we confirmed this with bulk RNA sequencing, as well as GWAS data. And this GWAS data is actually just on total lung cancer. It's not specifically EGFR mutant. And we found that low amphoregulin expression was actually associated with better outcomes. So this matched what we were seeing. So if using our own internal Xenium data, when we actually bin our patients based on high and low amphoregulin expression, we see a pretty striking demarcation between patients who respond to osimertinib and patients who do not respond to osimertinib. We confirmed this actually by doing an in vitro persister assay. So we took two different cell lines that are EGFR mutants. We treated these cells with osimertinib, so it should kill the majority of the cells, which we can see in this top row. And then we also treated these cells with osimertinib with the addition of amphoregulin. What we found is adding amphoregulin to this culture actually allowed for cells to resist the effect of osimertinib and survive in the presence of the TKI. So it's really it's really confirming what we saw in our spatial data, that amphoregulin expression, or amphoregulin is able to cause tumor cells to resist mechanisms of osimertinib. We were able to take those Xenium spatial transcriptomic data and actually prove it in vitro. So when we think about lung cancer, we can really think of things. So with immune checkpoint inhibitors, we really think of the tumor extrinsic. So what is the immune system doing, or the tumor microenvironment? But when people think about EGFR mutant lung cancer, they always really focus on what is the tumor itself doing, because it's a mutant cell line, and not so much what is the immune microenvironment doing. But because we're doing the spatial approach, we're actually able to look for tumor extrinsic factors that might be affecting this. So we've shown there's tumor intrinsic, but now we want to look at extrinsic. So we found that there's actually increased SPP1 macrophage levels that were highly enriched in the cold tumor region. And then they were also increased in early recurrence patients. And then ICAFs, which are really important for cancer progression, were actually decreased in the cold tumor regions and associated with later progression. And we can look at this with the distance. So if we think about the border of the tumor, like the epithelial region, we can look at what the distance is for SPP1 in macrophages and ICAF scores, and we can see that the ICAFs are actually increasing as we go further away from the tumor, whereas SPP1 is highest in the tumor itself and decreases as we go further away. Then using imaging mass cytometry on top of this, we can show that tumor or tertiary lymphoid structures, which contain B cells, T cells, are enriched in patients with late progression. And we confirm that both not only by imaging mass cytometry, but also with the Xenium data. And we also found that FOXP3 T cells are highly enriched in patients with early progression, and they're lower in patients with late progression. So these findings are really suggesting that it's not only a tumor intrinsic mechanism as to how these patients are resisting osimertinib, but it's also a tumor extrinsic. So that immune response is intrinsically required for an efficient anti-tumor response. So to put this all together, we really show that this tumor intrinsic mechanism we're thinking is amphoregulin mediated. So these tumor cells are producing amphoregulin, which is an EGFR ligand, which is allowing them to persist through the osimertinib blockade of the EGFR signaling, but also an adequate immune response with tertiary lymphoid structures, decreased FOXP3 positive Tregs, and also the right M1 macrophage, M2 macrophage milieu is really required for full clearance of these tumors. And when we put some of these biomarkers together, we try to integrate them and pull apart what's actually important, and can we decrease the number of biomarkers required to actually predict who's going to respond and not respond. And this one's quite a few biomarkers, it's about 10, but if we just bin this with the top 50%, lower 50%, we clearly see that patients that have a high score with high amphoregulin, low TLS, do significantly worse than patients that have a low score. And We're currently working on validating this with a more proteomic-based approach using these markers that we found with the spatial transcriptomic studies to actually confirm this on a proteomic level that could be implemented clinically to predict who's going to respond. So in conclusion, spatial transcriptomics really allows us to identify both tumor intrinsic as well as extrinsic mechanisms for the resistance of TKIs in non-small cell lung cancer. And these findings translated to both actionable clinical targets, such as amphoregulin, as well as clinical biomarkers, which I just showed on the last slide. And with this, I have a lot of people to thank. So my mentor, Dr. Kil Merchant, Ho-Chin Lee, who did the majority of the spatial analysis on this, Anton Villamejor, who's helped a lot with the validation work, Dr. Bomick and Manish, who did the in vitro amphoregulin persister assay, as well as Rick Mabane and Simon Nod, who did some of the xenium running, as well as segmentation, and some funding from the Department of Pathology and internal funding, lung cancer research initiative. With that, I'll take any questions.
These in the cohort, you have these like the non-small cell lung cancer treatment patients, right? Or whatever the bulk RNA sequencing you have. But you see one of the top thing hits that you were getting and showing on your slide 16 or 17 was ASCL1, right?
Correct.
And then you are even showing it as the journal score at the last slide in the conclusion. So it is associated with the cell lineage plasticity, right? And the neuroendocrine signature.
Correct, yeah.
So do you think that when you're treating these patients, they are from the non-small cell lung cancer, they are going towards small cell lung cancer, and maybe they may not respond to immunotherapy longer?
That's something we're really digging into. The fraction of cells that actually express ASCL1 is very, very small in all of the patients. So it's a very small subset of cells that are actually expressing ASCL1. And that's something we're digging into a little bit more because that struck us a little bit when we saw the data as well. It was less non-small like, but it's something we're looking into a little bit more. And the thing is, with a lot of these patients, we don't get many post-treatment biopsies. So it's a little hard to see patients that actually progressed. What does it like? Does it become more small cell like? This is all data that we have generated. So the bulk RNA sequencing is on our internal cohort. The only data that was publicly available is the GWAS, which we confirmed the amphoregulin expression with. But otherwise, all of it was produced in-house. Yep.
The primary tumor sites. Am I saying it right?
Yes, so when we generated the TMAs, we took about 4 punches per tumor,
Okay.
and we selected regions that were intratumoral and then also on the periphery.
What I'm
So-
asking, sorry, go ahead.
Go ahead.
What I'm asking is, will it be any different if there is a metastatic site and you were to harvest, tumor cells, if I may say so? Will it be any different, the signature, be it protein-wise or genetic, whatever?
I would assume that it would be quite different, at least in the TME component. So all of
Okay.
these are primary tumor sites. So a lot of EGFR mutant lung cancer is diagnosed with in the metastatic sites, but these are all primary tumor. I would hypothesize that the tumor intrinsic mechanisms might look the same, but the tumor extrinsics might be a little bit different. And that's something that needs exploration just because a lot of these patients do progress with metastatic disease. So it's something we're looking at. And then also with the TMAs, there's always the question of, are you sampling adequately? And all of our validation work that we're doing right now is being done on whole slides. So the actual diagnostic tissue. Thank you.
Okay, next up we have Darius Fugere. He's the SVP of Commercial Sales at Singular Genomics, and he'll be introducing some of the technologies that's used by our next speaker, Zarif Ajir, who will be joining us virtually.
All right. Well, thank you so much. Benefit of this is everyone's nicely caffeinated, so I get to introduce a new spatial technology to a caffeinated room. So I'm Darius. I'm going to be talking about a technology we've been developing called the G4X. I'm with a company called Singular Genomics, and we're really happy to be partnering with the AGCT core here at Cedars Sinai as our third early access site for the system. So G4X, really what's differentiated about the system is it's a very high throughput in situ multi-omics platform. It's about 10 times the imaging area with 40 centimeters squared per run and up to 128 samples that you can fit per run. One of the nice benefits of being very high throughput is we're able to bring down the cost from thousands of dollars per sample to hundreds of dollars per sample. And the hope here is that we're able to push spatial multi-omics into more of the translational and eventually into clinical application as a function of bringing down the cost. We also have subcellular resolution, 0.5 micron, so it's all precise single cell results, and everything's integrated. So we have transcriptomics, proteomics, and H&E on the platform. So our layers of readout that we have from an FFPE section, we do a fluorescent H&E, so eosin's already nicely fluorescent, but we have a fluorescent version of hematoxylin. This is nice. You can visualize the tissue morphology and connect to standard histology. We've got a 15-plex proteomics, and you can add on 2 custom proteins, and then 350-plex transcriptomics. This is a barcode-free approach. We use a novel tissue handling method that's scalable with the platform. So you just section onto basically a hydrogel. You can punch out of that hydrogel onto a tissue transfer array, and then put your slide on top, apply heat and pressure, and then you can nicely fixate all your samples for a run. You can get 128 samples per run, and you can really transfer everything. If it's serial sections in about 30 minutes, if they're all different sections, then that can be about a half-day endeavor. But it's nice. You don't need to use TMAs. You've got a couple different unique storage opportunities, either on the hydrogel itself or on the slide, can be stored for a couple months. Once they're on the slides, there is a wet lab workflow. We're doing rolling circle amplification on padlock probes. So we do that on the transcripts, and then we're actually directly in situ sequencing the foot binding region of those transcript sequences, and we have between 3 and 12 probes per gene. And then a future modality we're working on is direct sequencing, which is gap fill sequencing in between those probes, so we can look at variable sequence information. And then for protein detection, same thing. These are oligoconjugated antibodies, and we're actually imaging a single base. We can do 4 colors at a time over multiple rounds. The flow cells that we have with the tissues on them, and what I'm showing on the top is 10 serial sections in renal cell carcinoma. At the bottom, I think a cohort of different cancer types. We've got an X2 format. This is a 2-lane configuration. You get 10 samples on there, and that's 10 by 10 millimeter samples. So about 10 centimeters squared per flow cell, and you can run 4 flow cells. And the X4 is a 4-lane version where you can fit 32 samples on there, 4.5 by 4.5 millimeters each. And what's nice is actually each lane is fluidically independent, so you can run different panels in different lanes. We provide a integrated single-cell analysis viewer, and then of course, the results coming out of the system. So we do all your single-cell QC metrics. So on the high-performance computing that we have, we do onboard cell segmentation, and then output all the single-cell metrics that you care about. We have open file formats, so you can kind of plug into your preferred downstream tertiary analysis pipeline or open source tool. And then we have this viewer, like I'm showing here, where you can kind of visualize the different channels of proteins, transcripts, cell segmentation mask, and the H&E, and you can toggle between those and visualize that over the tissue. We have a couple different off-the-shelf panels right now: a kidney cancer, lung cancer, colon cancer, and breast cancer panel. The way that these are architected is you've got 100 tissue-specific genes, 150 immune cell genes and 50 stromal genes. Those are shared across the different panels. And then you can add in 25 genes pretty simply. You could also make a custom 100-gene module, or if you want, you can go full 350 genes on the transcript side. And lastly, we've demonstrated good performance of the system. So things that we look for: high, number of unique genes per cell, transcripts per cell, low false discovery rates as we measure by negative control probes and negative control sequences. We have excellent section-to-section reproducibility. And then we have everything that's integrated multi-omics, so you can look at protein RNA correlation at a single cell level, and then have the H&E as well that's already inline and registered. And then what's nice about the reproducibility is this, I think, unlocks the opportunity for serial section 3D reconstruction. So we've demonstrated, one flow cell, 6.2 million cell reconstruction in renal cell carcinoma. That was pretty nice for visualizing and understanding cell neighborhoods and cell niches in X, Y, and Z dimension. So that's kind of the basics of the technology. And we're sitting at a table out there, so feel free to stop by. And otherwise, you'll hear a little bit more here in the next talk. Thank you.
Thank you, Sarah, and the organizers. It's a pleasure to be here at this really wonderful symposium. So I'm going to talk about single-cell spatial biomarkers in lymphoma. I am a physician who takes care of patients with lymphoma. I also have the privilege of running our spatial molecular profiling core, so happy to chat with other folks as our core does provide some of the technologies that I'm going to present for all other kinds of disease processes, not just cancer. But today's talk is going to be focused on what my lab really is interested in, lymphoma. And so for a broad outline, I'm going to describe our work that's been published using in a type of lymphoma called Hodgkin's lymphoma, using really one of the earliest truly multiplex protein platforms, the imaging mass cytometry platform. I'm going to then introduce a sequential IF platform, the Luniform platform, that we have both these instruments in the core that addresses some key limitations of that first generation platform, and then talk to you specifically how we're using metabolic biomarkers as a new direction in a specific clinical application, which is that in responses to T-cell engagers, which are important new cancer therapies. So I will say that I'm not a pathologist, although I do learn a lot from our pathology friends, and pathology has done a lot. And just looking at something so simple as H&E, which is a chemical stain, there's a lot you can tell about tissues. And lymphoma is an interesting example when you look at the tissues, because the different types of B-cell derived lymphomas have very, very different immune responses, and they become a great platform for studying and optimizing different technologies. So on one end, on the left side, is something called Hodgkin's lymphoma. Hodgkin's lymphoma is a B-cell derived tumor, and here's what the Hodgkin's Reed-Sternberg cell looks like. Only about 5% or less of the tumor mass is actually cancer cells. Most of the tumor is actually a normal immune infiltrate. So it's one of the most immune infiltrated tumors. And you can see here's the CD3 stain, which is pretty much stains almost all the cells. And they have very high PD-L1, and not surprisingly, they have the highest response to immune checkpoint inhibitors of any tumor type, with about 70% of patients responding. On the other end, in the middle, you have our diffuse large B-cell lymphoma, primary mediastinal B-cell subtype, activated B-cell subtype, and germinal center B-type. And these are all these different subtypes of this family of large cell lymphomas. And you can see the response to PD-1 therapy goes down as the infiltration goes down. You can see that within the same family of B-cell tumors, you can have completely cold and completely hot tumors. And so our work initially studied Hodgkin's lymphoma because of the richness of the tumor immune microenvironment. And we asked a clinical question that we were able to do primarily because of our great collaborators who had developed this very nice cohort. So an important clinical problem in Hodgkin's lymphoma is when it relapses, you can cure it with something called an autologous transplant. An autologous transplant is, however, a very burdensome, very toxic treatment, puts people in the hospital for 4 weeks just to recover from the treatment. So if we had a way to figure out which patients would benefit from that treatment versus patients who are not going to be benefit, then we could select patients to give that treatment for and spare other patients that side effect. So they, our collaborators at BC Cancer developed a cohort of patients with Hodgkin's lymphoma, total of 95 patients, a subset of whom were cured, they were used as controls, and 73 of them who relapsed and then went on to autologous transplant. And then some of those were cured and some of them relapsed. So we have a cohort with all the clinical information of these patients, and we can now use this cohort to try to understand, are there predictors in the tumor and tumor microenvironment? This is the panel that we use to interrogate. This was done on the imaging mass cytometry platform. Since most of the cells are immune cells, this panel is primarily focused on immune cells. And many of them have been studied in the context of immuno-oncology or immune activation or immune evasion, but here we're just profiling the microenvironment. I will say that this cohort was the basis of a gene expression basis, a nanostring classifier that was actually published now almost 8 years ago, in which they showed that they could predict reasonably well using gene expression profiling who would have risk of relapse and who would not. So this is specifically the survival curves of folks after autologous transplant. You can see that they separate very nicely, but there are still about 25% or so of the patients in the so-called low-risk group who are still relapsing. And there are about 25% of patients in the high-risk group who are being cured by the therapy. So it's good, but not perfect. But this was the state of the art. And there, the limitations, there's lack of spatial information. It's really a bulk type of assay, but useful. And so when we did our spatial proteomic analysis, the key here is that we developed a spatial biomarker. And to our knowledge, this was the first published spatial biomarker which differentiates. Many times we present single-cell spatial data, but we reduce it essentially to something that could have been achieved with flow cytometry or with just singleplex IHC. So what we really wanted is an example of where it really matters that you do the spatial analysis. And so we created for each of the cell types a spatial score. This can be done in many different ways. This is not terribly sophisticated, but what we did is essentially we looked at each of the key cardinal cell types we found, and we looked at the protein expression on those cell types that are related to immune states, and gave it a spatial score. So we have the tumor cells in the center, and then the immune cells and their spatial score enrichment based on their distance. So we're integrating in a single metric the cells of interest and their spatial distance. And then these scores are treated just like any other variable you put in any prediction model that you would do for gene expression. And we are then able to then test numbers of these together to create spatial features, which we can show in this case. For example, take the very top one. It's the expression of CXCR5 on the tumor cells at a certain distance. But if you just take the protein CXCR5, for example, when it's expressed on the B cells in the tumor microenvironment, the non-malignant B cells, it actually has a protective effect. So this is a hazard ratio, and this is 1. So if a feature is to the right of 1, that is predicting to have an unfavorable outcome. And if it's to the left, it's predicting a favorable outcome. So it's not just the expression of CXCR5, it's expression of CXCR5 on a specific cell type at a specific distance that leads to that spatial score. And to summarize that work, basically what you come out with is that we could then concatenate all these different spatial scores that had predictive value and come up with a spatially derived predictive score. And this is just a comparison from that prior publication. And now where our prediction is much better, we can, with almost perfect certainty, find the patients that are going to absolutely not relapse and send those patients. And in the paper, we go on to take the data that we generate on the imaging mass cytometry platform, which is a great discovery tool, probably not the most efficient for clinical use. And in that paper, we then take those key spatial features, reduce them to a more clinically viable test, and show validation on multiple cohorts. And this was published in the Journal of Clinical Oncology as a potential biomarker for the use of outcomes. So with that background, I want to talk about sort of some limits and how we're going to go forward. And of course, IMC is still a great workhorse. It's still probably our busiest instrument in the core, and many people who have started projects or doing still IMC projects, and certainly happy to discuss the current utility of that platform. There are some limitations. The current plexing on that platform is limited to about 40 markers. Now, 40 markers is a lot, but sometimes you want to do more. And the resolution currently is about 1 micron, which means you get true subcellular resolution. But for certain types of tumors where you want to look at nuclear morphology, where that's an important distinction, you're like the way a pathologist does, that's not quite the resolution that you want. Typically, because of the speed of imaging, you want to do a limited field of view. So TMAs are awesome for imaging mass cytometry, but if you want to do whole slide, if you have a specific application where you need broader architectural features in the tumor or the tissue, and we talked about this to our customers all the time, what are you looking at? You're looking at cell interaction? Then any small area analysis is reasonable because cells are small. But if you want to look at larger tissue features that are being made, or looking at different broad structural things that are above the scale of your sampling, then you may need a whole slide methodology. So we designed a panel. This has already been revised. Excuse me. For an 82 marker panel, that's an immunometabolic panel. I'll talk to you a little bit why metabolic. For the Lunaphore system, Lunaphore is a sequential immunofluorescence platform that's automated. I won't go into the technology. The folks from Lunaphore are outside, and they can certainly explain it to you. But basically, it's immunofluorescence, just like you know it, but it's an automated system. It stains from 2 to 4 markers. Then it automatically elutes those and stains again, elutes it and stains again, and just keep going pretty much till your tissue can tolerate it, and different tissues can tolerate different numbers of cycles. And you can thoughtfully build your panel in a way that I believe the company says it's validated to around 40 markers, but we've pushed it to at least 80 and probably beyond. It depends on just the tissue and how you approach it. But let me tell you about why metabolism. So metabolism is, I think, something that we all remember from biochemistry. And I want to say that there are many, many different ways to study metabolism. You're going to hear about some of them today. And I'll just say briefly, each of them is sort of unique in what contribution they give. So we are not measuring metabolites. That's not usually something you get at with an antibody. But we can measure are protein levels of key rate-limiting steps in metabolic processes. So it's sort of like it's not a direct but an indirect measure of metabolism. It can give us a sense of if 2 different tissues are using largely different metabolic pathways, the proteins that control those metabolic processes will be differential. So that's the approach here. And this is just a summary that Joe did a lot of this work, who you already heard from today. So for example, GLUT1 is a receptor for glucose uptake. Some of these other receptors for protein, amino acids, and lipids in the microenvironment. So obviously, if you're highly expressing an amino acid receptor on a cell, there's more likely that you're using amino acids as a fuel source, and then some other rate-limiting steps. So why metabolism in the context of immunity? So this is, again, not work that our group has done, but many groups have done for many years, in that immune activation is tightly controlled. Besides the signaling molecules and the receptors that we look at by flow, is controlled metabolically, and metabolic processes are associated with T cell states, specifically activation, exhaustion, memory, are deeply controlled and programmed into the metabolic states. So for example, naive T cells here are primarily doing OXPHOS and fatty acid oxidation. When they get activated, there's a burst of glycolysis. And then if they clear the antigen and enter that memory state again, glycolysis is a relatively minor fraction, but exhausted T cells and the tumor microenvironment are thought to depend a lot heavily on glycolysis. There's probably complicated reasons, including the availability of substrates. But the key here is that there's quite a distinct metabolic state. So what we did is developed a Lunaphore panel for some of those metabolic enzymes. I've showed you some of them here just as a validation. So this, to orient you, is a tonsil. And tonsils are really useful because we validate all of our immune markers on tonsils, but they also have a very classic structure. So those who aren't familiar, tonsils have immune cells, B cells and T cells primarily, and the B cells aggregate in what's called follicles and then germinal centers. And you can see the CD20 staining here. And then the T cells surround it. So CD3 here is in this ribbon here around this follicle where CD20 stains. And then within the follicle, there's the germinal center, which is, you can see even on CD20, there's like an inner part here where that's a PD1 staining, and the different markers that mark these different germinal centers. And this is sort of our standard immune panel. You can see some only mark the sort of the rim of the B cells, some mark the inner part. But if you then look at some of these metabolic enzymes, and now again, pretend you don't know anything about metabolism, you can see that it's recapitulating those patterns. So there's a very tight association between these different lineage states, which are well established, and the different metabolic enzymes. And what's really nice is you can use that spatial information to sort of validate, does this make sense? Are these biologically fitting what you know about the known biology? And spatial is really useful. Forget all the fancy analysis, just looking at the image, and this is pathologists have known this forever, is that tells you a lot about what's happening in those tissues. You can see these metabolic enzymes have a very nice spatial correlation. I will just make one mention about the nuclear morphology. So when we do lymphoma work in particular, lymphoma is a very, very cellularly dense tissue, and nuclear morphology matters. And you can actually see, this is a Luna-4 image with the DAPI nuclear stain, that these two, this is CD20, so these are all B cells, and this is right at that edge where one part of the follicle is entering the germinal center where the cells are a little bit larger, and you can really see the difference in those nuclear morphologies in the cell shapes. And interestingly enough, ASCT2, which is one of those metabolic enzymes, is really differential. That really turns on when you change from one state or not. So now we've correlated both the metabolic enzymes, the morphology in these different B cell subsets. Just for folks who are not familiar with the Luna-4 platform, so here's an example of a lymph node we did. This is a lymph node in a patient with follicular lymphoma, and I'll tell you about why follicular is interesting in just a moment. But here's the B cell marker CD20, this is CD10, and this is CD3 in red marking the T cells. This is from the clinical sample. This is that same sample. It looks a little different in shape. I've put a star for orientation just because we've cut a little bit deeper into the block. And when just as sort of validation, the cyclical immunofluorescence can recapitulate many of the same features. Of course, it's single cell, so you can extract single cell data, do your UMAP projection of the different populations, and then, of course, do the projection of those populations back onto the space. So this is the UMAP spatial clustering, and now a projection of those UMAP clusters back into that space. So for example, red are endothelial cells, and you can see that they are in these spaces in between where endothelial cells should be. And then some of these T cells are also following some of those same patterns, with the tumor being the B cells in those sort of follicular spaces, why it's called follicular lymphoma. And so we've applied this to a project where we found IMC to be a little bit limiting for us, and that is this challenge of follicular lymphoma transformation. So follicular lymphoma is a low-grade lymphoma, and patients can live for many years. But when it transforms, it can turn into an aggressive lymphoma. Transformation is not heralded by any singular genetic change, although there are changes, anything we can do on gene expression. And so it remains to this day a diagnosis that a pathologist has to look at the tissue and see, do you make little follicles, or do you lose the follicular structure and you have a diffuse pattern? So it is inherently a spatial diagnosis, and it is one that is done on whole sections of tissue because just to give you a sense of scale, this is pretty much the size of a TMA punch. You can't do this on a TMA, because if you try to punch a TMA, you don't know if you're here or here in this part. So you really need to be able to look at whole slides to understand transformation. So we thought this was a perfect use case to try to say, when do you really need whole slide imaging? Now, if you come to my core, I'll ask you, do you really need whole slide imaging? Because it's a different workflow, the data files are different. So but if you do, then this is a great platform, and this is a clear example. This is a survival curve of patients who don't transform, they live, and this is in years, so this is 1 decade, 2 decades. They do quite well, but if you transform, their prognosis is very poor. So we did our immunometabolic characterization we created a bunch of samples that have different types of lymphoma from low grade to high grade. I'll skip over this. This was actually done on TMAs, extra large TMAs, but only to validate the markers. The study itself we have to do, of course, on large whole slides. We showed that when you look at the immunometabolic modules, here are all the metabolic enzymes, that when you cluster them, they cluster with different states, as you might expect. For example, high PD-1 expression is associated with activation, so that's this group up here. And medium PD-1 expression is associated with exhaustion. So to everyone who just thinks PD-1 equals exhaustion, those who are immunologists know that it's much more sophisticated, complicated than that. The levels of PD-1 matter, so you want to be able to read out not just on and off, but actually read them out quantitatively. And you can see that there's the metabolic clustering as well. You can, and metabolic clustering can be really sophisticated. You can say, oh, they're using fatty acid metabolism, this cell's using glycolysis. But it turns out that cells, when they activate, they kind of try to fire on all cylinders. So if you just use a very simple a sort of metabolic score, you can summarize it. And we did that for this analysis. And so this is the single cell metabolic expression, MEP, which is the sort of summary. And you can see as cells progress, the metabolic state of the T cells is changing. And we can look at how they move in the different clusters. Some of this is unpublished. So I will say that we did a lot of work to try to validate this with a different metabolic, or Joe and the team that he leads did a lot of work to validate this. So I'll just mention very briefly, there is a way using flow cytometry to measure single cell metabolic activity. And this is the Zenith assay. And this uses another surrogate. It's actually measuring puromycin incorporation, which is directly related to protein synthesis, and protein synthesis is directly related to ATP concentration. So here's ATP concentration, and here's puromycin. This is the original paper published in Cell Metabolism. So what we did is we tried to validate what we were finding in this system. So this uses fresh lymphoma specimens, and these were processed for clinical utility, so they're in the core with FFPE the way any tissue would be in the archive. But we had a sample that was fresh, so we could actually measure directly their metabolic activity. Because this is all, like I said, we're measuring things indirectly. So before we sort of say we're actually measuring metabolism, we want to do some kind of actual functional validation. So we split the samples, and we made these patient-derived lymphoma organoids, where we could culture the cells and then do the Zenith assay to look at the metabolic states. And we could, in culture, perturb the cultures with different treatments. And I'm going to tell you about that treatment in a moment. And then once we measured the metabolic states, and we really know now I'm actually directly measuring T cell metabolism, we then tried to go back to the FFPE specimens and say, oh, the patients that had high or low metabolism and had high or low response, what was the state measured in the FFPE clinically processed specimen? Because then that would convince us that we could actually go back and measure this on the clinical specimens, which are much more abundant than these ones that we get fresh, which are hard to procure on a regular basis. So this is where the T cell engagers come in. So I'll just say very briefly, T cell engagers are a really exciting new treatment for all cancers. And basically what it does, it's an antibody. Antibodies have two sites for binding. And so one site binds T cells, and the other side binds the antigen you want, in this case CD20, and it causes the T cells and the target cells to come together, which triggers a T cell response, and the T cells then activate and kill the target tumor cells. And what's really nice of our patient-derived organoid model is that it recapitulates that. So that's already been published. You can put the bispecific antibody in the culture, and there's T cells and tumor cells, and they will activate and they will kill the tumor cells in the culture model. So here we used a drug called epcoritamab, and you can see this is the live lymphoma cells on the y-axis, and you add different doses of epcoritamab, and you can see you get lysis of the tumor cells. Some of them are resistant. So this patient, this gold, can't read the number here, but the one in gold had no lysis. So we had some sensitive, some resistant, which is great because that's what happens clinically. So then we looked, again, using the Zenith assay in our cultures, what was the basal metabolic state of those T cells before we give the epcoritamab? And we call these ones at the top the non-responders, and these are responders. And you can see just at baseline, the metabolic state was different. in responders and non-responders using the Zenith assay. And even more striking is that when you gave the epcoritamab, the patients who were responders, the colors match here, had the highest activation of metabolism in the T cells. So basal state was different, and when you treated, you had activation. So that to us was very convincing that now the metabolic state is correlated with the response. So then we went back to the tissues and measured the metabolic state, which would represent this basal state. And you can look at here just two excuse me, cases. In gold matches this resistant case, and in red matches the sensitive case. And you can see the basal metabolic state as measured by our single cell metabolic assay on Lunaphore matches the difference you're seeing in the actual culture model. So we're really excited to think about this as a potential biomarker for predicting response to bispecifics, and hoping to do that. That work is ongoing as we get patients actually treated with bispecifics and even from the company to analyze on the new platform. I will just mention one last use case, and this is a really cool project I've done with collaborators in the RMI, where we used iPSC-derived macrophages, which is a really cool platform for potentially using a novel cell therapy. And this was work done in collaboration with Clive Svensson and his group. And basically, at just a very high level, you can see that we know that these iPSC-derived macrophages are monocyte-like, and they have one metabolic profile. And when they get injected into mice that bear tumor, they can enter the tumor and they can change their state and their immunophenotype. And they also change, not surprisingly, their metabolic state. So you can see that cells at day one have a certain pattern of of metabolic enzyme use, and then they switch over. They become, they start to express ASCT2, which is sort of classic for this M2 macrophage phenotype, and turn off some of these other metabolic states. So, and we can correlate that spatially. So you have these peripheral areas of the tumor where the macrophages initially settle, and then here's the macrophage in this necrotic center of the tumor and has that different physiologic state. So allowing us to now correlate spatial information with cellular state. And what we're hoping to add, and we're going to hear about what some new technologies can do, is actually now directly measuring metabolites in tissues like this that we've collected. Now we can put all of this together as a spatial dimension, the cellular dimension, cellular state, and the metabolic state and metabolic substrates that are available in that context. I think I'm out of time, so I thank everyone listening and our collaborators, and appreciate the opportunity.
Take a moment for a few questions. If you have one, raise your hand, and please wait for the microphone.
Fresh samples and then going back into the FFP section, then just trying to correlate it. Usually, like in the metabolic state, it can vary from cell to cell, right? How difficult it was, or I'm happy that you could correlate it positively with the FFP sections along with the frozen. But how difficult it would be, like, because I think the metabolism state, once the fresh samples you have, and when you have the FFP samples, it's quite different, unless you try to get a subset and find a positive correlation. What is the threshold you could?
Even the manipulation of freezing, thawing, culturing, hypoxia, normoxia, what you put in the media, all of that will affect metabolic state, right?
Yeah.
And so that's all noise and background you got to think about. Our supposition here was that the some of the changes that had happened to those T cells, particularly in the microenvironment, were sort of somewhat stable and related, because the challenge with immunotherapy, right, is that when your T cells are in an inactivated state, they kind of stuck that way. So we think that's probably why we can still see that difference. Otherwise, you're absolutely right. Normally, immune cells in a healthy system are highly plastic and dynamic, and so you'd have to think about controlling for that and looking for that, which is why it was so important to see if we could validate. Ultimately, in the biomarker world, ultimately, I stayed away. I mean, I stayed away from specific claims of biology. I didn't tell you this cell is using this enzyme or this, because that requires a rigor that I've not presented to you and that you have to do. For a biomarker person, it's important, but it's not that important, right? We just want to know, can I separate these groups? Can I utilize the biomarker? Very few biomarkers do we really truly understand their biology they're reflecting. But you're right, to do it thoroughly, you need to do some of those studies to control for those variables, absolutely. So each of the metrics between the cell type and the protein and the distance is a feature, and that final differentiator was, I think there's 5 features that are incorporated into that spatial score. So they took the multiple features which had both positive and negative predictive value, and they selected the highest ones to be a little bit more concise so they could develop a clinical test based on it. But I think there's 4 or 5, I can't remember. I think there's maybe 4. That are in the final score.
Okay, I think that's it.
And so I know we all are still excited to hear the talk from Zarif that we had to skip, and she will still be giving her presentation in the final time slot immediately before lunch. But for now, we have our own Sarah Parker to continue our talks on single-cell and spatial proteomics. She'll be talking about single-cell proteomic analysis of the aneurysmal aorta.
All right, while AJ gets my slides hopefully projecting to the Zoom. Yeah, so we've talked a lot this morning so far about single-cell transcriptomics. We've talked about single-cell and spatial proteomics from the perspective of antibodies and their use to target specific proteins. So now we're going to switch gears a bit and start talking about more of a discovery-based proteomic profiling approach using mass spectrometry, which is the forte of my lab and also the core of which I'm the co-director here at Cedars Sinai, the proteomics and metabolomics core. And because I'm also a cardiovascular physiologist, I focus on the heart and the blood vessels. So today I'll be talking about using aorta as a context with which we can better understand and develop the technologies for single-cell proteomics. So to get everybody onto the page of sort of some of the technological challenges that we face in applying these single-cell technologies to the proteome, I'm going to start off describing really what launched mass spectrometry into the realm of capability for single individual cell profiling, and that was this what we call SCOPE MS, or single-cell proteomics by mass spectrometry approach. So in its earliest implementations, what we were able to do was to kind of bypass some of the sensitivity limitations of our mass spectrometers by using multiplexing to boost the signal of any individual peptide, combining multiple cells together to sort of boost that signal. And we used little small molecule barcodes that allowed us to then detect and sort of parse out which sample each cell which cell each peptide came from in the ultimate data mix. So how this works essentially is you have your individual cells, you dispense them into a well of, say, a 384 well plate. You can lyse them, digest them, and then you would apply this small barcode label. And we can then take each of the individual cells labeled peptides, we can combine them back together for this slightly amplified sample that we can run on a mass spec. And even 5 years ago, taking 8 to 16 individual cells proteomes and putting them on the mass specs we had 5 years ago still really wasn't enough of a signal boost to get all that much proteome detection from the mass specs. So we started adding these carrier channels, which was a larger amount of bulk lysate that would sort of boost that initial peptide signal, enabling that ultimate ability to then parse out the peptides that were identified from each individual cell. So this worked really, really great off the bat and really enabled a lot of the early single cell profiling experiments. Almost all of these to date, even now, have been performed on maybe biologically simplified systems. So cells in culture, maybe slight, cell organoid systems. But we have some question about how well this approach is really ever going to work for truly heterogeneous tissue samples. Because if you kind of think about the principle, you're needing this big boost in signal strength that's brought about by multiple versions of that peptide coming in from multiple different cells or present and detectable in the carrier population. So when you start to get down to very rare cell types with unique proteomes, are you ever really going to be able to measure a peptide from one of those rare cells in this mix of the other samples? So it really is some of the same limiting factors that hit us in bulk, still hitting us with this particular approach to single cell proteomics. So because we're really interested in translational applications, sorry, I said everything that this animation just showed you and emphasized. Because our lab is really interested in translational applications, we started thinking about developing the technology to really apply to one cell at a time's worth of proteomic data. And as we started to think about that, the technology actually caught up to where we were hopeful of being able to get. And Bruker came out with the very first mass spectrometer capable of detecting protein levels at the really individual cell concentrations. And one of the challenges with the methodology still in place for us, though, is that to get these individual cell proteomes, it was still taking somewhere in the order of 30 to 60 minutes per cell to acquire the data. So, what's sort of been our first, I would say, major contribution to the field is a creative use of our liquid chromatography separation upstream of the mass spectrometry that allowed us to get our acquisition time down to a clean 15-minute all-in proteome. So this allows us to get to about 96 cells profiled per day. Just suffice it to say the performance goes really well, and because we are a cardiovascular lab, we started to test whether this technology is going to apply to sort of what we consider real-life cell types that we're going to want to profile. And so we showed that we can effectively profile the proteomes of human cardiomyocytes isolated from transplant patients, mouse cardiomyocytes from different models, and then we took a mouse aorta and did a whole cell dissociation, and we're able to see many of the sort of known cell types that parse out from the aorta. So this was really exciting. We knew that we are now in business for being able to do really true individual cell proteome profiling of properly heterogeneous sort of biologically real life specimen. And so pretty quickly on the heels of this paper, started envisioning how I could leverage this and apply it to some of the diseases that we're interested in studying in my lab. And the main condition that we study is aortic aneurysm. And for our purposes, we're studying aneurysm within the context of a genetic disorder or hereditary forms of aneurysm. And specifically, I study Marfan syndrome because it's one of the longest studied, sort of well-known causes of hereditary aortic aneurysm. So just to not belabor the biology too much, but aneurysms are dysregulated growth of the aorta. They predispose the tissue to dissections and ruptures. You can imagine if the main conduit moving blood from your heart to the rest of your body splits or ruptures, this is a very bad thing for you. We've known the gene that causes aneurysms in Marfan syndrome for 30 plus years. It's an extracellular matrix protein called fibrillin-1. That knowledge really has not yet led to an effective therapy besides prophylactic surgery where you basically cut the aorta, cut out the aneurysmal sections and sew the tissue back together. So we'd really like to try to start to do better by these patients. And I think in order to do that, we need to understand the mechanism much more clearly. And you can imagine with an extracellular matrix-bound mutation, almost all of the cells throughout the aorta here are going to be interacting with this mutated protein. And we have a relatively simple but still somewhat complex cellular milieu in the aorta. We have smooth muscle cells, endothelial cells, fibroblasts, so sort of your classical cells of the three major blood vessel layers. But we also have pretty substantial immune involvement and also involvement of nerve cells and adipose cells. So a good complex palette for us to start exploring the single cell proteome, and importantly, how that single cell proteome reacts and responds to the presence of these fibrillin aneurysm causing mutations. So here's the overall experimental design that we applied. Ended up needing to pool cells from 2 sort of wild type mouse aortic roots and 2 aneurysmal Marfan aortic roots. That gave us enough cells to comfortably perform our sorting into wells of a 384 well plate. We do a 1-pot sample preparation where we really minimize the steps of any kind of washing or transfer of samples, because every time a protein sample touches plastic, a little bit of that protein is left behind and you lose signal. Through this, at the end of the day, we profiled 3,475 cells, averaging about 500 proteins per cell across the experimental workflow. So we sort of did the brute force work of generating all these proteomes, and we're left with 3,400 data files that now needed to be analyzed and processed and made sense of. And so that's where our collaboration with my colleague Jesse Meyer came in, who you're going to hear from after my talk. But we put it through its paces with typical protein detection analysis. And then we really didn't know how single cell proteome data were going to fare within typical single cell analytical pipelines. We tried a few different ones and ended up with the ScanPy pipeline that Jesse has since built into a really excellent online workflow with a nice graphical user interface. So if you want to follow this QR code, you can see more about the pipeline that Jesse's built for single cell proteomic analysis. But suffice it to say, we eventually met with success, and I sort of met my biological sniff test. And we were able to identify all of the major cell types most of the major cell types that we know are present within the aorta. So, and in this particular case, they also parsed out to look a lot like the continent of Africa. So this is something fun about single cell and the Rorschach UMAPs. But so using just sort of a standard profile of marker proteins that we could use to identify individual cells and assign them back to their Leiden clusters with which they co-segregate, we see a large population of different smooth muscle cells up here, all in the upper left portion of this UMAP, all expressing varying levels of classic smooth muscle cell markers. And then we see cells that clearly express markers of fibroblasts, endothelial cells, adipocytes, and macrophages. So achieved a pretty good comprehensive profile of the mouse aortic proteome. And importantly, then we were very excited to dig into what are the molecular and cellular differences in these different cell types between our 2 genotypes. And for the interest of time, I'm not going to go super deep into this, but we did find some really intriguing differences. We also were able to include mice of both male and female sex, and we know that sex is actually a modifying factor in aneurysm severity. Males tend to have aneurysms about 2 times more severe than females on average. Interestingly, we see some sort of overall differences in the composition of endothelial cells and fibroblasts between males and females. Males tend to have more of these 2 cell types. And then we do see changes in sort of all of these complex subtypes, subphenotypes within the smooth muscle cell umbrella, finding a couple that are poignantly either more prominent or less prominent in the Marfan phenotype. So presence of that fibrillin mutation is participating in driving smooth muscle cell phenotypes in different directions. And that's something that's not exactly novel to the field. We know that this happens, and it happens in lots of different vascular disease states. And these data are going to allow us now to dig deeper into what the mutation is really doing to some of these different smooth muscle cell types, and how those transitions maybe could be prevented therapeutically. And then another really interesting question we can ask, especially because we have multiple biological replicates under each of our experimental groups here, is start to look at proteome, differential protein expression within a particular cell type between the genotypes or between the sexes. So for instance, we look at endothelial cells and we compare all of the proteins in of an endothelial cell from a Marfan mouse to an endothelial cell from a wild-type mouse. And we were, I think, relatively underpowered still, even with an N of 3 per group, to get a whole lot of information, a whole lot of significance among our differential proteins. But one thing that stuck out to us is that the endothelial cells did seem to show some of the largest degree of differential expression among their proteins, interestingly, by genotype. Interestingly, when we look at differential expression by sex, we actually see that fibroblasts show very sex-dependent protein expression, which was also something interesting that we have not yet followed up on. But we were really curious to know, more about what some of these highly dysregulated endothelial cell proteins might be. And with only 20, you can't really do a pathway analysis, but we just looked at them one by one, and it started to really pop out that a lot of these related to either sort of endothelial cell adhesion proteins like PCAM, NO-related production proteins like nitric oxide synthase. And then we see these are tended to be downregulated in the Marfan endothelial cells relative to wild type. Conversely, when we look at contractile proteins, we see some upregulation of sort of what are otherwise classical smooth muscle or mesenchymal cell markers like myosin heavy chain 11 and tropomyosin 2. So this started to smell a lot like endothelial to mesenchymal transition to us. This is a phenomenon that's known to occur in cancers. It also, it results in an increase in permeability of the endothelial layer, allowing in more easily infiltration of immune cells, fluid barrier disruption. And so this is a biological event that we're really interested in. And there's some evidence to suggest that this has been found in other forms of aneurysm, but this would be the first evidence that endothelial to mesenchymal transition happens in Marfan syndrome. We wanted to start to validate this observation, and so we looked to the literature for other data sets, and we were able to find a single-cell transcriptomic data set that was performed on the same strain of Marfan mice that we used in this study, published in this paper. And what we found when we dug into their RNA data and their single-cell endothelial cell transcriptomic data, they find two populations of endothelial cells, and we see some trends that kind of support our observation from protein, but they're certainly not as strong. And we also see that for these overall endothelial markers, this endothelial cell type 2 shows lower overall level compared to the endothelial cell type 1. And that cell type also has slightly higher expression or differential expression of some of these smooth muscle and mesenchymal markers. So, partially supporting our hypothesis, we see that the Marfan male mice have the largest population of this lower PCAM and NOS3 expressing subtype in these transcriptomically defined cells. And we also looked at two of the transcriptional regulators of endothelial to mesenchymal transition, SNAIL1 and ZEB1. Not really detectable at the proteome level, but we did see them in this RNA-seq data set, and we see that those show a very clear pattern of elevation in the Marfan samples. So overall, kind of corroborating our initial observation from the proteomic data that was really smacking us a little bit more in the face, that endothelial to mesenchymal transition might be happening in these mice as one of the major ways that these cell types are responding to the fibrillin mutation. So this is something we're working on following up with spatial transcriptomic or spatial proteomic data to see if we can really confirm this in mice and then see if we can actually see it in some human samples. But now that we have this RNA data set in our hand, we also were very curious to ask the question, how do the transcriptomically defined cells compare to our proteomically defined cells? And so we took the transcriptomic data and just passed it through the same ScanPy pipeline. It had previously been analyzed using Seurat, so to avoid any major differences in the analytical approaches, I just ran it through from raw through our ScanPy workflow, and essentially came up with many of the same cell types. This particular data set has an additional population of valve cells because of the way they isolated their roots, but we see a lot of the same cell types, multiple smooth muscle subtypes, et cetera. So we wanted to know how well the shared genes and proteins between these two data sets sort of overlap onto each other across our different clusters. Oh, sorry. We are going to look at that, but first I'm going to tell you about specifically the similarities and differences in our known disease-causing gene fibrillin. So here we have, these are just the overall UMAP plots of our protein and our RNA. We see fibrillin here is really seen mostly in the smooth muscle population at the protein level, whereas we see it almost ubiquitously, at least in the cells of the vascular media in the transcriptomic data set. And I think this probably has to do with the fact that at the protein level, if fibrillin is seen, it's probably because it's coming down bound by the cell, right? The cell is recognizing it in the matrix, and it's sort of, as we do our isolation, it might just be coming along for the ride as we pull it out of the tissue. Whereas obviously at the transcript level, it's indicating that that cell is making gene that will ultimately get secreted and made into this protein. And then another really standout factor that we saw, though, is that, and this may have to do with the genotype-phenotype association with this particular mutation that caught in this mouse model, we see that there really is no difference between a Marfan cell and a wild-type cell in terms of the amount of fibrillin they're producing at the transcript level. But when we look at the protein level, we see much more, almost 2 times more protein in a wild-type mouse relative to a Marfan mouse. Now, these are heterozygous mice for this mutation, so that really indicates that something's going wrong with the production and secretion of the fibrillin, potentially, and that we're detecting that at the protein level and not at the transcript level. So finally, now we wanted to know how well does this transcriptomic data set compare to some of these proteomic data set profiles that we're doing. And there's 2 different ways that we could find out there in the various tools for data analysis. The first we looked at was within Surat. You can take your ScanPy file, you can modify it, you can open it up in Surat, and Surat has this compare marker kind of function that it allows you to do. And basically, when you project a marker of a cell based on the protein onto an RNA data set, you can identify the major cell types present, but you really don't get any of the subtleties that we're seeing in subphenotypes, like the 7 smooth muscle cell phenotypes or the 2 endothelial cell phenotypes. So you get sort of the big picture, but you kind of lose some of the grainier small picture, and that's really where a lot of the meat and the detail is. That was true when we do just sort of a gross cross comparison, and it's also true when we actually look at an integrated data set using the Liger tool, where now we take common genes, we merge everything together, and we analyze it as one unit through the pipeline. And even then, we really only get high-level match, and you can see this in the whole data set. This is a pretty complicated Sankey plot, but basically you're seeing cells from the initial clustering that was done of the data set independently for either RNA on the left or protein on the right. And in the middle are the integrated clusters that were detected by Liger. And you can see, if you spend enough time with this figure, you will see that smooth muscle cells map back to smooth muscle cells, but there's no real clear pattern of like a cluster of smooth muscle to another cluster. They're all kind of disseminated across the interior Liger clusters. So you kind of, again, you lose that key subtlety that you have with either data set individually. To get even more clarity on this, we really zoomed in on just the smooth muscle cells, since we think these are major players in aneurysm biology. Even when we get down to the level of the individual smooth muscle cells, again, when we analyze them by Liger, we get 7 unique clusters of integrated transcriptomic proteomic cells, but you just really don't see any clear pattern of how the initially transcriptomically or proteomically defined cells map back to these integrated clusters. So the different analyte types are differentially defining the similarities between the cells. And when you combine them together, you get a sort of completely third different picture of who these cells molecularly are across the grouping. Of course, these different Liger integrated clusters do show differences by genotype of the mouse that donated the cells. And when we look into the markers for these, some of them make really good sense biologically, and they weren't picked out by either analysis alone. And these include the angiotensin converting enzyme, S100A4, and tropomyosin 4, which are all markers of dysregulated smooth muscle cells, and were all among the populations that were elevated in the Marfan. So there might be something here. We're following up on it with validation studies, and we're really excited to see what these kinds of integrated experiments can divulge for us, but important to say that a transcriptomic data set does not necessarily tell you the same thing that a proteomic data set does. And that's really the crux of my talk. So single cell facilitates analysis of complex tissues. We are detecting new and potentially biologically exciting information that differs from what you might learn from the transcriptomic alone. So thank you.
Sarah, we'll take a couple of questions. Please raise your hand and wait for a microphone.
Like it is adding another layer of complexity to your analysis in your system. So how do you address that? That's the thing, like, because you were trying to show the transition for some of the RNA molecules when you just started with the proteome, and then when you came to the RNA, it changed, and now overlapping even, it's not happening. So how do you address that, and how you will be profiling it downstream?
Yeah, I mean, it's a great question. We ask ourselves that as well. I think we go back to the central dogma and then how that informs what we need to know about our samples, right? So if you need to know, the transcriptional regulators of your system, you can look at transcriptomics because you want to know what genes are actively being transcribed and what that means about the activation state of the cell. If you want to know the functional state of the cell, who it's primed to be right now, that's the proteome, right? If you want to know how well transcriptomics is matching translation, you can look at the integrated data. I mean, that's right now is the best answer I can give. And as a biologist trying to solve a translational problem, I'm going to say whatever gives me the most practical solution that leads me to a biomarker or a therapy. But yeah, it's a good question.
So one of the pathways that you were mentioning is EMT, right? Transcriptionally, you can look at it as EMT markers, so ZEB1, SNAIL. Downstream, what are the downstream targets of that? Meaning it could be TGF, I don't know, it eludes my mind right now. One of the things that I worked with is C-terminal binding protein, which is the downstream of that. Are we able to unravel the question that he's asking by looking at those proteins that are potentially the EMT downstream markers and use that as a quote unquote biomarker or therapeutic target?
Yeah, I think that's a really great question and an idea. So SNAIL and ZEB1 are sensitive to TGF-beta, right? And then downstream from those two transcription factors are the mesenchymal markers like myosin heavy chain, ACTA2, and et cetera. We know that TGF-beta is disrupted by fibrillin mutation. I kind of left that out. But so, starting to kind of piece those elements together into a pathway story is one way you could use the two different data types. So thanks. All right, we should probably move on to Jesse.
All right. Good afternoon, everyone. Thank you all for coming today. I'm excited to tell you about some of the recent work from my group related to single muscle fiber proteomics and sarcopenia. In my lab, we're committed to driving healthier aging with a focus on improving and preventing sarcopenia and Alzheimer's disease by better understanding metabolism. To achieve this, we generate knowledge from data through visualizations, statistical analyses, and machine learning. And our favorite data to use for this pipeline is proteomics data, but we also explore the synergies of multi-omics and single-cell omics and electronic health records. So today I'll share with you a project that spans all three of these boxes where we ask the question, what causes sarcopenia? But before we dive into the data, I'll give you a quick overview of what we're covering today, starting with some background about muscle aging and why proteomics can give us a unique lens into that process. Then we'll talk about something we encountered unexpectedly, protein isoform confusion with aging. And that leads us to something else surprising, the possibility of a novel type of muscle fiber. So with this roadmap in mind, let's get started with the background. Sarcopenia is the loss of muscle mass and strength with aging. As you can see on these plots, on average in our 50s, we start losing our strength and power until we're left with about half of our strength in our 80s. Sarcopenia can be slowed with resistance training, but it cannot be stopped. It is inevitable. It will happen to everyone that ages, and for that reason, we should all care about it. This loss of strength leads to a lack of mobility, which then leads to increased morbidity rates. So therefore, preventing this loss of strength would increase your healthy lifespan. Your muscles are made up of two different types of muscle fibers. There's fast fibers that are responsible for high power output, for example, if you're sprinting or carrying something heavy. And you also have slow fibers, which are responsible for low power, such as posture and standing. This heterogeneity means that the molecular approaches that homogenize muscle will miss the fiber type specific detail. And we know that detail is important because several studies have suggested that sarcopenia is a disease of only the fast fibers. This is data from my collaborator, Chris Sundberg at Marquette University. And in his lab, they dissect out single muscle fibers from human adult volunteers, and then they can measure the power from those single fibers. And I think this is incredible because these fibers are tiny. They have an average diameter of 50 microns. But this data is very important because, as you can see, it shows us that over many fibers from many people, there is a dramatic loss of power with aging. So the aged people are in old and the young people are in blue. And you can see that this happens in both males and females, and it's a big problem. I'm clicked on the wrong thing. So because we want to understand only the fast fibers and they seem to be functionally affected, we want to look at molecular changes in those fiber subtypes to determine druggable molecular mechanisms. And in 2017, a seminal paper was published by Marta Murgia and others in the Matthias Mann lab, where they described for the first time human single muscle fiber proteomics. And in total with this paper, they measured 152 single fiber proteomes from 4 young and 4 old males. They detected only 6 statistically significant changes, and this paper has been highly impactful, and it's been cited over 300 times since it was published 8 years ago. Unfortunately, the methods that they used for sample preparation were very laborious, and the data collection was also very slow. And this means that we need new methods to allow population scale studies of single muscle fiber proteomics. Shortly after I moved my group to Cedars Sinai, we started applying the single cell sample preparation methods that Sarah mentioned to our single muscle fiber samples. And in 2023, we published this technical paper describing the workflow applied to 53 fibers from 1 young and 1 aged female. The way this works is our collaborator dissects out those single fibers. They put them each into a single well of a 384 well plate, and then they ship that plate to us, and then we use those methods for 1 pot single cell sample preparation. And this is very important because it allows us to prepare samples exponentially faster than those prior studies we mentioned that use the end-stage tip protocol, which is much slower and requires a lot more manual effort. Further, by leveraging that dual trap single column high throughput LC approach that Sarah mentioned, we're able to collect our data from 96 fibers per day, which means that our method is nearly 10 times higher throughput with regard to data collection. So we're starting to scale to a level where we can measure lots of fibers from lots of people. But before we started this project, we weren't sure if we could directly use the proteomic data to differentiate the fiber types, or if we needed to do a parallel Western blot to measure the myosin-heavy isoforms. So this is one of the first things we looked at, and this plot is showing you those whole proteome profiles from each fiber compressed into 2 dimensions with UMAP. And each one of these points is one of the 53 fiber proteomes, and you can see that we find 2 clusters with the unsupervised Leiden algorithm. And when we overlay the major myosin-heavy isoforms that delineate those fiber types, we see that the group on the left is correlated with the myosin-heavy 7 amount, and that is the slow twitch isoform. And the group on the right is correlated with the amount of myosin-heavy 1 and 2, and those are the fast twitch isoforms. So overall, this tells us that we can differentiate fiber type just based on the proteomic data using these canonical markers of fast and slow fibers. Now I'll describe an entirely unpublished project where we used this technique and designed what was at the time the largest single fiber study ever implemented, and it was the first in females. So in this case, we collected samples from 5 young females and 5 aged females. This time we had an average of 100 single fibers from each person. We performed those 384 well plate-based sample preparations, and then we analyzed these samples with the timsTOF Pro 2 with 15 minutes per injection using capillary LC. And the data came out absolutely beautiful. This time we lost some of the old fibers, so we had slightly more fibers from young participants in the final data set. and the median number of proteins per fiber was higher than we had seen in that original study with around 700 proteins per fiber. There are some differences you can see in the depth that we obtained from the old and young fibers, but in the end, we're left with over 1,000 fibers where we had quantified at least 200 proteins. So this is a great data set that we can use to start investigating sarcopenia. All right, now that we've set the stage and walked through that background and experimental design, let's dive into what we found, starting with the first major discovery, which is something we're calling protein isoform confusion with aging. We can determine the fast and slow fiber types based on the proportion of myosin-heavy isoforms in each sample using this simple equation here. We call a fiber pure if the fraction is over 80% of the signal in the myosin-heavy 2 or 7 channel. And here's what this actually looks like for all of the young fibers. So remember, there's about 570 fibers here. Each one of them is a point along the x-axis. And the y values give you the proportion of each of the myosin-heavy isoforms for that fiber, just looking at the 3 main isoforms, 1 in yellow, 2 in blue, and 7 in purple. These fibers were ordered by increasing myosin-heavy 2. You can see that this blue line is always increasing. And generally, we have either a high percentage of 7 in purple, these are our slow fibers, or we have a high percentage of 2 in blue, and these are our fast fibers. So to summarize this plot, we have about half of our fibers in young people are the slow type, about 1/3 are fast, and about 1/5 are the hybrid. Now let's look at the same plots for the fibers from old people. You'll notice here that the proportion of slow fibers is similar, but the proportion of the pure fast fibers has decreased from 31% in the young people to 20% in the samples from old people. And you'll see that there's an increase in the hybrid fibers that makes up the difference. So this is well known that the percentage of fast fibers will decrease with aging and the percentage of hybrid fibers will increase with aging. So this is sort of a preliminary proof of concept or quality control analysis of our data. Beyond those myosin-heavy chains, there are several other protein isoform pairs that are expected to match either fast or slow isoforms, fast or slow fibers. For example, this circa pump or ATP2A1 and 2. However, it's not known whether these proteins should become more hybridized with aging. And this plot is showing you a histogram of the proportions of that ATP2A1 relative to its isoform across all of the fibers, with the young fiber proportions in blue and the old fiber proportions in orange. And what I want you to notice is that the young fibers are more likely to have a proportion of either 0 or 1. So they're more likely to be pure. And the old fibers are more likely to have an intermediate proportion of the isoforms. You can see that in the middle here with this lawn of these kind of orange-looking bars. So this perfectly highlights the concept of isoform confusion. As we're aging, our fibers don't know what they're supposed to be anymore, and they're expressing intermediate levels of these isoforms. Again, although this is known for the myosin-heavy isoforms and some of the light isoforms, it's not reported for circa pumps, and it's not reported for any other proteins that we looked into. So we wondered if this was a widespread phenomenon across more protein isoform groups. And to test this, we devised an unbiased pipeline looking for protein isoform group proportion distributions that might differ between young and old. Sorry about that. I don't expect you to catch all of this, but I'll point out that there are 509 protein isoforms that fall into 142 groups. And we used a KS test to determine whether their distributions differ between young and old fibers. And then we used very stringent filtering of the p-values, corrected p-values, to determine which isoform groups become confused with aging. And when we used this pipeline, we discovered many confused isoforms among the proteins that are known to distinguish fast and slow fibers, including that circa pump that I showed you before, but also TNNT on the right and a number of other contractile proteins down on the bottom here listed. So now these plots are a little bit different. We're showing you the ratio of the isoform ordered by increasing purity of that isoform, and there's 2 lines, one for all of the fibers from the old people and one for all the fibers from the young people. You can see that the shapes of the orange and blue lines are statistically different as determined by that KS test, and you'll notice also that generally there's a less pronounced transition for the old fibers than there is for the young fibers. Beyond those canonical fast and slow isoforms, we were surprised to find 23 additional protein isoform groups that were dysregulated with age. 2 example proteins are shown here with ECI2 involved in lipid metabolism and RTN4 on the right, which is an important protein for multiple cellular processes, and it's found in the endoplasmic reticulum. Among the other dysregulated isoform groups listed below, you'll notice that many of these groups are mitochondrial or metabolic proteins. So to summarize this first section, I've showed you that we may have found a potential mechanism of age-related isoform misregulation that could be undermining power in the muscle. What could be happening is the fast fibers might be progressively losing their distinct proteomic identities, and then they're adopting slow-like features, which is reducing their maximum force output. In this next section, we'll introduce a novel muscle fiber subtype that may explain some of those observations. As context, while we've been working on this paper, another study came out where they also analyzed the single fiber proteomes of over 1,000 single muscle fibers. And in their paper, they found evidence for only 2 fiber types. And you can see that represented on these UMAP plots. And their fiber types were cleanly correlated with the amount of myosin heavy 2 in the middle or 7 on the left. There was some amount of myosin heavy 1, but that was mostly only in these fast fibers that were dominated by myosin heavy 2. So it's essential to note that this study only included healthy young males. So that's a key difference from our study that I described. And in disagreement with their paper, when we look at our proteomic data, we find 3 fiber types with unsupervised clustering. And when we look at our myosin-heavy isoform markers, we see that one cluster on the top left is primarily the slow type, and the cluster on the bottom is predominantly fast. And then we have this third cluster on the right that appears to be a mixture of both fast and slow myosin-heavy isoforms, and we're calling this our dysregulated cluster. So notice that this dysregulated cluster is more prevalent in the fibers from older individuals. Remember, our baseline rate is 57%. So this 43% young is a substantial deviation from that. And also remember that that previous study on the last slide only included those healthy young males. So it's possible that this fiber type is actually a result of something that's specific in females or specific to aging or some combination of that. So what is this fiber type? This analysis uses Wilcoxon rank sum tests to compare the markers of each cluster. And I have overlaid here the main 8 markers of this cluster. In the middle here, PRDX3 and LGAL-S1 are proteins that may signal inflammation, oxidative stress, or compensatory regeneration. The 4 proteins on the edges, here and here, are all metabolic proteins. And the increase in all of them at the same time is actually contradictory. On the top, CKM and enolase are thought to be fast-twitch glycolytic markers. And on the bottom, the myoglobin and carbonic anhydrase should normally be slow-twitch oxidative markers. So this may be an indicator that our dysregulated cluster is actually metabolically confused. It's known that these fibers can switch their type. So we wanted to model the underlying process of type switching. And trajectory inference is a method that can allow us to do that. It's a metric of omic similarity among the fiber profiles, and it can enable us to predict how the fibers may change from one state to another. So here we used a common method called PAGA, trajectory inference, to determine those relationships. And in this graph, the thickness of the line, the edge, tells us something about the relationship between the groups. So you can see here that the strongest relationship is between the fast and slow fibers, with weaker relationships between fast and slow and the dysregulated group. When we look at the same analysis using only the young fibers, we can see something slightly different, but still similar. Here it's more apparent that the dysregulated group is the odd group, and there's a stronger connection between fast and slow. And interestingly, if we infer relationships between only the old fibers, PAGA determines a shift in the relationship between groups to indicate roughly equal paths between fast and slow or fast and dysregulated. So this suggests that the fast to dysregulated connection might be becoming more strong as we age. We can use those relationships from PAGA to obtain a pseudotime, which you can think of as a guess of how the fibers might transition amongst each other over the biological process of interest. And if we pick the fast fibers as the root node on the bottom here, we can plot gene quantities over the inferred pseudotime. So on this top graph, low pseudotime is blue and high pseudotime is yellow. So you can see that we're either going to slow or the dysregulated group. And when we look at the contractile apparatus, fast and slow proteins on the top of this graph, and glycolysis and TCA proteins on the bottom of this graph, you can see that fast and glycolysis are going down when the slow contractile proteins are coming up with the TCA proteins. If we look at the same proteins on this other path going from fast to dysregulated, we can see that the contractile protein patterns are the same, but now the glycolysis pattern has shifted. So we're actually seeing an increase in the slow contractile proteins with an increase in the glycolysis proteins. And remember, these slow fibers should actually be oxidative. So this could be further evidence supporting the idea of confused fiber identity, especially with regard to metabolism. Tying this back to the first section, these two findings appear to be related. So here you'll remember this style of plot from the first section. And the blue dots now are the single fibers from that dysregulated third fiber type. The green are fast and the orange are slow. You can see that there's two lines, one for the fibers from old and one for the fibers from young. And now you'll notice that most of the fibers in that dysregulated group are those with these intermediate levels of the circa isoform fraction. So this kind of ties the two stories together, and if this holds up, there might be an interesting connection, and we're working on validating that further because there are some contradictory cases. Importantly, we think that these observations in skeletal muscle are impactful because they're consistent with a broader information theory of aging that was put forth by David Sinclair's group. This is a figure from a Perspective article that he published with the citation here on the bottom. And this idea posits that during development, if we start at the top, our bodies are producing these cells with well-defined identities. And then they lose their identities as we age, and that causes tissue and organ dysfunction. In their original work, they showed this in the context of epigenetics, but we might have protein evidence at the isoform switching level. So we're really excited about this as a potential broad mechanism of aging. To summarize all of our unpublished results that I told you about, we found the story of protein isoform confusion, where the old fibers have more intermediate levels of various protein isoforms. I also showed you that this transition from fast to dysregulated gets stronger in the fibers from old people. And when we look at the trajectory from fast to dysregulated, we find this contradictory increase in the slow contractile proteins along with the glycolytic proteins. And altogether, these three findings, I think, support a story of protein fiber identity loss with age that's consistent with this concept of the information theory of aging. I didn't have time to show you that we also found a single protein, MYBPH, that by itself can classify young versus old fibers. We think this is quite interesting, and I haven't seen this in many proteomic data sets. Not much is known about the function of this protein, so we're looking at that now with some knockout models in mice. And we're also investigating the possibility of whether this third fiber type may be considered a type of post-mitotic senescence, because some of the proteins that are upregulated in this fiber type seem to be senescence markers, in particular this LGAL S1. And there was a recent paper that reported that, in fact, muscle fibers can become senescents with some evidence for that. So we're trying to explore that as well. But overall, I hope I've convinced you that single fiber proteomics gives us a unique lens into the biology of sarcopenia that might help us find mechanisms that can allow us to improve our health span. This is one mostly unrelated slide before we conclude. For those of you that are conducting single cell omic data analysis, this is the platform that Sarah mentioned. This is a really interesting platform because it allows you to interactively design your data analysis with drag and drop nodes. And when you're ready to finalize your output, you can easily share everything, including your data and your analysis pipeline and the results that you produced all together with your publication. We can give you a short link to do that. So it should increase the reproducibility and lower the barrier to doing your own data analysis. And I forgot to mention that it also includes an interface to the Cell by Gene Interactive Data Explorer. So if you want to get your data into that, this is an easy way to do that. So check it out with this QR code here. The URL is also on this slide at the bottom. So now I'd like to thank everyone involved in these projects. Cameron has been leading the analysis of this single muscle fiber proteomics data. He's here today if you want to talk to him and you have more questions. Simon collected the data from these fibers using that dual trap single column setup. Yuming helped with the sample preparation. Amanda led that first technical paper, and Lex is leading the development of that web platform that I mentioned. This has been a close collaboration with Chris Sundberg's group at Marquette and multiple people on his team, including Isabel, Laura, and Carlos. We're grateful for support from the NIH and also the ASMS Research Award. We're also grateful to work with so many people here at Cedars Sinai in the single cell group, especially Sarah Parker and Jenny Van Eyck and all these other people. And I've included my contact information here at the bottom in case you want to talk about any of these things. Finally, I'd like to thank all of you for your time and your attention. Now I would be happy to take any questions that you have.
Please raise your hand if you have a question, and we'll bring a mic to you.
That's really interesting work, and I just wanted to ask something related to the the fibers that did not, kind of change into or switch into senescent mode, if that's what we're calling it now, but are old fibers of the same type different than young fibers? Like, how much did you guys go into that? I mean, you mentioned there's one protein, or one gene, I guess, which can classify old versus young so far that you're pretty sure about. Have you looked into the overall state of sort of fibers that retain their identity, and if there's something there in terms of aging that can also explain what's happening with sarcopenia?
Yeah, that's a great question. That's actually the only thing that we started out trying to do with this data set. But I had to cut it because I didn't have enough time and I thought these other stories were a little more interesting. But we did look at that. Statistically, it's a bit difficult because you have multiple measures from each individual, but we can use a linear mixed effects model in order to get rigorous p-values for those changes. And in agreement with what I said at the beginning about how the disease is primarily in fast fibers, we see almost no changes in the old fibers, but we see about 55 statistically significant changes in the fast fibers. So there is some remodeling happening, and we're working on summarizing those differences, and I think we need to scale up. I think we need even more statistical power. We're hoping to get an R01 related to this. We got a good score, it's just in limbo. And we propose to do something like 15 or 30 people per group, and I think that'll get us a lot closer to the actual changes that are happening.
Hi. So, so you showed at the beginning, you talked that the power on a whole organism basis, the loss of power is more significant in females. It declines down to a slower percentage of their starting value. But on the single cell level, males and females, the male cells, they're stronger overall, but they have a bigger decline. And the female cells, they have a smaller decline, and they both decline to about the same amount. So if you just thought about it on a single cell basis, you'd say that actually it's more of a problem in males, not in females, at least at the single cell level. Is there some androgen-dependent benefit that is being lost, or can you comment on that?
Yeah, I can try. I'm not a muscle physiologist. That's not my role in this part, so I don't have a great answer for that. But I do know that there are other things happening. For example, your entire muscle size would matter. It's possible that females have less total muscle mass to start with, and that causes a difference between these measurements and the whole limb measurements. But I don't have a good answer. I'm sorry.
Thanks. So I think we need to move on. So I know your schedule says that lunch is next, but we are going to try one more time for the talk that was skipped earlier from Zarif Ajer, who is joining us virtually.
All right, so hey everyone, thanks a ton for sticking around. I know we're the last thing left between you and lunch. And so this will actually be a joint effort from me and Kang. Kang is a postdoc at the Levy Lab, and I'm a current undergrad over at Caltech, and I work in the Levy Lab as well. So I'm just going to go ahead and share my screen and give me 1 second. Okay, great. Yeah, so today we're going to be talking about some of our work on trying to unravel the heterogeneity in the colorectal cancer tumor microenvironment through some strategies on integrated histology and multi-omics. And as a quick note, sort of some of the crux of our work here will be using the G4X data that was described previously. So as a review, remember that G4X is sort of paired spatial proteomics and RNA and histology. So we'll see more of what that looks like here and how we sort of use that for some really cool findings. And before I start, could I just get a confirmation that folks can hear me?
Hear you, but we
Sorry, you can hear me, but you can't see the slides. Is that the case?
Yes.
Okay, here, I can stop sharing and try it again. Um,
Oh, we can see now.
okay, excellent. All right, then I will get started. So yeah, and so just as a quick review, right? Kang and I are both part of the Levy Lab. And so our goal really is to use all these tools, whether it's histopathology or sort of molecular imaging, to try to improve the process of pathology. And so really, right, when we look at all these tissue samples and we sort of generate all this high dimensional data, you know, our interest is in how can we use sort of multimodal data to actually make advancements in tools for clinicians as well as disease understanding. And this point on multimodal data, I just want to zero in on it for a minute. Right, we've sort of gone from a paradigm of just looking at imaging and sort of different patterns and morphology to running all sort of diverse molecular tests, you know, from gene expression to mutation profiling. And so we've seen that sort of revolution, that sort of transformation in sort of human pathology, but we've also seen it in AI for pathology, right? So as a lab, we really try to develop tools that can help clinicians make predictions about prognosis or about diagnosis. And we've seen that the AI models we use, they also, just like humans, they also drastically improve when we can give them more data. So for example, instead of just looking at imaging, can you also look at spatial molecular information, or can you also integrate proteomics? And we've seen that these sort of models do better when there is more data, which, you know, which might seem like an obvious conclusion, but sort of validating that what has really been one of the key milestones in AI for pathology in the last few years. And so, as I mentioned, right, our goal is to sort of bring together all these different diverse data types to improve our mechanistic understanding of disease. And so we focus on colorectal cancer, as well as to sort of develop new tools to empower clinicians. And so from this point of view, spatial omics, right, has been super interesting to us because it is inherently multimodal, if you will, because you get both the underlying H&E imaging as well as the gene expression at all these spots across tissue. So we've worked a lot with Visium data as well as Xenium. And so we're always sort of on the lookout for new technologies that can add to this toolkit. Because, right, as we've all seen in the last number of years, spatial omics and multi-omics have sort of had a pretty massive impact both in research, and then we've sort of started, I would say, edging towards the clinical adoption of some of these. You know, for example, in colorectal cancer, there's a tool called Quant CRC that's sort of gaining traction. And then there's also some approaches that have been around for longer, like CDX2 staining, like immunoscoring, and so on. And so you know, right, when we think of spatial omics, we've spent a lot of time trying to think about, look, what is left for spatial omics to truly have widespread adoption, and for it to make an impact in the clinic. And so what we've sort of come to understand is that the high resource cost of these assays is one of the main limiting factors, right? So right now, it's difficult to collect large-scale spatial transcriptomics datasets, for example, because, you know, a single Visium or a single Xenium run can cost you thousands of dollars. And so it's cost prohibitive, and the actual sort of the actual assay process themselves takes a lot of time, and you know there's often sort of issues with the protocol. You know, tissue gets torn up, right? We've had some experiences with that, and so essentially we're not yet at that high throughput, low cost scale yet with most of these technologies. So the data sets that we're collecting are not as rigorous. The sort of biomarkers that folks can come up with, you know, are not as rigorous, and they're not as robust. So number one, definitely the resource and sort of cost investment for collecting some of this data has been hindering its adoption. But then also, I just want to sort of appreciate the computational complexity of analyzing this massively sort of multimodal, high resolution, you know, sort of data sets. You know, you can imagine with a multi-omic data set, you might be looking at, let's say, like 50 different samples, and for each one, you've got, you know, information on like 5,000 different spots. Like all this data is inherently super high dimensional, and you know, folks come up with new computational pipelines for analysis, but that part is definitely not a solved problem yet. And so from this perspective, G4X is really exciting to us as a group because it sort of has the promise of, multi-omics with integrated histology, but at a lower cost and high throughput. So whereas previously we were talking about order of magnitude thousands of dollars for samples, folks have been able to sort of do the G4X per sample for in the neighborhood more of hundreds of dollars. And so that has been really exciting for us, as well as the potential for sort of paired RNA and protein, has been another point that sort of has drawn us to want to try out this technology. And so we have recently started gaining access to a pretty large scale cohort through the ColoCare study. So essentially, ColoCare is a multi-institutional sort of effort that has been going on since about 2018 or 2019. So you've got data for thousands of patients from all different demographics. And so the diversity in there is certainly what we're interested in. And so we have started accessing ColoCare samples here at Cedars recently. We've currently got a pilot effort underway where we've started retrieving a bunch of samples, and we're getting ready to profile them. And so thus far, we've pulled out 4 sort of samples that we've been able to run G4X on, and say we're going to go over some of the results that we're seeing and why we're excited about it. So here are sort of some of the demographics for the initial pilot cohort that we're looking to pull. And as I mentioned, right, what we're sort of hoping to get out of it is the histology and G4X. But then we're also looking to try to do some 3D sectioning, and we'll talk more in a bit about why 3D can be exciting. And then we're also looking to run Xenium and Visium on these samples as well, just so we're able to effectively compare what we're seeing from G4X versus Xenium versus Visium, and sort of assess the trade-offs of each modality, and use each modality to validate the others. And sort of our goal with pulling these samples and running these analyses once again, is to try to derive sort of more robust spatial biomarkers, and see if there's any improved mechanistic understanding that we can get, particularly looking at metastasis and progression. So this is a cohort that we've started collecting. As I mentioned, it's 4 samples so far, sort of 2 with metastasis and 2 without. And so right with the G4X, we're looking at the H&E, we're looking at those RNA markers and some protein markers. And then we've also been able to profile these for Xenium as well. So that's RNA markers, as well as H&E. And the additional point I'll make here is that with Xenium and G4X, we're really getting down to the cell level or the subcellular level. So it's not just spots like Visium. We're getting increasingly granular now, and that's been really exciting for us. Okay, and I'll let Kane go ahead now to talk you through some of the analysis that we've done and what some of our initial findings from that have been.
Hello, everyone. Can you hear me well?
We can hear you.
Okay, thank you for the information. And my name is Ken. I am a postdoctoral fellow in Libby Lab. I am very, very sorry for not being on site to present this talk in person. And also it is my pleasure to give a presentation to introduce some of our preliminary results for G4X data. So first, I want to give an overview of our analysis. So we have 3 different phases in our analysis preliminary. So the first phase would be the validate phase. So essentially, we try to use the protein of G4X data to kind of do an unsupervised clustering, and then we use this cluster to label the cell types, roughly speaking, and then we use these. What we want to know is that is the mRNA expression in the G4X kind of make sense with the cell types clustered by the protein expression or not. So that would be like an internal quality control for this phase. And the second phase would be to compare. Particularly speaking, we want to compare the G4X RNA and the Xenium RNA. So our expectation for this is we want to see some general similarities important expression, and we also want to see some of the difference as well. And these differences, if we can investigate these differences, we can really see some nuanced description of biological insights. And the third phase is explore. So we kind of try to find some interesting findings that is in line with the current literature. And the idea is that we also want to confirm with the studies out there that, okay, so G4X is pretty good and it can be used for further analysis. Okay. So before we dive into the analysis, I just want to talk a little bit about the spatial domain clustering that we use for cell type labeling. So the algorithm we use is the latent clustering, which is a very regular workflow that we use in single cell RNA analysis. But in this case, we integrate the spatial domain analysis to make sure that the clustering algorithm can be aware of the spatial location within the tissue of the cells as well. So we use the information from the protein expression, which translates to the protein graph, and then we have the H&E information, specifically talking as the location of the cell in the tissue. We have a spatial graph, and then we put them together, and we kind of do the clustering. So the results of the latent clustering is just about anonymous cluster, right? We know that, okay, so these cells are clustered together and these cells are not clustered together. But based on these clusters, we can really identify the upregulated genes of these upregulated protein, to be exact, of these clusters, and then we can really label these are tumor cells, or these are normal epithelial cells, or myofibroblasts, or smooth muscles manually. Okay, if we move to the next slide. So here are the results of their protein-based cell type labels based on the spatial domains. And we see that the tumor regions are really pop up from the tissue. And also we also can see their cancer-associated fibroblasts, which is represented in the blue dots. And we can also see the lymphocytes in the red dots. So my take here is that in the tumor microenvironment, the major components that we can see here is the cancer-associated fibroblasts and the lymphocytes. Actually, we can also see a minority of the myelocyte cells in these cells as well, but it's really not popped up, and we think that these are maybe related to the recruitment of the cell in the system, in the body system, to coming to the tumor microenvironment for a broad tumoric effect. So for the validation, what we want to do is to see if the mRNA expression in the G4X is really in concordant with the protein. So how do we do this? So we use the cluster of, for example, epithelial cells, and then ask if the cell have the upregulated genes related to the epithelial cells as well. So in this case, we use the tumor cluster for that. And what we are expecting to see here is to see the epithelial cells markers, specifically for the epithelial lineage, and some markers that are different from the normal epithelial cell as well. So in this case, we see the EpCAM and the CDH1, which essentially encode adhesion protein in the adhesion process for the epithelial cell interaction. And these are really pops up in the tumor regions. And we also see some of their more cancer specific markers that can distinguish the cancer cell with the normal epithelial cells. For example, in this case, the MYC gene and the VEGF-A gene, which makes sense because MYC is a very famous oncogene, and the VEGF-A is also a very famous oncogene for the growth factor for the angiogenesis to the tumor. And also we have the TS-band8. This particular protein is a transmembrane protein that is essentially related to the epithelial mesenchymal transition, which stabilizes the beta-catenin pathway. So I think this result is in concordance with the protein expression at least for the tumor regions or the tumor cells. Then we go to the validation for the lymphocytes, and to be honest, we have to admit that this lymphocytes labeling is the cluster of both the tumor infiltrating lymphocytes and the normal lymphocyte as well. So it's what you're seeing here is actually the markers for the lymphocyte that is present in both the lymphocyte of normal tissue and the tumor infiltrating lymphocyte, not really particular specific for the tumor infiltrating lymphocyte. So we have the CXCR4, which is the chemokine receptor in the lymphocyte, normally expressed in the lymphocyte as well. And we have the interleukin 10 receptor A, which is a pro-inflammatory receptor. And we also have the IgH-A1 in cause for the immunoglobulin heavy chain A1. And we also have the HLA-DRA, which is a human leukocyte antigen. This gene is interesting because they also highly the high expression of this in tumor infiltrating lymphocyte can be related to the prognosis, but they can also be expressed in the normal lymphocyte as well. And CD74 is kind of like the biomarker for B cells and some of the subpopulation of the T cell as well. So in general, I think these markers are in concordance with the protein label lymphocytes. So this is a very interesting analysis. So I did some upregulated genes in, we did some upregulated genes in cancer-associated fibroblasts as well. So our expectation in this analysis is that we can see some cancer-associated fibroblasts that express the non-specific fibroblast lineage markers. And we also want to see if the CAFs in this case can express some of all the other genes that is not really expressed in the normal fibroblasts. So in this, we see the FN1, the collagen 1A1, ACTA2, and VIM. They are all the biomarkers related to myofibroblasts and fibroblast lineage in general. But we have the IGF-BP7, which is a binding protein to the insulin-like growth factor. They can be related once they secreted by the cancer-associated fibroblasts, they can really promote the cell invasion or the cell migration we see in the microenvironment. And we also have the PDGFRB, which is the platelet-derived growth factor receptor B as well. So these receptor is also related to the pro-tumor microenvironment. So in general, we see that the CAFs have some similarity and difference in the transcriptional expression. And we move to the phase of compare. So essentially, we also have the xenium data that is paired with the same sample, in different section of the tissues. And we are curious that what is the difference between the xenium and the G4X, and is there so if we can see the common general patterns, is a very good cross validation between the two different technologies. And also, we want to see if they have some differences as well. So here we visualize some of the protein, like eyeballing to see how they are similar, how they are different. And we think that they have the very similar general patterns. For example, the VEGF-A, which is only expressed in the in the cancer cells. We can also see it in both the G4X and the xenium. And the FN1 is the specific marker for the myofibroblast lineage. We also see it in both the xenium and G4X, and similar to the ACTA2. But we can also see there are some differences, and these differences can be attributed to the section difference, and also for the technological differences as well. And then, so here we kind of show some of the deeper analysis in one sample, in particularly differential expression analysis and also the pathway analysis. So we kind of want to contrast the tumor with the other cell types, including like CAPs or lymphocytes or normal epithelium. So to see what is the pathway that is popping up, and what's the RNA that is popping up. So what we saw here is we see the EpCAM or CEA-CAM5. These are also adhesion molecule that is highlighted in the epithelial markers. We can also see some of the oncogenes as well, like the MYC and the TS-PAN8, which is a good sign. And also for the pathway analysis, we found that they have some significant pathway related to cell motility, cell proliferation, and some of the branching of morphogenesis, and also dysregulation in RNA synthesis, miRNA, and also hypoxia. So all of these pathways are suggesting malignant behavior compared to other cells, which in this case is normal epithelium or smooth muscles or caps or lymphocytes, which is making sense. Okay, so then we move to the explore phase. So in this case, we just make some observation and do some quick analysis to see if we have some findings that are concordant with the literature. In this case, when we wandering around the map, this to be clear, this map this time we do a little bit differently. We use the mRNA expression to do the spatial clustering. So we find that in the normal epithelium cells, we have a particularly different subpopulation that is popping up at near the basal membrane of the normal epithelium, contrast to the other cell in the toward the villi. So then we do the differential expression analysis, and we found that most of them are epithelial markers that is kind of the same with the villi one and the basal membrane one, but we have one of them pops up is the OLFM4, which translates to olfactometin 4 protein. This protein is particularly expressed in this particular cluster and related. I think it's related to the differentiation markers, but the results in colon cancer is a little bit mixed. Some say that it could be related to cancer, some is not. But one can be sure that it's related to the stemness of the cells. In particular, in line with our observation here, we found this cluster to express OLFM4 in only for the normal epithelial or the basal cluster. And this is also in line with the theory with the stem cell in the crypt of the gland of the colonic glands, and then they move toward the differentiated, fully committed epithelial layer. So my take here is that although protein expression can help us do a pretty rough clustering and very specific, but mRNA expression can give us a pretty sensitive clustering that we can look into the heterogeneity of the certain cell types, which can be very, very useful for dissecting the tumor microenvironment heterogeneity or tumor cell heterogeneity as well. So next we talk about our second findings. That's the cancer-associated fibroblasts surrounding the tumor cells. So we zoom in two different areas from different tissues to see what the tumor microenvironment adjacent to the tumor cell looks like. And what we found here is that the caps and the lymphocytes are the major component of the tumor microenvironment, except for the cancer cells. So we also see that the caps kind of surrounding the glands formed by the cancer cells, which is actually in concordance with one of our collaborators' studies, Dr. Abdestani, published in the 2025, which is pretty interesting. So this again can confirm the quality of the protein expression, but on the other hand, if we can do further step to dissect these caps using mRNA, because we know that the subtype, we have a lot of different subtype in caps, like inflammatory subtype or myofibroblastic subtype. So that would be definitely one of the future steps of our preliminary analysis. So with that, I hand it back to Zarif.
Yeah, okay, awesome. So thanks, King. And I hope that folks found that interesting, right? So as you can see, we're able to sort of use G4X in concordance with Xenium to show that both assays are picking up sort of reasonable molecular profiles, and we're able to use the G4X sort of the cross-modal behavior to find greater heterogeneity in the tumor microenvironment, and we're able to validate some of the findings that other folks have suggested. And finally, just to wrap it up, I won't go through all of these, but I just want to give you an idea of in the future what some of the sort of analyses you could do are. So we've worked on inferring sort of molecular markers from underlying histopathology. We've done that with Xenium and Visium. So conceivably, in the future, we may once we collect larger cohorts with G4X, we might be able to try to infer using AI the actual protein expression patterns just from the underlying histopathology, and sort of in the future maybe move towards virtual assays for spatial proteomics, sort of ways to get an idea of the proteomics before you actually do the molecular testing yourself. There's also, I think, a lot of work that can be done with modeling sort of spatial proteomics.
I won't be able to go to the meeting.
Oh
no.
Yeah. So a lot of work can be done with sort of modeling proteomics with graphs to predict outcomes and to sort of predict prognosis and recurrence, for example. And the final point is that when we start to look at the same tissue from different views, so yes, you could look at proteomics jointly with RNA, or you could take a 3D look at sort of proteomics and RNA altogether. We think that that multimodal molecular information will help us do prognosis prediction and recurrence prediction even better. So here are some of our early work, looking at spatial domains in 3D, for example. And so that was with RNA. One thing that we're looking to do is sort of compare 3D spatial domains with RNA versus protein. And so the sort of 3D sectioning that we're going to do with our current cohort is an exciting way to do that. So that's all. Thanks, folks, for sticking with it. We'd love to answer any questions, but we hope that we convinced you that G4X and similar technologies are exciting ways to better understand the tumor microenvironment, that multimodality and multi-omics is key, and that finally AI isn't magic, but when we can use it to analyze some of this high-dimensional data, then we start to see some really interesting insights.
Thank you, Zarif, and thank you, Kang. I think we can accommodate probably one question maximum. It looks like we don't have any questions in the in-person audience. I don't know about the Zoom chat. Okay, well, thank you so much, Zarif and Kang. And I'm sorry, we are just about to start our lunch break, so probably that's the reason. Thank you so much. And I would like to announce that we will have now 15-minute break, 15-minute lunch/bio break for people here in person. And we will see you back in the auditorium at 1:15. And Zoom folks, please tune in at 1:15. I should probably post that.
Okay. So I am Mitra. I am the Proteomics and Metabolomics Core Manager. I just want to, on behalf of the organizing committee, to thank everyone for participating in this single cell and spatial proteomics. And thank you for your patience with some of our technical issues this morning. So we have been fortunate that we can have an expert in spatial proteomics. She kindly accepted our invitation. Peggy Angel is tenured associate professor at Medical University of South Carolina and co-associate director of mass spectrometry imaging. She has developed multiple mass spectrometry imaging approaches to spatial biology, all of which are designed for use on clinically archived human specimens of tissues, cells, and fluids, and are developed working with clinicians. Notably, she is the inventor of a spatial method targeting the collagen proteome on formalin-fixed peripherin embedded tissue that integrated 2D collagen proteomics maps with other modalities. So please welcome Peggy, and thank you so much, Peggy, for again accepting our invitation. Yeah, sure.
All right, it's a huge honor to be here today. I really appreciate this symposium. So today I'm going to talk about leveraging spatial omics to unlock novel markers of precision medicine in tissue cells and biofluids, and just detail some of the work that we've done over the past years. Here's disclosures. So one of the interesting things that happened last year was that Nature announced that spatial proteomics was the method of the year. And so this was actually a result of maybe 15 to 20 years of really hard work and movement toward commercialization that produced a lot of different modalities for researchers to use in exploring the spatial environment. And so what you'll notice, though, when you look at some of these different images is that you'll see a dark space. And so this is what we're interested in the extracellular space. And so this is a picture of a heart valve, actually, and these dark spots are cells. And you can see that I could say more than 95% quite easily is the extracellular microenvironment. And so this is what my laboratory focuses on. In the way that I think about it is that just like our environment around us influences how we feel, the cells are immersed in this three-dimensional microenvironment, and it influences how they signal. And so what we have done is done a lot of work defining methods to look at the extracellular microenvironment in formalin-fixed paraffin-embedded tissues. But what I really like about the extracellular microenvironment is that it is a very unique and exciting space because it is made out of proteins. It has a significant chemical biology locked in post-translational modifications, largely glycosylation. And so the blue colors that you see here are glycosaminoglycans, and these greenish colors are collagen fibers in the heart valve. It's also very multiscalar. And so what this means is that when you look at the tissue components, you can see these very large strings of collagen fibers wound with elastin coils. This is a Verhoeven van Giesen stain. And you can see single cells embedded within this tissue. And what you'll notice is that these collagen fibers are actually at sometimes smaller than the actual single cells. And the elastin nets that form around the collagen fibers are even smaller. So there's always a lot of space to push for even higher spatial resolution of what we're doing and what I'm going to show you today. And so our primary question that we ask in my laboratory is, how does the extracellular matrix shape cell recruitment, localization, and signaling? And we largely look at that from a biomarker point of view. But if we look at this particular tissue here, what we see is that there are immune infiltrate patterns localized in very specific spaces on the tissue. There's cellular deserts where there's nothing there. And then you, of course, have your structural features. In this particular case, this is a breast tissue, and it actually should all look mostly like these adipocyte cells. And so there is this large biology associated with how cells move and localize within the tissue. The last 150 years have mostly been stains. So pathologists use hematoxin eosin stain, and so the pink that I show you today on most of those is going to be collagen. Verhoeven van Giesen stain, which I showed you on heart valve, and then Movat's pentachrome stain and trichrome stain. And so all of these are chemical stains that really don't get to the molecular composition of collagen structure. So what we know from doing conventional global proteomics, largely pioneered by Alexander Naba, is that these extracellular proteins are made out of collagens and then again heavily post-translationally modified. And so collagens make up about a third or more, actually that's more than a half, of the extracellular proteins in the microenvironment. They're heavily modified by hydroxylated proline. And the other ECM proteins that shape these collagen fibers are N-linked modified, which is a focus in my laboratory, as well as phosphorylated. So the challenge is here, how do we define this extracellular microenvironment in formalin-fixed, paraffin-embedded tissues, integrate wholly with the laboratory, and no extra sample collection from the patient to suit our analytical needs? So that's another point of my lab. If I'm going to develop an analytical method, I want it to integrate very closely with the clinic and not have them do anything different. And so the secret is that what we do is we use enzymes and we don't use antibodies. And so we have tissues on a standard microscope slide. We use immunohistochemistry-like workflows. We spray enzymes and not antibodies onto the tissue, and we use mass spectrometry imaging to scan across the tissue, and we get this highly multiplex signal here. All of these are peptides originating from the extracellular microenvironment. And so each one of these peaks represents an image channel. And so these peaks can be put together by machine learning and studied for biomarkers in comparison across the tissue or among different patients. Now, mass spectrometry imaging works using a laser that is scanned at discrete points across the tissue. And at each X and Y coordinate, it generates a mass spectrum. Each one of these peaks, again, is an imaging channel. So it's actually a lot of data. Typically, these images are around 30 gig or more each. And so you can see when you get to a large clinical study, you have an enormous amount of data present in the tissue. And so what we're using to produce all of this signal is collagenase. We're using the collagenase originating from Clostridium histolyticum because this is unbiased to species. It'll work on a mouse, it'll work on a zebrafish, it'll work on a human. And what it does is it targets connective tissue and proteolyzes it. And what we're doing is detecting those different peaks. We also get around 40 to 60 other ECM proteins that are involved in regulating the collagen structure. And it reports on these proline hydroxylation site modifications. One of the things that we have found is that when you change the protease, you have to optimize your mass spectrometry. So the collagen peptides are very different. They have a high glycine proline content. Because of their high glycine content, which is a low molecular weight amino acid, we have a different type of cutoff that we use for proteomics, and we actually have a higher percent hydrophobicity. Collagenase-produced peptides are also different because they have lower charge states than a tryptic peptide. And so we recently published a paper looking at optimization of collagenase-produced digest of collagens. So it's a completely different type of proteomics, but can be used for other enzymes as well. So this is an example of what we can get from one formalin-fixed paraffin-embedded breast tissue. So we can do single patient proteomics. We are pulling down glycopeptide sites, typically around 25%. And when we generate the images from these, we typically get hundreds to maybe over 1,000 different images that we can put together using different machine learning algorithms. And we don't tell the machine learning algorithms beforehand where the localization is on the tissue. And you can see here that these different colors, which are connected with different proteomes, are mapping to the H&E that's located over here. So they're following that same histology. This is an example in liver. This particular tissue had a higher significant sampling of fibrillar collagens, but we're still getting these other types of ECM proteins. And again, a very rich image structure that heuristically we can use with machine learning methods to segment out the different tumors and understand those. So we did show that this was highly reproducible. So this is on unstained FFPE tissue. So Denise Rushenarong, who's here at Cedars Sinai, also went through and showed that you could do collagen imaging on tissue that was already stained by an antibody, and we'll see an example of that in a minute. So this is an example of what we're looking at with these. And so each one of these peaks, again, correlates to a particular domain of the collagen code. And so this is a liver hepatocellular carcinoma. We have tumor. We have bridging fibrosis and normal. And what we see is that the same protein is differently modified and localized to different spaces on the tissue. We see the same thing in breast tissue, and in fact, colon tissue, prostate tissue, brain tissue. And what we're mapping is these changes in post-translational modifications. And you can see that they follow the H&E staining, but now we're actually getting to molecular information, and the one here indicates the site probability of that particular proline. So what does this mean? Well, when we actually pull apart the collagen structure is packed with these really rich domains that change biology by interacting with cells. And so different parts of the collagen structure can stimulate angiogenesis, fibrosis. It can interact to trigger downstream signaling. The hydroxylation of proline is an enzyme-driven modification that holds the collagen structure in this triple helical format. So we have within this triple helical bundle, we have all of this information on these different biologies that it can trigger. The hydroxylated proline holds these together through hydrogen bonding, masking or unmasking these sites that change the biological outcomes. And so in this particular case, there's several domains that are already known. And so this is an integrin binding site. And when it binds, when it is hydroxylated, which tissue engineers use an O to indicate a hydroxylated proline, it has high affinity binding and triggers a lot of downstream signaling. So each one of those sites has the potential to modulate how the cells see this collagen network spread throughout our bodies. Well, there's actually over 200 or so, 235 different prolines in the collagen structure. And if you think of each one of these as an independent potential that can happen, we have a lot of complexity. And so this also indicates that collagen-type proteins shouldn't be thought of as a single protein where we collapse their peptides into one protein to quantify them, but we also need to look at their domains to see how those are changing during the disease status. And so what do cells see as they move through the matrix? Well, they actually see these different domains and they actively interact with these. I didn't include the movie for today, but I'm just highlighting the different points where the cell is actually reaching out and retracting as it moves through the matrix. And this is also, I want to comment that this is the bare matrix. We know there's a lot of glycosylation that happens there. So it's really no surprise that in the past, collagen expression has been shown to be predictive of disease, and actually across many cancers. The problem has been that in the past, what we have seen are alignment, collagen type, collagen fiber regulation. We've seen RNA transcription, but now we're getting to the molecular composition. And so this is I think the beauty of what we can do with spatial proteomics is we can do this staining for this enzyme on the tissue, prolyl 4 hydroxylase 3. And on the same section, we can actually map the collagen proteome and understand that the P418 site is not modified relative to these other sites that are contained within the structure. So you also have now you have the enzyme expression by antibody staining. You have the transcriptome data. Both of these are indicating that there's active transcription happening for post-translational modifications, but it's not until you actually get to the protein structure that you can see these critical changes in collagen modification. And so what that has led us to is using this in a workflow that's multiplexed with not only looking at collagen fiber measurements, but combining it with different cell marker studies. But we will typically look at glycosylation, collagen, and then trypsin, because trypsin, when we digest that, it actually goes after a cellular type of proteome. And so this is an example combining this with geomics. And so here, what we did is we did geomics followed by and glycosylation imaging, and then collagen imaging. And so now we can get a more complete picture of what's happening within the tissue. So I wanted to show a couple case studies on breast tissue. Again, like I said, we have found that collagen domains have predictive value across many different disease states. And so if anyone has a favorite disease, ask me about it, and I probably have some data. So one of the dichotomies in breast care is that during clinical risk measurement, we're always measuring the stroma. We're looking at the stroma for diagnosis and survival. We're looking at stroma changes in therapy, but we really don't know the molecular composition of this stroma. And so what we have in our hands is tissues collected over 6 or 7 or more years. There's about 420 at least different patient donor samples, some in duplicates. We have normal breasts from healthy women from the Susan G. Komen Tissue Bank, ductal carcinoma in situ, breast cancer progression, and triple-negative breast cancer. So we have a whole range of different breast tissues that we can actually start trying to understand the molecular changes that happen in the breast microenvironment. And so this is a study that was done by Denise previously in my lab. And so here we had tissues from the Susan G. Komen Tissue Bank. This is normal breast. It actually comes with one of the most extensive virtual tissue banks that you can access at any time on your phone or anywhere if you have a question about any of the data. So our hypothesis in this particular study was that core fucosylation was increased in breast tissue at risk. And so the reason we thought that that was a great hypothesis is because we knew that TGF-beta, when that's core fucosylated, increases stroma. And so we thought, well, if we're seeing increased stroma based on BI-RADS breast scoring, then that's going to correlate with core fucosylation. And then there's a lot of literature showing that core fucose, the enzyme that makes that core fucose, is elevated in breast cancer, and we have increased levels of breast cancer core fucosylation associated with tumor progression. And so what we're looking at here is this particular fucose, which is marked as a red triangle. That's at a N-acetylglucosamine that is attached to the protein structure. And then we're comparing that with multiplex glycomics workflows to outer arm fucosylation. And so here's an abbreviated workflow. And to our surprise, what we saw was that actually core fucosylation was associated with the adipocytes, and the outer arm fucosylation was associated with the breast stroma. We were not expecting that. And so we did find one study that was done in 2014 in Serum that was looking at, that also reported outer arm fucosylation in breast cancer, but the controls were male dominated. And so we could also look at the BI-RADS categories for scattered fibroglandular and heterogeneously dense. And here we were able to see that outer arm fucosylation again distinguished these two categories. And you can see that that these glycan patterns are aligning with the stroma. Whoops. And so we can take those a little bit further, and this is another example where we can see that a single glycan, and we had a couple different glycans where outer arm fucosylation gives a high distinguishment of the differences between the BI-RAD categories. So we knew that glycosylation patterns were unexpectedly different in normal breast. So we also looked at the collagen peptide patterns. And again, we're looking at these from the standpoint of collagen domain. And this was done by Jacqueline, who recently graduated, but she was using QPath to pull out only the stroma from these tissues. And so it's a stroma to stroma measurement based on clinical characteristics. We did a templex staining. I'm not going to spend too much time on that. But when she looked at the collagen network across the normal breast, what you can see, we can just focus on this particular tissue here. You can see that there is an intensity variation signified by a red color varying across the breast stroma relative to its H&E. So the pink here is going to be the stroma content, the whiter is the adipocytes. And so We compared these versus fibroglandular and heterogeneously dense. And what we found was out of more than 500 different proteins, we found 47 significant peptides that could predict the category of breast density. And so what that to me tells me is that we can actually start getting better at understanding breast cancer risk and actually having a molecular measurement for it versus just a portrait. Now, the exciting thing is that we're taking this to look at transitions to breast cancer. So this is ductal carcinoma in situ. It forms these lesions in the breast microenvironment that are not associated with risks such as solid cribriform or chemetonecrosis, which looks like this donut here, that is associated with breast cancer risk. This particular tissue has a cancer site located in it. So we compared pure DCIS to DCIS versus invasive breast cancer and invasive breast cancer. And again, what we see is that these peptides that are involved with these lesional systems in ductal carcinoma in situ and non-obligatory pathway to cancer are different than those involved with invasive breast cancer. We are very fortunate to have a collaboration with Rob West out at Stanford. And so we have access to the resource for archival breast tissue. And so here what we have are patients who've been followed, who've been diagnosed with ductal carcinoma in situ and have been followed for up to 15 years to see if there was a recurrence of ductal carcinoma in situ or an occurrence of invasive breast cancer. So clinicians count this as the same thing. It's increased risk. And so what we saw was, again, that there was very specific collagen peptides, which I'm not giving the M disease here on purpose, that can distinguish recurrence in these patients. And also we found that there was very specific peptides that could also link to disease-free survival time. So now we're starting to build this portrait of across these transitions from normal to breast cancer, we have these collagen peptide differences. So the final breast project is triple-negative breast cancer. And so this is around 16% of all cancer diagnoses associated with populations of African American and high BMI. It's a very aggressive cancer with limited therapies and frequently goes to metastasis, which further drops the survival rates. So we were lucky to have this cohort of around 80 tissues that were annotated by Graham Kolditz, giving us around also 80 different clinical characteristics, and we focused on survival here. So we used multimodal multiplex techniques to look at both the collagen fiber measurements, cell staining, as well as glycans and collagen regulation within this cohort based on survival. So this is just a picture of the different stroma markers that we used, and we're using a laser labile or a UV labile peptide tag by Ambergen. So these are some examples of some of the staining that we can see. We can do highly plexed, because as I showed before with mass spectrometry, you have a very high capability of multiplexing as you can go out very deep into the mass range. So what we use these cell markers for was to look at this previous expression that had been reported with triple-negative breast cancer. And so we saw that CD20 differentiates triple-negative breast cancer survival. When it's high, better survival. When it's low, poor survival. This correlates with literature. We also saw that PD-1 showed the same example where we had PD-1 positive infiltrates with improved survival and low expression in those that did not survive. We measured the collagen fiber curvature, as that has been reported before as well. So this was measured across the entire cohort. And what we saw was that in normal or in higher survival, there was a wavier pattern correlating with previous data that had reported wavy patterns around more normal tissue. And then as tumor grows and expands and starts to take over the microenvironment, it actually causes straighter collagens. And so we saw the same thing in our data where we had a wavier collagen form in those that survived versus a straighter in those that were deceased. And so based on this, we felt that the cohort was validated, and we moved to looking at new predictors of outcome. And so this is just one example of the glycans. This is data that's now submitted. This is a hot tumor, high infiltration. This is a whole talk that I can give on these two tumors. But what we see was that there's a particular glycan that is core fucosylated that actually links with poor outcomes as well. And in our paper, what I'm not going to show here is actually there's more outer arm fucosylated associated with poor survival than there are core fucosylated. So it's corresponding with the normal breastwork. So we also looked at collagen peptides. And so again, we have hundreds of these types of peptides. And you can see not just from survival probability, but looking at the tissue maps that we have very intense expression in those particular patients who have deceased. In this case, it's metastatic breast cancer. We can look at these in a couple different ways. So here we have one peptide that's modified at two different proline sites. And when we look at its singly modified counterpart, we see that it's not associated with survival probabilities. We also have quantification for these. But these also present different spatial characteristics on tissue. And what we found was that it was more informative for us to measure the entire tissue in breast cancer versus trying to outline only the tumor. And so I think that that's something that changes dependent on which cancer we're studying. But the highlight here is that we have different post-translational modifications associated with survival that are shown in different locations on the tissue. Now, I previously told you that there was different domains in collagen that we've already mapped. And discoidin domain receptor binding are domains that have been mapped by the tissue engineering community. And then in the cancer field, there's been a lot of work looking at discoidin domain receptors themselves and showing that their activation involves immune exclusion by reorganizing the collagen fibers. And so we think that we're presenting a more holistic picture once we are able to combine both the cell markers of discoidin domain as well as those of the collagen peptides where high expression indicates poor survival. And so this is just an example of the tumor microenvironment. Again, this is modified discoidin domain, which is poor survival. You can see it's located very specifically in a tumor region. And then here's the unmodified that's more extensively present throughout, where yellow indicates high expression. So overall, we found around 14 different peptides, which I thought was a reasonable number, seeing that we're finding hundreds of different peptides. So one question, although I'm not going to spend too long on it, is like, what do we do when we find a new peptide? How do we validate that? Well, we have a paper out where we found a specific peptide that I'm going to call the G pager. It was linked to previous mutations in Ehlers-Danlos and bone disease. We actually incubated it with cells and showed that there was a change in proliferation and chemotaxis. We also showed that it activated the transcriptional programs and it increased DDR2 particularly, as well as TGF-beta1. And so here we're comparing the modified versus unmodified or alternatively modified peptide incubation. But the cool thing that I thought about it was there was a clear evidence that these collagen domains activate transcriptional programs. So that means that there's a lot more that we need to find out. So I just want to end with leveraging the mass spectrometry platform for more than just tissue. We have a number of different studies where we have found clear biomarkers. This is in colorectal cancer. This is looking in an impoverished region versus a more affluent region in the same state. And we can distinguish very clearly between different stages. The imaging data also shows us where those markers are located, which is critical. So this is another study in hepatocellular carcinoma, again, where the imaging shows clear expression patterns in the tumor. This is in better outcomes versus poor outcomes. So you can get the quantification from this as well as the area under the receiver operating curve. And these types of area under the receiver operating curve numbers are not unusual when we're looking at collagen peptide variation. And so what we wanted to do is say, can we come up with a non-invasive way to target collagen peptides? Well, this is a really hard problem by chromatography. You have to do extensive work, but what we developed was basically a middle-down peptidomics method. And if you want to ask me about it later, I'd love to talk about it. But with this method, what we're able to pull down in one shot, and it is a simplified workflow, is several different markers that many groups have spent a lot of time working and reporting papers on. So we can pull these down at one time, which means that we can have stronger biomarkers that we can then link back to the tissue microenvironment. We've also adapted this method for looking at detection of extracellular matrix from cells and cultures. So these are using LabTech 2 slides that fit inside the instrument. This is an example looking at spironolactone in primary cardiac fibroblasts. This is looking at induction of senescence in a lung carcinoma. So this is the applications that can be used to study the extracellular microenvironment. We also have examples of looking at genetic knockouts in that paper. So in 2019, right before COVID, Anand Mehta, myself, and Rick Drake, which we call the Mad Labs, got together and we thought we're gonna go after this U54 and we're gonna do simplified workflows for glycans. And so we each wrote an aim on that grant, and it's really launched, I think, a whole new way to use the imaging platform. And so from this, we can take our typical scanning tissue approach, and we just apply that to either serum spots, plasma spots. You can apply it to cells, and you can get an array readout that gives you visual data as well as peak data. So it's extremely informative and adaptable. So this is just an example of looking at direct detection of glycosylation from cells. So this was published well after I graduated, but it's something that I did participate in the study. But we found if you labeled this particular nitrogen and glutamine, which is essential in cell culture, it actually carries forward into N-acetylglucosamine, N-acetylgalactosamine, and sialic acids. And so with this, you can actually start measuring in the mass spectrometer very quickly, and this is showing only one glycan, but the emergence of a glycan during oxidative stress. So now you can do this in an array fashion over many days and get a highly omics type of data. We haven't yet combined it with looking at the EndoF, which would be core fucosylation, and PNGSF, but you can see that there is an emergence of this particularly branched type of glycan that's also associated with tumors. So one of the things that we invented, and last year we sold to Brooker, was the glycotyper. And so this is based on 40 years of glyco literature. And so the problem is that the techniques to look at glycosylation have been very complex. And that's a hindrance to people who are, interested in wanting to study those. And so we started this with a graduate student, Allison Black, just hand spotting an antibody. And she carefully learned about how much antibody to spot to capture a target glycoprotein that we could then quantitatively profile for their glycan changes during disease. This was then transferred to a company called Glycopath. And here we developed the surface chemistries and techniques that allowed us to print small spots on a slide of antibodies, capture glycoproteins, and then use our simple workflow of spraying PNGSF to release the glycans. And then now we have the specificity from each of these glycoproteins because it was captured, and we can look at their glycopattern as a marker of disease state. So I'm not showing here a huge amount of work that was done in actually automating the whole workflow, going from adapting the instrumentation to having it automatically report a quality control analysis and then add to a database. So it's an extremely powerful approach, and at the end, each one of these slides is scanned in around 5 minutes. So I wanted to show, particularly to this group, you can do these very fast assays where you can have pretty much anybody in your staff who knows how to pipette. They can pipette an array of plasma, for instance, if you have plasma from different disease states, and look at the glycopatterns. And then from that, you can move on to using this glycotyper where you capture the different glycans. And so here we're looking at, can we predict primary graft dysfunction before this happens during lung transplant? So when lung transplant happens, a certain percentage of the patients will develop primary graft dysfunctions. And we found that glycans were a great way to predict this actually, and we started with just plasma, and then we moved to actually connecting it to the IgG protein. So it gives you that additional biomarker specificity. That's it, around 37 minutes. I want to say that the next talk is going to be amazing. Here we're going to talk about single cells by mass spectrometry imaging, and I'm not going to say any more about that, but I'm terribly excited. But, yeah, thank you.
Question. Okay, I'll ask mine and as I'm walking over there. So mine's kind of straightforward. I think you've an impressive and compelling picture of the value of glyco modifications for biomarkers. I wonder this is a really basic question, like, are there interventional strategies? Because clearly these things seem like do you think they're passengers or do you think they're actually pathogenic?
I think they're pathogenic. I mean, they have the cell has, immune cells have what's called a glycocalyx. And so that glycan structure is very interactive with the environment about it. And it's also rapidly modulated. So it can be modulated in under minutes. And so I believe that's what Dr. Dressman is going to show in the next talk, is his continuing work on looking at how these changes happen and how they contribute to pathogenic cell response.
Hi, Dr. Pezzi. It's an exciting talk, actually. I have one question. So most of the data you showed here is related to the breast cancer with this technique. So I wonder, you have any other biomarkers for other field? Means if it is go for an infectious disease or any cancer related, other than the cancer related, most of the things, what you are showing here is a cancer related things, actually.
Sure.
How much you are hopeful to get the expression of the biomarkers in other diseases, actually?
Yes. So we have some studies that we've done, for instance, in mouse models of immune treatment in myocardial infarct. And so those are showing very distinctive regulation of collagen domains. So I can show you pictures of that if you're interested. We've done it in lung fibrosis. And so one of the studies there that was published was looking at normal lung versus low-grade lung adenocarcinoma. And what I expected was collagen's only going to be up in cancer because you see fibrosis. But what actually happened was that there was very distinct collagen signatures associated with the healthy lung. And so I'm always begging for samples, so if anyone's interested. But yeah, it does translate to other diseases. And if you think about it, all of our tissues have extracellular matrix, so there's got to be an involvement somewhere.
Okay. The thing is that what I understand, if it is going to work for other things, means other infectious disease or other diseases, then it is going to be a hallmark detection method for everything. So we have a problem, we have a disease, we have a health dysregulation, we can go for this kind of procedure, and anybody can detect the disease. So it is having we are having the detection method in the middle of the thing. One side is a therapy, other side is a diagnosis.
Yes, exactly.
So if a physician get a patient, and if he thinks, he or she is having this kind of issue, so he can go for AI-driven machine learning. So my next question is related to, you never highlighted anything to the which kind of softwares or who are developing these softwares, basically that detecting all these signals in the machine learning software. Is there any particular language or any other lab is involved here? So because all the things are what you are showing are going to be detected by the software engineers. So that's a big part here, what I understand.
Oh, absolutely.
Yeah. So is there any lab who is developing this software, or it's readily available in the
There's
market?
a lot of laboratories developing it, but I think that what's going to happen is AI is going to completely subsume all these lab-driven softwares. And so I was really excited by the work done by the G4X team in producing those. I think it's got to be a collaborative type of work and not a single lab. When I started imaging, it was a one-shop stop, meaning you did everything. But now we need dedicated teams who can actually also communicate among institutions to build better biomarkers and a better understanding of human health. I agree with you. Yeah.
Impressive talk and intriguing data. Just a small question, because you were trying to relate it where you showed something in the colorectal cancer, and then maybe the main was a breast cancer, right?
Yeah.
If
I
you
couldn't resist.
yeah. So you said before you started the talk that you may be having different in most of the cancers you may have done it, right?
In most of the cancers, I'm sorry.
Other cancers, maybe you have some data. But what I'm trying
Yes.
to hint on or ask about is when you try to see these modifications, especially with respect to the collagen, let's say I do a pan cancer analysis. How
Mhm.
conserved are these modifications, and how I understand the biomarker
Yeah.
concept, but if I try to relate it to the enzymes which are exactly doing these modifications, how relatable that is, and how you can have a conserved modification or a PTM.
Yeah, I think some of them are going to be conserved. I've put together some rough slides that they're not for presenting, but just like, for example, in kidney cancer, like, is it in the tumor or not tumor? In lung cancer, tumor or, healthy lung, right? So we can put those types of patterns together, but I think that speaks to the complexity of collagen regulation because it doesn't always follow the same pattern. There's some that do. But when you look at literature, you see the same thing where you'll see a collagen type protein is helpful in one type of cancer in one stage, and then in another stage or another type of cancer, it's regarded as associated with metastasis and poor outcomes. And so I think that that molecular level and putting together that kind of landscape of cancer would be really valuable.
Thank you.
Yeah, thank you.
Well, I think we should thank Dr. Angel for her wonderful talk. Really, really exciting new way to think about what we can learn from spatial.
Okay, so our next speaker is James Dressman from Sean Bendel lab in Stanford. His talk is solo cell overcoming resolution barriers in MALDI imaging with high throughput single cell microarrays.
Well, it's always hard to follow Dr. Angel, but I first just want to thank the meeting organizers for giving me this platform to present my research, and I want to thank you all for being here to listen. And so my name's Jake Dressman. I'm a postdoc in Dr. Sean Bindel's lab at Stanford. And a lot of the work that I'm actually going to be presenting was done during my graduate work at the Medical University of South Carolina under my PI, Dr. Anand Mehta. And so I'm going to be presenting a platform that we developed called Solo Cell that uses single cell microarrays to perform MALDI single cell analysis. So I think it's kind of safe to say that the evolution of single cell technologies kind of drives our understanding of biology. A great example of this is the evolving model of hematopoiesis in immunology, where when we had the ability to only look at a handful of markers at a time, we had rigid classifications. But as more single cell approaches came about and they became more routine within the lab, like mass cytometry, single cell RNA-seq, or single cell ATAC-seq, that turned into this kind of model, this continuum, as we added more information to it. But all of these approaches measure DNA to RNA to protein, which can be highly correlative. So that kind of poses the question of what other biomolecules can we actually analyze at the single cell level to provide more information. And so a great example of another biomolecule is the post-translational modification in glycosylation. So I was first introduced in glycans at a small biotech company where we made monoclonal antibodies. We would measure glycans on these mAbs for each batch to make sure that there wasn't any changes, because glycans can affect serum half-life, its antibody-dependent cellular cytotoxicity, and complement-dependent cytotoxicity. So if these changed from batch to batch, it would negatively affect a patient's outcome in the clinical trial itself. So a great way to analyze these biomolecules is through multi-mass spec imaging, like Dr. Angel was kind of saying. This has a very wide array of different analytes that you can measure, from metabolites to lipids to glycans to peptides. And not only can you analyze one analyte per sample, but you can start multiplexing things. So Dr. Angel, again, published this approach back in 2021, where she used sequential enzymatic digestions to analyze the spatial distribution of glycosaminoglycans, in glycans, elastin, and collagen, all from the same tissue sample. And more recently, this was actually published last month by Erin Seeley. She reported a way to image 8 different modalities from the same tissue. So you can extract lipid data, metabolite data, in-glycan data, O-glycans, proteins, and peptides. Again, all from the same tissue sample. Another aspect of multi-imaging that is, I think, overlooked is its broad sample compatibility. So I was first introduced to glycan imaging at the Medical University of South Carolina during my graduate work, where we performed a lot of in-glycan imaging. This is where we use the enzyme pingase F to cleave off in-glycans on the surface of archival FFP tissue or fresh frozen tissue to spatially map its distribution on the tissue slide. But since then, we have adapted the approach to look at things other than tissues, such as biofluids and cultured cells. And a large part of my thesis work was centered around further expanding this approach to non-adherent immune cells through this antibody capture array approach. It's a very simple method. We take a protein reactive slide, we spot down an antibody on it, and then incubate cells over top of the array, where they're captured via antibody receptor interactions. And once we have the interactions, so once they're captured on the slide, we can take them through our in-glycan imaging workflow, where we get a robust in-glycan profile. And once this paper was published, we kind of realized we were a little narrow-minded. This is a MALDI imaging platform, so we can start multiplexing and getting more information from these captured cells. So things like tryptic peptides, lipids, and glycogen can be added to this workflow by simply taking what we already know from what we can multiplex on tissue and applying it to the captured cells. And so here's an example of first taking a lipid profile from captured cells, washing away the matrix, applying pingase, and we can get a nice in-glycan profile. And then we can spray another enzyme called isoamylase, which chews up the internal glycogen within these cells to make them into ionizable fragments, and we can get a quantifiable readout of the glycogen stored within these cells. And then finally, we can get a tryptic peptide profile after we've sprayed trypsin. And what's nice about this approach is if you perform these things in the proper sequential fashion, every step enhances the next. So you have to delipidate the cells prior to in-glycan analysis. And if you want to do a tryptic peptide, get a tryptic peptide profile, you have to, or you don't have to, but it helps to remove the glycocalyx because it increases access for trypsin to start cleaving those proteins. And so this approach, like I said before, it's a 1.5 microliter spot of antibody. You get thousands of cells within a capture region, and you image the region, and then you pool all that signal into one readout. So this is missing a lot of information because within those thousands of cells, there's a lot of heterogeneity. So that got us thinking about how we can make this approach a targeted single cell approach. And so one of the limitations to MALDI is its resolution. Routinely, MALDI imaging is conducted at between 10 and 20 micron resolution. So when you apply that to a tissue section, you're not imaging single cells. You're imaging cell clusters or cell neighborhoods. And so to kind of get around this resolution barrier, previously, single cell resolution has been conducted by sparsely seeing cells on a slide and then continuously rastering the whole region, and then pulling out ablation marks that are only over one cell, and then excluding ablation marks that are over multiple cells or no cells at all. Some disadvantages to this is the obvious low throughput. You can only pack in so many cells on one slide before you get a lot of overlapping ablation shots. Oftentimes you do have to use adherent cell lines. And this also requires a pre and post image to co-register the slides on the cell versus the ablation marks over top of the cells, which isn't necessarily a trivial task to perform. And then what this also doesn't include is analyte delocalization. So when you start spraying these enzymes on these captured cells, you can they start moving from their point of origin. So glycans and glycogen are very hydrophilic. These enzymes require humidity to actively cleave, and you can get a little bit of delocalization. So if you have cells over top of one another, you can have signal from one cell bleed into the next. And so our solution to this is kind of this one shot per cell solution, where we're taking that capture spot that I was talking about previously, shrinking it down to a size that could only physically capture one cell, and then taking those capture spots, spacing them apart from one another in a way that we can have one laser shot per cell without any overlap from the adjacent cell, to have this kind of simplistic high throughput approach for single cell analysis. And the way that we are fabricating these microarrays is very low tech. We're using microcontact printing with PDMS stamps. This approach has been around since I think the mid-90s, and it's very robust. And so you simply just take a PDMS stamp, you ink it with our capture protein, where it binds via weak electrostatic interactions. You take that stamp and stamp it on a protein reactive slide, let it incubate for a little bit, remove it, and you have your imprintation of the stamp. And then you can incubate your cells over top of it to have single cell capture. And so here's a fluorescent image of one of these microarrays. We're using a commercially available PDMS stamp that has spot diameters of 30 microns, spaced 60 microns apart. And this is very easy to fabricate and make. These spot diameters can actually get down to the sub-micron resolution, so spot diameter between 10 and 30 microns is very easy to fabricate. And so it's kind of fun playing with some theoretical numbers here. Like I said, PDMS is very customizable. You can make a stamp that is a full slide, so 75 by 25 millimeters. And if you have spots that are 10 microns in diameter, spaced 50 microns apart, you could get theoretically 750,000 single cells on one slide. And then you can add in well chambers to partition off the slide, so you can start adding in more samples. And PDMS is reusable. Once you stamp the slide, just with a little bit of ethanol, water, and some sonication, you can remove any residual protein, and then it's ready to get re-inked again, and then you can keep on stamping. And so once we incubate cells over this array, we actually get a pretty decent bit of capture. And so I was very excited when I initially saw this image, but quickly realized the next problem, which was selecting the single cells for laser ablation. And if any of you have dealt with Flex Imaging, which is Brooker's imaging software, manually doing this would have been very monotonous. And so one of the reasons why we're getting a lot of doublets, triplets, or no capture at all is because these aren't ideal single cell parameters, we're using a commercially available stamp here. So if we had spot diameters of 10 or 15 microns, we'd probably get a lot more single cell capture here. And so I kind of took this problem of single cell targeting and presented it to my lab mate Furkan Bayram in Dr. Anand Mehta's lab. And he came up with this great solution to use a convolutional neural network to target single cells. And so a CNN is only as good as its training data. So he manually labeled 20,000 images or capture images as single cell or not single cell, and then trained a convolutional neural network and validated it to recognize only those single cells. And from there, he built a framework that was a very user-friendly framework or program to allow our user to easily use the CNN to make these decisions. So it starts off with a high-resolution image around 10x magnification. We then, it prompts you to select an area of where your captured cells are. You then manually select 7 adjacent cells to calculate the average distance between your cells, and 2 farthest points to calculate the slope of the grid. And what's nice about this is because this allows the program to be grid aware, and it can partition off each capture region to then feed to the convolutional neural network to make its decision of single cell or not single cell. You can then, it then prompts a histogram to allow you to actually toggle the stringency. So if you want a higher competency in your single cell selection, you're going to have less false negatives, lower, less false positives, I guess. And then it spits out a list of your single cell coordinates. And these are just XY coordinates of cell 1, 2, 3, 4, and so on. And here's also another readout of the program where we have our selected single cells. And from here, we can take every single, or like all the coordinates and upload it to the Flex Imaging software. It's already compatible with this. And then we had just hit go on the instrument. And so here is live acquisition of single cell in glycan data. And every one of these laser shots here is over top of a single cell. And we're operating at around 6 cells per second. And one of the reasons why the laser's moving around in this kind of snake-like sporadic fashion is because Furkan programmed a nearest neighbor algorithm. So to optimize the stage movement to increase the throughput. And once we have the data acquired, it's very easy to process. You can start with imaging lipids, and if you want to then go to glycans, you just resave the file, and the order of your single cells acquired stays the same. So cell number 13 from analyte 1 will always be cell number 13 of analyte 3. And each pixel in this case represents a single cell. So here is an image of kind of a representation of the current throughput. We used a 24-well module here to partition off each sample. One well can capture a little over 2,000 single cells. And here is an image of the post ablation. So we have our matrix image where the laser has actually ablated, and in each one of these squares, in each one of these squares we have our single cell to kind of confirm accuracy. And this took about 6 minutes to acquire. And to further compare this to conventional methods of high-resolution imaging, if we would have had this same region, this 5 by 7 millimeter capture area, run at a 10 micron resolution, we would have 350,000 pixels versus our approach, which is targeted single cell analysis, a little over 2,000, which is a 100-fold reduction. And this reduces your acquisition time. It reduces your instrument degradation as well as your data storage. And then when you go to process and see your data, it takes about 5 minutes to run within the Skills software system. And then to confirm that the cells can actually go through multiple rounds of imaging, here's a high-resolution image of cells. In A, it's right after capture and post-fixation. B shows cells, those same cells after they've gone through lipid imaging, a car noise wash to delipidate them, and even antigen retrieval. So we can boil these cells and they're still on. And then C is finally, this is post-glycan imaging. So they're still there for further rounds of investigation by MALDI. And here's representative spectra of a single cell. So we have an A, its lipid profile, and B, its glycan profile. And when we start to compare the signals between single cell and bulk, it's actually very representative. So here we have the average signal from 3,250 single cells compared to bulk analysis. And we can see that the R-squared value is 0.95 and 0.93 for N-glycans and lipids. What's also nice about this is capture proteins can be modulated. Antibodies work great when you're going after a highly abundant marker, and they're specific. But what we also found was very nice was having a universal capture protein. So every cell out there is glycosylated. So lectins turned out to be a very nice universal capture protein where we can stamp down CON-A, which is a lectin that binds mannose residues, which is found on every glycan. And if we incubate, and we incubated a lot of different types of cells, and they were all captured. We have Jurkat cells, PBMCs, and 4T1 cells here. And so with this combinatorial approach of antibody and lectin, you can run these cool experiments like this, where we can take a PBMC sample, capture CD4, CD8 positive cells, also monocytes with CD14, and B cells with CD19, and perform bulk lipid analysis. But then also on the same slide, perform single cell analysis with CON-A lectin. And when looking at the data, this is the single cell lipid data where we don't know, where we're indiscriminately capturing cells, so we don't know their actual phenotype. We see two different populations partition out. And then when we overlay the bulk data, you can see that it's kind of sequestering based off of cell lineage. So we have our CD4, CD8, and B cells on the right, and then our monocytes on the left, so myeloid and lymphoid derived cells. And here's just a confirmation of comparing the individual lipid ratios between myeloid and lymphoid cells for both single cell and bulk data, just showing that there's a Pearson correlation of 0.85, again confirming that the single cell data is recapitulative of the bulk data here. And finally, what we envision for this is to start adding in more analytes. Right now, this is validated for lipids and N-glycans, but we would like to add in immunophenotyping approaches where we are either using ambrogen with the Merlet probes or potentially pairing it with other mass spec imaging approaches such as multiplex ion beam imaging and even fluorescent approaches with Codex. And and as well, this could be a long shot, but adding in spatial transcriptomics just to see how much we can take the cells through. And with that, I just want to thank my lab groups back at MUSC as well as Stanford and all the funding sources. And I'm happy to take any questions.
Thank you. Wonderful talk, James, Jake. I really liked your suggestion that you would get even better results if you start delipidizing, removing glycocalyx, and then moving forward to proteins. And here is my question. Have you tried, there's a little bit of weird sound.
yes, I've done that once, and I think it still needs to be optimized a little bit, just because of delocalization. I think the extent, because with triptych digest, it takes 5 hours to overnight, and I think that much time in a humid environment allows for some delocalization. I didn't get that far. Once I started looking at the images, I was, yeah. 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 Mhm.
Do you have any restrictions in terms of what you have to do? How do you deal with these
So
cells?
primarily I've dealt with non-adherent cells, so I haven't really had to optimize trypsin because, yeah, I get your point with if you want to remove cells from being attached to a slide, you're going to have to trypsinize them. That's going to cleave some of the glycoproteins, and that could alter your signal. So yeah, that's something to consider. Yeah. Yeah. Yeah, I mean, non-tryptic way, mechanically dissociating, you're probably going to get a lot of shearing. Yeah, but yeah, so it's something to consider. Yeah.
Yeah, thank you so much for the invitation to present. High throughput space-time, it's a method for single-cell metabolomics that our group is developing, and I also will talk a bit about CD40 cells. So I'm currently a postdoc at UCSD in the Alexandrov lab. We only moved to San Diego actually last summer, and before that we were working at EMBL in Heidelberg in Germany, and we mainly focus on developing techniques and methods for spatial and single-cell metabolomics. And why single-cell metabolomics, you might ask. So first, although metabolomics, it's quite understudied, when you compare to single-cell RNA-seq or proteomics, actually has a lot of potential because metabolites can directly impact function, the state, and the phenotype of cells. And then if we go into the single-cell level, we can even considering applying metabolomics to environments where metabolism is very important, tumor microenvironments or even in the immuno-oncology area. However, single-cell metabolomics is still growing. And if we just look into the number of publications we have been having in the past years, we see that right now single-cell metabolomics is where single-cell transcriptomics was 10 years ago. So there's still a long way to go. And this is what my group is trying to do. We're trying to kind of address the gaps that are currently, we currently have in spatial and single-cell metabolomics. And this is just a highlight of our recent, most recent work. As you can see, we cover a lot of areas from improving metabolite identification to study the best protocols for small molecule detection. And here I would like to highlight our main work is it's these single-cell metabolomics methods where we have improve the throughput and reproducibility. And this one, one of our most recent works, it's about studying lipid fluxes using single-cell metabolomics coupled with isotope tracing. And all of these data we are generating and results can only be possible with multi-imaging mass spectrometry, which you already heard about in the previous talks. So I'm not going to go into much detail about it, but just so that you know, we have a tissue section or cells. We have a laser that is scanning through our sample, and then for each one of these scanned ablated spots, we get a spectrum, and in our case, that spectrum contains hundreds of ions, and these can be converted into ion images. So for each one of the ions detected in our samples, we can have an image with a spatial distribution of this ion, also their relative intensity. And so this is where one of our main projects comes in. It's a Metaspace. It's a software for annotation of metabolite annotation, because as you can imagine, like you run multi-imaging mass spectrometry, you have hundreds of ions, and then you have the complicated task of annotating each one of them. And so we developed like this cloud software where you basically take the ion images like generated with single-cell metabolomics, and we match the ions to different databases according to like what suits like your data, if it's like you're looking for lipids or for metabolites. And then we give you like the possible metabolites for each one of the ions, along with some like FDR measures, so you know like how confident we are in these results. And this is like a free and open software, so anyone can join, use, upload their data sets, and some of the data sets are even public, but they don't have to be. Like you can have like your data sets private mode. And so after this, I'm going to now talk about high throughput Spatial, this method that we developed for single-cell metabolomics. So initially, we started by developing just Spatial, a method for spatial single-cell metabolomics. And in this method, we start by having some cells adhered to like glass slides. And this is a method that combines microscopy with multi-imaging mass spectrometry. So we have like these cells adhered to glass slides, and we do a round of microscopy where we get our cell morphology, so we can, and also like the general image of like our slide, so we can do segmentation. And then after this, on the same slide, on the same cells, we do multi-imaging. And this is followed by another round of microscopy where we can see exactly which areas of our slides were ablated by MALDI. And so by combining this initial microscopy with the MALDI imaging, like ion images, and like post-MALDI microscopy, we get the single-cell metabolite images, where for each cell in our slide that was ablated, we get the ions that were detected in that cell and also their relative intensity. So initially, Spatial was developed actually to detect lipids, and it had like what we consider like a low throughput, so we could only do 8 to 16 wells in a slide. And so recently, we have developed what we call now high throughput Spatial. So right now, we can have like 40 samples on a single slide. This could be multiple conditions and like respective like a technical replicates. For this, we actually use like some custom glass slides that we had to design and etch ourselves. And we also like have a custom software that we made like in-house, so we can like process all this data. And also like one of the main highlights of this high throughput Spatial is that we first we showed that can also be applied to metabolites or small molecules, so not just for lipids. And so in order to develop this high throughput Spatial method, we started by taking two cell lines, HeLa and this NIH. And this is what the cell lines that we use to develop the method. So we basically were able to detect around 135 ions in our like cells. And as we can see here, like the ions cover like the main metabolic classes. We have from amino acids to carbohydrates to like purines and pyrimidines. And like we detect between 20 to 70 like ions per cell. And also all these like ions or metabolites that we are detecting come from like very relevant like metabolic pathways. And also like one very important thing is that we can like differentially detect like these ions between the two cell lines. So it's not just about saying that we detected, we can also like quantify them, although it's always like, it's not the absolute quantification, it's relative, but still we can still like differentiate between the two cell lines. And in case you were wondering, like it's actually like a quite reproducible method, which is has been like one of the past few struggles, like in the single cell metabolomics like field. But here, like I'll show an example of the two cell lines that we used. And here we have like two replicates for each one of the cell lines, each one of these dots into one of the different metabolites. And we can see that like the coefficient of correlation between these metabolites is like quite high, like higher than like 0.97. And this actually, you can verify this for most of the replicates in our case study. We have like quite high correlation between all the replicates. And here, so when we talk about replicates, usually we talk about like wells that come from the same slide, but we can also like compare different slides. And here we can see that even like in this view map, that different slides also have high reproducibility. And finally, still talking about this high throughput SpaceM, this is like one of the first case studies we did. So we took these 9 cancer cell lines from the NCIH 60 panel, and we ran them through the high throughput SpaceM method. Here we actually detected more metabolites, probably because we were using like more cell types. So we detected around 200 metabolites. And we can see, like in the here in this view map, that high throughput SpaceM is able to distinguish very well between all the different cell lines. And besides that, since we are talking about metabolism, we can also find specific markers for each one of these conditions, and even some very here, some very markers for really small populations. But as you can see, it's only expressed in a fraction of cells, but it's quite intense. So it's truly like single cell level. And finally, I just wanted to now talk a bit about CD4 T cells, which is actually like my main project. So I started this project during my PhD at EMBL. And my goal was to take high throughput SpaceM and apply it to CD4 T cells. Also, this was already talked about this morning, how metabolism is very important for T cell function. So T cells, when they are in their naive state, they are considered to be metabolic quiescent. They basically rely on oxidative processes. However, upon activation, when the cells need to react to some foreign stimulus, they need to go through this metabolic shift, and this metabolic shift is really important for to sustain this activation, proliferation, differentiation, also all the effector functions that the cells need to do in order to fight whatever is going on in the body. And although immunometabolism is the field that basically studies this, how the metabolism regulates immune function, and is also a growing field. I think it's growing faster than single cell metabolomics, but there is still a lot to be explored. And in my project, I wanted to focus on single cell atlases of immune cell function, T cell function. So I was working with naive CD4 T cells that were isolated from the blood of healthy donors. And I was dividing them in different conditions. So I was either keeping them in their naive state or just activated them to simulate this shift that I have just talked about. But I was also, on top of that, modulating them with some compounds. So I tried to use 15 metabolic modulators, so 15 drugs that have metabolic targets. And on top of that, I also used 10 prescribed drugs that we don't know if they have any kind of metabolic effect. And after this, letting them grow for 3 days, I ran them through the whole high throughput SpaceM pipeline, and then I could generate a single cell atlas. So the idea was to take these 15 metabolic modulators, create an atlas, and then take these 10 drugs that we don't really know what is that metabolic effect, and kind of project them onto the atlas and see what is there more likely metabolic effects. Out of these 15 metabolic modulators, not all of them really worked well in T cells. So in the end, I was left with 7 metabolic modulators, but they still cover a big part of the main metabolic pathways. Like we have like from glycolysis inhibition to respiration to glutaminolysis inhibition. So we still have like it's a small atlas, but like has covers some of the most important pathways. And you can see here, here we can see like how some conditions like are really separated from the activating condition, which was our control. It's the one like in pink here. However, some conditions they didn't have such a strong effect, so they are not really like separating that well from the activated condition. So in order to like create a metabolic state for this atlas, I did supervised Leiden clustering. I thought this would be more robust than just taking the assigned conditions like and create the metabolic states. So I run Leiden. As you can see, most of the conditions they were assigned to one Leiden cluster, but again, all these conditions that don't have clearly like a strong effect when compared to activated, they still have like some kind of like mixed assignment of different Leiden clusters. But it was still possible to assign to each one of these Leiden clusters a metabolic state. And so this metabolic state was assigned based on the condition it was most overlapping with, and also like the markers that we found for that condition. So for example, here we have we have 2DG here. You see most of the cells like that were like treated with 2DG were just assigned to one specific cluster, and we renamed that cluster as glycolysis inhibition. And the idea to rename these clusters was just so then when we would do the projection of our test data, it would be easier to kind of infer what was the effect of those drugs. And that's what I'm going to show you now. So then we took all these drugs that are commonly prescribed to treat like some conditions, some diseases, and we took them, we run them also through the whole like high-throughput Spatial pipeline, and then after that we assign them to basically we try to see which one of these clusters of metabolic states were more similar to these different conditions. And here I just wanted to focus, we have plenty of them, but I just wanted to focus on baricitinib and tofacitinib. These drugs are commonly prescribed to treat autoimmune conditions, and they actually have a very similar target. So they target like CHUK proteins, and they are known to like inhibit the differentiation and proliferation of CD40 cells, but their metabolic effect is a bit unclear. But here we can see that actually they like show a very similar like metabolic state, and so, like a significant percentage of them, like around 20 to 30%, were assigned to a metabolic state of purine biosynthesis. Sorry. Which is quite very interesting, given that one of their like main effects is to inhibit the proliferation of CD40 cells. Here, I also just wanted to show how it looks like when you actually project them into the UMAP of our like single cell atlas. This is like how it looks like. Yeah. So finally, I hope I showed you that high-throughput Spatial is like a method that can detect both lipids and metabolites. It can also like differentially detect different cell types, and can be used like in monocultures and also co-cultures. I didn't focus on co-cultures, but in the original paper, like they do some co-culture of HeLa cells and this NIH. And it can also be applied to primary cells, as I showed, like specifically to CD40 cells, and kind of like provide metabolic states when they are treated with different compounds. And it can be, of course, it leads still a lot of improvement and more data, but it can be like used to build like single cell atlases of like metabolic states. And with that, just want to thank everyone in my team and thank you for your attention. Yeah.
Very nice talk, well presented. Two small questions. First, I know that you applied it to the CD4 T cells and tried to use the effect of the drug combination. Can you apply this like, is the technique so developed that I can I can take a tumor from a cancer type, I can digest the whole tumor and put all the cells to do this, or not yet?
Not yet. So we are actually now developing a way to also combine these with immunofluorescence, so that you can have like cells from multiple from different cell types, and then we can also apply Spatium, but to that. But we are still there. So for now, you basically need to have in each well, you need to have a different cell type. The way it's done now.
I can't use a pool of cells.
Not yet, but we are now we have a PhD student working on that. So he's developing a way to combine these with immunofluorescence, so you can have multiple cell types in one well. But we are still, yeah.
Do you think if I digest a tumor and I try to use flow cytometry or immunoprecipitation methods, maybe like just the pulling out the CD4 cells, CD8 cells, and putting them into different wells, and can I then do it?
Yeah, that would be possible. Yeah, that we can do it now.
Affinity based, I
Yeah.
can do it, or the flow cytometry. Second question, you use the CD4 T cells and tried to show the combination of the different drugs and tried to show the metabolic effect of these drugs on these CD4 positive cells, which I understand with respect to repression or activation. In the public domain, people have done a lot of single RNA sequencing on these CD4 positive or CD8 positive cells. Have you tried to use that data and correlate it with any of the known drugs that people are using and see whether there is a functional relevance to what you're doing?
Not yet. We have not tried that.
Thank you.
Yep.
The next speaker is Dr. Hannah McCullough from UCLA, presenting Dissecting the Evolution of Myeloid Leukemia of Down Syndrome using single cell and long-range spatial transcriptomics. But first, Lauren Wiggins from ONT will be introducing the platform.
All right, can everyone hear me? Perfect. All right, so I promised I would go fast so you get to the more exciting talk after me. But I wanted to talk quickly about using full-length sequencing for your single cell and spatial products with Nanopore. And we have a full support team that's all local, and we'd love to chat about any upcoming projects, any questions you have. We have a booth outside today and tomorrow, so feel free to swing by. And like I mentioned, I wanted to chat about why you might want to incorporate long reads into your single cell and spatial projects. And most simply, it's because of what you might miss from a short read view of the transcriptome. And essentially with short reads, what you're capturing is just a small chunk of one end of the transcript. So it's estimated that you might be missing up to 80% of the transcript information. And what does that account for when you're able to capture the full length? What do you gain? You can get things like alternative splicing, information on isoform switching, full-length TCR and BCR analysis, and fusion gene identification. And this information can be really important because up to 95% of human multi-exon genes are estimated to undergo alternative splicing, and around 15% of human hereditary diseases and cancers are also associated with alternative splicing. The type of information that you can get from either single cell or your spatial projects can look like this. So on the left, we are clustering full-length isoforms. So instead of a single cluster in a UMAP plot, you can get distinct isoform clusters. And on the right, we're looking at the mouse midbrain example from 10x Visium platform. And specifically, we're looking at two isoforms of SNAP25, and you can see that there's a pretty clear preference for the 201 isoform over the 202. And all of this information, both on the single cell and the spatial side, would be missed with only short read sequencing. We do have an end-to-end established workflow, including a bioinformatic pipeline. So it requires only 10 nanograms of input. It's a really straightforward, about 3-hour workflow. And you can expect about 80 million mapped reads per Promethean flow cell. And this is the instrument that they have in the core facility here. And what's interesting is that you can really use any samples that you've already generated as long as you've kept those full-length cDNA preps. So if you have these samples stored in your freezer and you want to go back and look at the full-length transcript information, you can do that and go straight into our library prep. We're also compatible with other technologies like parse, so it's not just strictly 10x as well. And again, we are happy to chat about any of your projects with the support of the core facility here, and I will pass it over.
Okay, so thank you so much for having me and my team here today, and thank you already in advance to your Genomics Core for getting us started with our work on single-cell long-read sequencing with the nanopore technology that we're very excited about incorporating into our studies. So today I want to talk about our journey to the omics methods through a specific project dissecting the origins and evolution of myeloid leukemia in Down syndrome using single-cell and long-read transcriptomics. So my long-standing goal in my career has been I need to cure childhood leukemia. And there's a lot of progress, but what does it really take to tackle this to every patient, every cell, every time? And I really want to say that it took the development of all the single-cell omics technologies that we can really look at these events in the individual cells where they are happening, and we can do this in human and not just in the mouse models. So in a simplistic model, what is leukemia? It is that a self-renewing cell, like a hematopoietic stem cell, gets locked into a self-renewing state and doesn't differentiate into all the beautiful blood cells that it supposed to be doing. Or it could be a more downstream progenitor that somehow hijacks part of the self-renewal machinery and now learns to self-renew instead of terminally differentiating, is now able to sustain self-renewal of a blast population throughout the lifetime. And for me, I wanted to cure leukemia, but when I went to med school and then started doing research, it was puzzling. I didn't know how the normal cells worked. How can you fix, how can you know how it's broken if you don't know how it should be working in the first place? And I think that has really taken us to the journey to the single-cell omics to really understand how is a human hematopoietic system built and how do these individual cells work together. So especially thinking about pediatric leukemias, it's very important to understand the ontogeny of the hematopoietic system, because we're finding that many of the leukemias actually have the pre-leukemic events that already happen in utero. And most of what we know about developmental hematopoiesis comes from mouse and zebrafish and adult model systems, and we've learned a lot from them. And what we've learned that hematopoiesis begins in waves. And the first waves that typically start in the extraembryonic tissues here shown in the mouse yolk sac, you generate these transient populations, transient red blood cells or macrophages that are meant to keep the embryo alive and developing, and some of them are sustained through adulthood, but you don't generate a real hematopoietic stem cell. The real hematopoietic stem cell comes in a later third definitive wave, comes from a hemogenic endothelium intermediate, and then seeds through the fetal liver and ultimately to the bone marrow. So if we think about the human developmental hematopoiesis, it's very similar in the basic layouts, but there's some very distinct differences that we need to be aware of. First of all, if we know that the first two waves start in the extraembryonic tissues, the extraembryonic tissues, the yolk sac and the placenta in the human macroscopically look completely different. So if we know how important the niche is, how do things really work in the same way in the mouse and human? We really need to answer these questions also in the human itself. The other important factor is the length of pregnancy, and we know that 9 months of pregnancy is longer than 3 weeks of pregnancy. But what people don't often think about, that it's not just stretching it for 3 weeks to 9 months, but when the mouse is born at 3 weeks, when you take the same developmental landmarks, the human is only at about 9 weeks of development. So we're barely through first trimester. So human gets incubated much longer. So now when we want to think about how to learn the instructions of making the perfect hematopoietic stem cell from pluripotent stem cell, or how to understand a leukemia that originates in utero, it's not easy to model this in the mouse, and we really need to kind of delve into the human and to the niches in human to really be able to understand these processes. So at the time when single cell RNA sequencing was coming, we had a very unique opportunity to collaborate with a group in Germany who had access to very early human developmental tissue, starting from 4 and 5 weeks of development at the time when hematopoietic stem cells are first born. So we created a map using 10x genomic single cell RNA sequencing. And this is compiling a lot of tissues and analyses together. But we found first the key goal is how do I even know if I'm working with the hematopoietic stem cell rather than one of those progenitor cells? So we went to the tissues where we know hematopoietic stem cells develop, and we identified a unique signature for the emerging hematopoietic stem cell. And we narrowed it down to these 6 genes, these 5 transcription factors, and the progenitor gene SPINK. And using this signature, we could find the real hematopoietic stem cells at any tissue during human development at any stage. So what we learned is that HSCs, first we can find them in the aorta gonad mesonephros region where they are born. Then we can find them in the extraembryonic tissues. They're actually circulating through the vitelline and umbilical vessels, and then back to the embryo, to the fetal liver, where they appear around 6 or 7 weeks of development, and then they get incubated there, but not just incubated. They're learning, they're maturing, they're gaining their functional abilities to do their adult job, to engraft into bone marrow, start taking signals from the niche, and adapt a more homeostatic state. And during this migration and maturation, HSCs are truly, they're developing, they're switching their lineage predisposition from a megakaryocyte erythroid biased HSC to a more multipotent HSC that also makes lymphoid cells. They become from a highly proliferative, highly metabolically active fetal type HSCs to a more homeostatic state. And all these things are important for their functional properties. So when we analyze these HSCs, we could find signatures of these key genes reflecting these processes that really shift dramatically when HSCs are transitioning from AGM through extraembryonic tissues, through fetal liver, maturing in the liver, and then in the umbilical cord. And why do we need this information? So the first immediate reason for that was that we wanted to compare what are we actually making from our pluripotent cell. At that time, nobody had made a transplantable hematopoietic stem cell, and we just didn't know how much in the boonies we are. So we collaborated with a group in Australia who have really spent their life in perfecting the protocols. And indeed, we found that their HSCs that they claimed are HSCs, they matched very closely to these nascent HSCs in the AGM stage. So they were on the right track. They just hadn't gone all the way through maturation. But by using the signal what we were learning from our single cell map, they were able to improve their protocol and now obtain transplantable HSCs from iPSCs, which is a big breakthrough in our field. And now what we're trying to do in my lab is really teach them how to go all the way in this developmental maturation through the fetal liver niches to the bone marrow niches, and really go through these developmental waves in culture. So what about our original claim that we want to get insights into leukemia? So I want to talk about one specific leukemia type, the myeloid leukemia of Down syndrome, that I think is both fascinating from the basic science perspective that really links the developmental hematopoiesis, and then as a kind of a model of a leukemia that originates in utero. So Down syndrome, or trisomy 21, the extra dose from the smallest human chromosome causes defects in multiple organ systems. And what's not commonly even talked about, that there is at least 150-fold increased risk of leukemia in the first years of life. And this contrasts dramatically to the highly reduced risk of other cancers in Down syndrome. So what really causes that? And they can be either myeloid or lymphoid leukemias. The myeloid leukemia always starts in utero. So there's something unique about the trisomy, the fetal environment, the fetal hematopoietic stem and progenitor, that all that together highly predisposes to this pre-leukemic state called transient abnormal myelopoiesis. So in all these trisomy 21 fetuses, the hematopoiesis is slightly abnormal. There's a expansion of progenitor population. There's defect in lymphopoiesis. There's too much of erythroid megakaryocyte cell, but nobody really had a finger on it, what is going on. But then we learned that as a newborns, 20 to 30% of Down syndrome neonates have acquired these GATA1 mutation that marks the first pre-leukemic stage of the myeloid leukemia of Down syndrome. So somewhere during fetal development, they acquire mutations in this GATA1 transcription factors, and this causes an expansion of these highly proliferative megakaryocytic blasts. And even if it's not a real leukemia, this alone can kill the patient. 10% of them can die, or they can have organ complications. So it's nothing to be just laughing about, even if it's not leukemia. Interestingly, this TAM phase typically resolves in the first few months, but in about 20-30% cases, a clone persists that then acquires additional mutations in cohesin complex regulators, epigenetic regulators, and signaling molecules, and now it transitions to a full myeloid leukemia of Down syndrome. But the key question was, how does the trisomy 21 trigger these hematopoietic defects in the fetal niches that predisposes to mutations specifically during fetal life? And then when, where, and why do these GATA1 mutations ignite that then initiate TAM? And what dictates the behavior of TAM? Sometimes the TAM is silent. You can only detect by very sensitive sequencing. Sometimes those blasts expand and can cause liver fibrosis and kill the patient. And then what causes the TAM to be transient? Is it just it's a progenitor that is at the end of its lifespan, or is it something related to the birth when the environment changes? And then what causes it to persist? And what changes at the cellular level when this transient progenitor, what does it acquire that it's now thriving in the postnatal niches? And then even though this leukemia is quite sensitive to chemotherapy, if there is a relapse, the prognosis is often very poor. So to get insights into all these stages, we acquired tissues from both fetal stages, from elective terminations, from these TAM patients as neonates, and then MLDS patients at diagnosis 1 month after treatment, and in some cases also after the relapse. And this illustrates our patient cohort. And if we just look at their MGG stainings, we can't really distinguish whether it's a TAM or MLDS. They look very similar blasts. So let's get some more insights from the single-cell RNA sequencing. We chose the 10x Genomics 5' single-cell RNA sequencing. We already knew that we are interested in isoforms that start from their own TSSCs, so we wanted to start from the 5' end, and also the new GEM-X kit that allows targeting 20,000 cells and was better also for more sensitive populations, sounded really very intriguing. So we mapped all these cell types to previous annotated data sets and identified clusters that map to either TAM blasts or MLDS blasts. And we can see that after treatment, this blast cluster disappears, comes back after relapse, and it's not really there in the control. So we felt like we had the ducks in a row to be able to study the right cells. So first question was what kind of blast are they really? We previously knew that there's a kind of a loose FAX phenotype, CD7 positive, CD33 positive, ITKA-L negative, that can identify TAM blasts and MLDS blasts, but it's not completely specific that you could use it 100% accuracy. So what are these blasts really? So again, we projected previously annotated data sets, and it suggested that both blasts most closely map to the megakaryocyte erythroid progenitor, to erythroblast, and sometimes to the EO mast cell progenitor. So not so surprising to what we had predicted what the potential cell of origin could be. But then when we compared these blasts to their normal counterparts, we were able to find both similarities as well as unique differences that really allowed us to define signatures that we can with high accuracy define whether this is a normal megakaryocyte erythroid progenitor or MLDS or TAM blast. And here specifically in MLDS, we can see induction of unique genes that are not really expressed anywhere else in the hematopoietic system. At this stage, there are many shared genes between TAM and MLDS, and then some that are very high in TAM, but then go down when progressing to MLDS. So looking at the individual patient level, we can see that these very highly specific MLDS genes, they are expressed in almost every patient. Here we have PRAME. It's a known cancer testis antigen, and it is very high in MLDS. GABA receptor 2, a neuronal gene gets induced surprisingly in the hematopoietic system along non-encoded RNA. Then some shared genes include IRX, Lin28B, a fetal gene, as well as some interesting surface molecules that may have role maybe in the ability for immune system to recognize these cells. So we're finding the signatures. So now that we know who are the blasts, how could we find where they come from? And when we mapped this FACS phenotype, we were able to find that in the fetal liver, even the euploid tissues had the same kind of a progenitor with the same FACS phenotype, but it was highly expanded in the T21 fetal livers. And when we mapped them in the in our single cell data, we can find that in the fetal liver, they also mainly match to the megakaryocyte erythroid progenitor. And this really suggests that this might be the cell of origin where the TAM first arises. And when we looked into our highly specific markers, especially the ones that are high in the TAM stage, such as IRX, CD70, and CD80, we could find that in this later second trimester livers, in the T21 livers, we can already see an induction of these markers. So we are now about to find out if these actually have already acquired mutations or if this is the 11th hour before they acquire the mutations. So what exactly is happening in this tissue? So the key first event is the GATA1 mutation. So let's look into this GATA1 mutation a little bit, because here the isoforms really start paying a difference. There are two isoforms of GATA, long and the short. And the mutations in MLDS and TAM, they always eliminate the long isoform and lead to only persistence of the short isoform. And this can either, physiologically, the short isoform comes through exon skipping of exon 2, which leads to that a second translation start site is being utilized. Here we can see the isoforms, the presence or absence of exon 2 in the normal case. So using the nanopore long-read sequencing, we were able to actually now for the first time really look at all the stages of erythropoiesis and see that as expected, GATA1, both of the isoforms come up in the MEP stage and then stay through erythropoiesis, but the short isoform goes down earlier than the long isoform as expected, and the long isoform completes the differentiation. So now we have an idea of the normal physiology. If we look at the mutations, we can see that there can be either splicing mutations that essentially eliminate the long isoform completely, or there can be stop codons here that at the protein level create a short isoform. So it's a little bit different now how we can follow these cells afterwards. So here's important, we're looking at, now we're using our single cell data to detect the GATA1 mutations. We can see here the stop codon in both our TAM and MLDS stage, but then the additional STAC2 mutation we had hard time seeing in the standard short read data. When we added the nanopore long read sequencing, we could now both see the GATA1 mutation, but also the STAC2 mutation in a much, much higher intensity. And we could now even follow that these mutated cells, both GATA, STAC2 mutated cells, they can even differentiate slightly beyond the blast cluster, giving us insights that we wouldn't have had if we couldn't track these mutations with high accuracy. Another way how we can track the molecular progression, we can't always find a perfect mutation to follow, is look at the copy numbers. And here we can see monosomy 8, trisomy 8 enriched, and here a ring chromosome 7 that matches perfectly in the relapse patients. So now we will be waiting for the next step to looking at the relapse samples and the PDX samples to see if there's clonal evolution and how the isoforms change. And then in the few final remarks, we go to why is this being triggered? Initially, we found that already at the HAC levels, things are going slightly array. They are more megakaryocyte biased. They are more immature. They express more highly chromosome 21 genes such as transcription factors, RUNX, and interferon alpha 2. They are highly metabolically active, and they expand these megakaryocyte erythroid progenitors. So why do they get then mutations? One interesting insight, again, from the long read is that we knew that MLD is associated with the isoform imbalance. The short RUNX1 isoform in particular is overexpressed in these leukemic cells. We can see now in our fetal livers that this short isoform of RUNX gets highly upregulated already at the HHC level and stays in the MAP. And what about the GATA1? This is where the mutations occur. When we looked at the long and short isoforms in the T21 and control livers, we found that there was actually shortage of the short isoform of GATA, and maybe suggesting that maybe the mutation that is looking in this is trying to rescue a deficiency. So this is just there for food for thought for the future. We found new things using our single-cell transcriptomics, new things about the distinct phases of both the predisposition, the initiation of transient abnormal myelopoiesis, and then transitions into the myeloid leukemia stage, unique signatures, unique isoforms, and unique ways of mapping the mutations as well as copy number variations. For future, we want to go deeper into these isoform imbalance. We've just scratched the surface. Want to really understand why things are happening in the fetal liver. Why is the fetal liver niche in particularly susceptible of fertile soil for these mutations to occur? And what are the external factors? Why 20 to 30% get these mutations? Is it hypoxia through dysfunctional placenta? Is it maternal infection? And finally, because we can't everything answer in vivo, we're using the pluripotent cell model where we can also interrogate the system. So a lot of more work to do, a lot more omics assays to incorporate long reads, short reads, spatial. So looking forward to working with all of you together. And my final 2 seconds. So key people, Stephanie, Yue, Paul from my lab who worked on this project, and really want to thank very, very highly from your Cedars Sinai Genomics Core, Ben Bastin, Casey Hanson, who have really helped us get this going, and the amazing support from Oxford Nanopore, Jay Mehta, Sean Evans, and Crystal, and also 10x Genomics providing us the baseline to get going. So I'll stop there. Not sure if there's any time for questions, but happy to answer.
Is about parents' age and the causal, cause of Down syndrome because of the aberrant eggs. Have you considered age as a factor in studying these approaches?
So very interesting point. Of course, we know that the maternal age is a very risk for the trisomy. Okay, but then once you have trisomy, 20 to 30% get the TEM. So there's something in utero, whether it is maternal infection, whether it's the maternal diet, whether it's the dysfunctional placenta. There's varying degree of maternal defects, and how then could maternal age be affecting these things, preeclampsia, other things. We think the mutations happen kind of a second trimester, late second trimester, around that time. So looking into maternal factors. So we are getting age, self-reported ethnicity from all our mothers, and we have little data at this point, but ultimately we're trying to understand these factors that could be playing a role.
such as UK Biobank or similar databases, could you potentially get more information? I'm not saying you would,
Yes,
but
I'm sure we would, and I'm sure people smarter than me have probably looked to certain degree, people who are more involved in the clinical and doing larger data sets. We haven't delved into it specifically, but certainly there are bigger data sets. Like we're more looking into the nitty-gritties of our few tissues. But I think very important things, because the best thing we can do is to stop that the ball never goes rolling. When you are there kind of tracking the relapse and things, you're a little bit too late in the game. But if we can understand those factors, that would be the best thing. Thank you.
Our next speaker is Vicki McCulloch-Bowden from Cell Signaling, presenting Uncover New Single Cell Biology with CST Intraseq Technology.
Thanks, you guys, for inviting us here from CST to talk to you today about our new technology, Intraseq from CST. Before getting into that, what I'd like to do, though, is just give you a brief interview of antibody validation at CST and why it's important, especially when we want to develop something new, a technique like our Intraseq technology. So I guess the first question is, do you trust your antibodies? And I'm sure a lot of you would give me the answer, no, I don't, because you've probably had a lot of troubles with them. Why is this important? Obviously, antibodies are a huge reagent, a huge tool in you guys performing your experiments. And the U.S. research antibodies market was estimated to be approximately $560 million in 2020. This represents a significant portion of the global research antibody market, which is expected to reach about $5.3 billion by 2027. That's a lot of antibodies. And there's about 1.6 million commercially available antibodies that target 96% of human proteins. But a significant portion of these antibodies do not recognize their intended targets, or they recognize nonspecific targets. And as you look at the sort of the top graphs here, you can kind of see as you get more complex in your applications, moving from Western blot to IP to IF, that fewer antibodies that are supposed to work in these applications are actually doing so. And this is estimated to cause approximately 375 million to 1.75 billion, which is a wide range, I know, wasted yearly on nonspecific antibodies. So that's why our antibody validation is important. And just sort of to kind of take home and prove that fact again, this is taken from a paper in 2018 in which the researchers just took 10 different antibodies from 10 that were commercially available against the NLRP3 target protein. And they're from different companies. They set up a set of criteria of what was important to them, specificity, sensitivity, and citations, which I know is important to you guys as well, to see which one best met their criteria. Well, what you expect to see is a band about here around this large size, about 118 kilodaltons. If you look at these, about 5 of these antibodies don't even give you anything. A lot of them give you a lot of nonspecific reactivity of some smaller size proteins. There are about 5 of them that do give you antibody reactivity against proteins at the right size, but some of them you might get nonspecific and the negative controls versus the positive controls. So when you're using your antibody, what is it actually recognizing if it's not recognizing the specific target it's supposed to? Well, when they looked at what antibody met their criteria the best, it's the one surrounded in the red box, and that happens to be a CST antibody. And I dare say that because of our antibody validation, that if you took 10 different antibodies from CST and compared them to other companies, I feel like we could finish in the top. So when you're designing your experiments, you need to be confident that your antibody will work as expected and detect your target unambiguously. Obviously, if it doesn't detect your target unambiguously, you might be thinking it does, looking at your data, trying to analyze it, and coming to conclusions based on something that's not specific to what you're looking at, not specific to your biology. It needs to work as expected, or you're going to spend a lot of time optimizing your experiments, which hopefully we can do in-house as an antibody provider. The way to ensure this is to make sure you partner with an antibody supplier that has knowledge and expertise to perform stringent validation of their antibodies. This is important because if you don't have the proper biological knowledge, don't know how to pick the correct positive and negative controls. You don't know how people are using this in the research and what applications they need. As a quick story, I once had a customer who had taken an antibody, used it in ChIP-seq. It's not one we validated in that application ourselves, but they did. And wanted to get a larger amount from us. It was a little, cost prohibitive for them at the time. So they went looking at other antibodies. And I wasn't real familiar with this target. I just did some searching, found only a handful of antibodies available out there. Not many had an application or images available on their web sheets, their data sheets. But when I found one that did, in the Western blot, they actually had a positive control, what we in-house use as a negative control. And I trust our scientists a lot more than I trust other scientists. So if they don't know the biology, then they really can't test the antibody for you. This must include a process in which all applications are validated independently. So I can't simply do this great degree of validation in Western blot and then go, do I see a signal if I look at IHC? I need to do the same amount of validation independently for each application. And this has to be a transparent process. You need to be able to look on our website or call us up and go, hey, how did you validate this antibody? And for us or any other company, so that we make sure that it meets your criteria. So how is antibody validation at CST different? And I alluded to, first of all, you need to kind of look at each application independently. And I think a lot of you guys think that when we make new antibodies, that we just kind of throw spaghetti at a wall, pull out our clones, test them in Western blot, and say, okay, what works? Then let's go test them in other applications. And we don't do that. We do each application independently. We do this through a set of framework, and this framework is pulled out of a paper from the David Rim Lab at Yale, in which we have different sets of, or different framework validation steps for each for our applications. And that includes our binary strategy, which is like an on-off switch. So it's your positive and your negative control. And a lot of people will look at knockout models in this binary strategy. And a lot of antibody companies, this is the least that they'll do, right? That you'll see the positive and negative controls and not much else. Or you look at a range strategy, and that will work for something like IF or IHC, in which we're looking at different tissues that have low, medium, and high expressing amounts of the protein that we're looking at. Our orthogonal strategy will look at data from things like proteomics or RNA expression in order to make sure that our antibody data meets that and gives us the same type of results. Because we're an antibody company, we often have many antibodies against the same target, right? I know that's confusing for you guys as well. But sometimes we have antibodies against different antigens, intracellular versus extracellular. So as I validate a new antibody target, I can use, if I have another antibody that recognizes the same protein and works in the same applications, I can just look to make sure they're just behaving the same. Our heterologous strategy allows us to look at a recombinant strategy. For example, if I'm looking at family members against the same similar proteins, making sure there's not cross-reactivity, or if there is, you're aware of it. And then complementary strategies, because we make a lot of antibodies against post-translational modifications. So we need to make sure that we're looking at antibodies that specifically recognize those modifications and not others, or not the total protein. But we go through that process, and then we pass and fail each application independently so that we can put out products that are going to work best for you. Now, this is a rather busy slide, but what I want you to take home from this is our experimental models. These are the tools in our toolbox, okay? And we use those for each strategy, for each application we're trying to validate. And the main take-home message should be there's not just two tools in our toolbox. We don't just look at positive, negative controls or knockouts. We're looking at other things as well. You can't build a house with just a hammer and a screwdriver. Other tools to be able to do so, and that's what we use in order to validate our antibodies. Now, when we're using our antibodies in an application like IntraSeq, we have to look at antibodies that are conjugated. In this case, we oligonucleotides. We want to make sure that our antibodies are working appropriately before and after the conjugation process. So in this example, we're taking an antibody against CD3. We're looking at the unconjugated clone in IHC, seeing it works appropriately. And this is just one of many different tissues we're looking at in this process. When we conjugate the oligo to it, we just want to make sure it works, again, the same way that the unconjugated. So we see the performance is comparable before and after conjugation. In this example, we're looking at a CD68 target in which we see that before our conjugation process, the unconjugated clone is specifically recognizing the parts of the tissue that we expect it to, But when we conjugate the oligonucleotide to it, you can see there's a lot of nonspecific binding. There's poor signal to noise ratio. So that's not an antibody we would use in any application, right? It's not going to be appropriate for something like a spatial biology platform or one of our kits like our Signal Star. Very responsive keyboard. So if we look here, an example of this is looking at flow validation of our antibodies. And again, if we're looking at a conjugated antibody, we have to compare the before and after. This is an example of what we'd use for our validated antibodies in that application. Here we're looking at keratin 818. On the left side of the panel, we're just looking at a secondary antibody, and this unconjugated primary, and we see when we look at our positive control compared to our negative controls that you get that shift because your antibody is specifically recognizing the protein in those cells, but not your negative control. When we come in and conjugate that directly and perform the flow cytometry, we get the same results with our positive and negative control. So we see similar performance. So this is something we can move forward to use in our technology. But in this particular example, looking at our IRF7 unconjugated clone compared to our conjugated clone, we see that, in fact, when we do the oligo conjugation, we're starting to see that we're getting some binding into the negative control. We don't see that shift that we see with our positive control on the left. So the conjugation has affected the performance of the antibody. So we're not going to use that in something like Intraseq or any other application. So I've alluded to Intraseq. What is it? Well, let's step back and think about, the history of single-cell transcriptomics. And CiteSeq is something that's been used for a while, looking at proteins that are on the cell surface of your surface of your cell. And once you take your antibody, conjugate a barcode to it, so that allows you to look at proteins on the surface of the cell, right? And that barcode depends on the platform that you're using. And it allows you to sort of phenotype your cells by looking at cell surface cells cluster into different types of cells. But what you can't do is look at what's happening inside the cell, because you have to be able to get the antibodies across the membrane, be able to look at the proteins that are inside the cell, obviously, if you're going to look at things like intracellular signaling. Unfortunately, a challenge of that is when you permeabilize the membrane, you're going to get RNA degradation and loss. So what our scientists at CST have done is developed our Intraseq technology, which stands for Intracellular Proteins and Transcriptomic Sequencing. And this is a kit in which we've essentially established developed a buffer, it's proprietary, which will protect your RNA from being degraded, okay? This was developed and validated by CST. It's using the 10x Genomics Chromium single-cell 3' reagent kits with feature barcoding technology, and you can use it on their 10x Genomic Chromium instruments. It's actually a very simple protocol. So what you would do is you would take your cells, you're going to do whatever treatment you're going to do with those cells, do some kind of induction with some kind of activating chemical, which causes some phosphorylation or pathways to be activated. You might want to come in with some kind of inhibitor, some kind of drug to see how that affects it. So then you can take those cells and you will then, it's a very simple process. You're going to fix them overnight. It's about 5 minutes of bench work. Fix them with our methanol fixation step. At that point, if you'd like, you can't get on the instrument. You can get into your cells and save them for 7 days before you move on with the process. The next step in your protocol is to use this SC block. This SC block is that proprietary buffer we developed at CST. It's optimized so that you obtain great quality single cell readout of both your RNA and your proteins, so you don't have as much degradation or loss of your RNA. Then you're going to come in with your antibodies that are conjugated to the 10x Genomics 3' oligonucleotides. And we have a kit that I'll show you the next couple of slides in which we have a cocktail with 31 antibodies. These are both human and mouse reactive. We also have 7 individual antibodies you can purchase that are strictly human reactive if you want to spike in, or you can buy any of those antibodies in the cocktail as individual, depending on how you want to mix and match and set up your experiment. You let that incubate overnight, again, just a few minutes of bench work. Let it incubate overnight and then come in the next day, do your washes, and that's the point where you're going to kind of take your cells, put them into the 10x Genomics Chromium workflow. So only a few days of work, an hour of bench work or hands-on work. This product has been released, it was released in September. So at this point, we have this, like I said, this one cocktail mix. We should be coming out with another one in the next month or so. And like I mentioned, it has those 10x feature barcodes. It is 31 CST primary antibodies that react with both mouse and human proteins, and validated by us on the 10x Genomics Chromium instruments. No matter what you use, whether it's the individual or the cocktail, you have to purchase the buffer kit. So our Intraseq 3' assay kit, that's going to have your buffers, that's going to have that SC block, your washes to be able to do your experiment. And then you can also buy the individual antibodies, whether they're ones that are in the cocktail or ones that are separate antibodies that only recognize the human proteins that are separate from the cocktail mix. So this is just an example of what's in cocktail mix 1. And you can see we have a number of surface proteins we're looking at. We have a number of intracellular targets here, which are a lot of them are immunological targets. And then we have many of our, what we call PTM related targets, and that we have the total protein and then a phospho-specific antibody that is in the kit. For example, we have, the STAT3, phosphostat3 tyrosine 705, phosphostat3 serine 727 that you can look at. We have some of the histone and phosphohistone and methylhistone, acetylhistone antibodies in the kit as well. We have our individual antibodies that are human reactive, that if you want to add those in or do them separately, you can. And that includes some of our cell surface markers like CD68, CD3, as well as a few intracellular targets and our isotype control. So that's what Intraseq is. That's what you can get right now. Like I said, we're coming out with new kits available in the next few months. And I just like to go over a few kind of proof of concepts to show you, okay, this does work as advertised. Here, for example, we're looking at our population of PBMCs. We're looking at TCF17 and NFAT1. And if you look on the left-hand side of the panel, you can look at the RNA expression. When we look at TCF17 cells, you can see in the different populations, your monocytes, your T cells, et cetera. T cells in particular, you can kind of see that expression in red, which shows high levels of expression of your RNA. But when you look at the things at the protein level, you get a more uniform expression and a more accurate representation of the proteins that are actually being expressed. And you can see there are other cell populations that do express a little bit of TCF1, TCF7. And if we're looking at NFAT1, it's just the same thing. If we look at our cell populations, in our monocytes, we're seeing a little bit of cells. In our NK cells, we're seeing a little bit of expression of RNA, that in those red cells in the population. But when you look at things at the protein level, you see that more uniform expression, especially in your NK cells. And now you can kind of see more expression in your B cells as well. So you're starting to see better expression and more accurate representation of what's happening in these cells. So that makes it better able to phenotype them, right? Because you can't always do that just looking at gene expression. And another example of a proof of concept, we've taken our CD8 cells. We're looking at the T cells here. If we separate those into different populations, we have our naive T cells, our memory cells, and our effector cells. Obviously not anything novel. But what we've then taken is looking at the RNA expression or the protein level expression of different targets that are found in our Intraseq kit. If you look at TCF RNA expression, again, you see the little red dots kind of spattered throughout the population of cells, but it's kind of hard to see what's really happening if there's a specific subpopulation that has expression of TCF7. But when we look at that at the protein level, we can then find that it mostly is in the naive T cell subset of the CD8 positive cells. So we see that better looking at the protein. If we want to look at TBX21, we get the same kind of results looking at only the RNA. We can see a very small smattering of those red cells throughout the population. But when you look at things at the protein level using Intraseq, you can see that you have a major representation of the TBX21 in the effector T cell subpopulation. Same if we look at CREB. We're looking at phospho-CREB on the protein side. If
Okay. Can, can they hear us right now? Okay.
We got a thumbs up. Wonderful. Okay, so hi everyone. So first thing I'd like to say is that intestinal epithelial biology is really my area of expertise. I'm not really much of a bioinformatics guy, and I think it's really good that I'm here because I'm an end user of the multi-omics, although I'm not sure if I'm allowed to say that. I think the name's been updated, but I really am a end user of this, so I came in relatively naive to it, and I must say that it has been really useful in my research. Before I go a step further, I really do want to thank members of the bioinformatics core here, Casey, Sarah, and Josie, for being really helpful in this process. Again, it is in my area of expertise, but they were able to make it really accessible to me. So I'm going to give an overview of the intestinal epithelium and the research question I was into, and then I will then go over the multi-omics results. So this is the intestinal epithelium, a schematic of it. So here you can see it really starts with the stem cells here in blue. So the way the system works is these stem cells divide, they turn into transit amplifying cells, and then eventually they turn into differentiated cells. Now, the most common differentiated cell is an enterocyte, which makes up about 80% of the intestinal epithelium. Then you have goblet cells, which secrete mucus, and these account for about 10%, and these are helpful for motility. And then you have about these purple cells here are enteroendocrine cells. These are incredibly rare. They're about 1% of the intestine. These secrete hormones and are sensors of the intestine. And then finally, in blue at the end, you have Paneth cells. So the cell types that I want you to remember are the Paneth cells, the enteroendocrine cells, and the goblet cells. So for a long time, this was really difficult to study in vitro. Most of the time we used a cell line called KCO2s, which were used to study intestinal permeability, but nobody could ever really study these differentiated cell types. And all of these are implicated in both health and disease, so it's really important that we can study these in vitro because this intestinal epithelium doesn't exist in a vacuum. You have the microbiome, which is, all of the microbes that reside in our intestines. They really heavily influence the functioning of this tissue. You have the mucosal immune system. So this is more of a cog in a wheel as opposed to just a cog on its own. So studying it in mice, etc., was fine. Knockout mice was all good, but you could never really figure out the intrinsic properties of this tissue. So all of this changed back in 2009 when Hans Clevers published his incredible paper, which showed that you could take the LGR5, i.e. the stem cells of the murine intestine, and you can make intestinal organoids. And back in the day, really all you needed was just a couple of markers. So here you had your goblet cells illustrated by MUC2, you had lysozyme, which is Paneth cells, chromogranin A, which was representative of enteroendocrine cells, and that was more or less it. Just as long as you could show one marker of the various differentiated cell types, that was more or less enough. And then, people just did simple stuff like EM and other various, one antibody at a time, just to prove that they had the cells. So just to give an overview of what I do, so my interest really is generating iPSC-derived intestinal organoids. They're slightly different to biopsy-derived organoids. In very simple terms, what I like to do is obtain blood from Crohn's disease patients that either have a very specific genotype or phenotype. This blood can be reprogrammed back to a stem cell-like state and then directed to form human intestinal organoids. Now, at the time, again, when I did this back in 2011, I think it was, I did the same as everyone else. Just used the same old markers, said, yeah, I've got intestinal organoids, I have enterocytes, goblet cells, enteroendocrine cells, Paneth cells. But that was really about it. Now, what this picture does not show is that this enteroendocrine cell was one cell, basically out of, 25 fields. So there was one enteroendocrine cell. This was going to be virtually impossible to study in vitro with only one cell. The Paneth cells, again, this was the best of the best. There was only a handful of these, so it was going to be so difficult to study these. Goblet cells a little bit more, but this was very variable. So I repeated this again. This was the standard at the time. All I was showing here was that whether it be from fibroblast or lymphoblastoid cell lines being the donor cells, had my own reasons for doing that. Again, just the 4 cell types. So how are we going to move on from this? So at the time, which was kind of in the vogue, was just to look for a very generalized response. So here I put interferon gamma onto the human intestinal organoids. This is microarray data back from 2015. But the take-home message here was that more or less the system works. So when I treated cells with interferon gamma, they clustered. All of the genes that you would expect would be upregulated. And this is what most of the people were doing. So if you had IL-22, I mean, take your pick of the cytokine of the microbes you were interested in, it was fine. But how were you going to study the intestinal epithelial subtype? So one more technical piece about iPSC-derived organoids is here. This is the intestinal epithelial tissue. The green is the mesenchymal cells. So there's 2 cell types in contrast to the biopsy-derived organoids. There's different percentages every time. So what we were able to do basically was, to cut a long story short, we were able to purify the epithelial component and serially passage it every 7 days. So this is what they look like. One thing I want to point out is developing this took about 9 to 10 months. It took forever to figure out the initial passaging strategy that would allow us to passage, for a long, long time. And so that was the first thing that we could do. We could really just pass these, but they were undifferentiated. There was virtually no Paneth cells, no enteroendocrine cells, no goblet cells, no anything. There was a paper came out in 2013 which showed that if you were to add in DAPT, which is a notch inhibitor, it would lead to the generation of the various secretory cells in the intestine. So to maintain them in an undifferentiated stem cell-like state, you would went on, notch on, which is more or less what we do. And then what we decided to do was just add DAPT and cheer. So these are the 2 things that we thought were really going to do it. Now, we did this without success for about 2 years, I would say. This protocol basically, in a way, was very, very difficult to reproduce. And again, they only did the most superficial, they did qPCR. Again, they did not look for the individual cell types. So what we decided to do was add in DAPT. Now, at the time, our sole research focus was on Paneth cells. We thought, right, okay, there's a whole host, and for our own reasons, because Paneth cells are highly implicated in Crohn's disease, this was all we were interested in. So basically, we added in DAPT, and eventually, as I say, after about 2 years, we finally figured out the culturing conditions. So by increasing the concentration of cheer and adding in DAPT, we now got about 25% positive lysozyme positive cells. So we were really happy about this. And of course, if you can do it once, you can do it 7 times. Again, we always got about 30% of Paneth cells. We went a little bit further. Again, we looked, all of the genes that you would expect to be upregulated in Paneth cells were, again, in comparison to the undifferentiated cells, always massively upregulated. So we knew we were in a good place. And again, we got some really nice images of human defensin 5 here being present in the organoid. So we thought, right, with all of this, we've done everything that we wanted to do. We've really shown it, qPCR, immunocytochemistry, right, now we're going to strike on and really do single cell seq in these cells. So this is really what the experimental overview was. It was 3 individual patients, or 3 individual control people. So we made their iPSCs, made their organoids all at the same time, purified their epithelial-only organoids, and added in DAPT. So in total, we added in 6 samples for the multi-omics. And once we did the initial analysis, well, here we could see that we had 2 clusters. One, this was from the undifferentiated group, and this was from the differentiated. So we had 2 blobs. So we were pretty happy about that. In terms of annotation, we got it from a CMGH paper. So what they'd done here was they had obtained samples from 3 donors from all of the different areas of the intestine. So from the duodenum, jejunum, ileum, ascending colon, descending colon, and transverse colon. So as many areas as they could, 3 individuals. And what you'll notice here is that from the total number of cells from the small intestine, they got about 6,500 cells. What I really want to direct your attention to is the number of Paneth cells that were ultimately obtained. From about 6,500 cells, they obtained a grand total of 49. Basically 8, 15, and 26. So in every single cell paper that I've looked at pertaining to this cell type, there is a negligible number of Paneth cells all of the time. So I don't know if they're not amenable to this type of technology, but there is some reason why they're never present. Now, unfortunately, and again, I have to give credit to the bioinformatics team, they tried so hard to find them. The Paneth cells, unfortunately, were not there. We looked. We confirmed via flow cytometry before the cells were ultimately sorted for this. We looked at them afterwards. Everything suggests that those Paneth cells were there at the time, but for some reason there was an issue. But of course, there's an upside. So when we looked closely at this and we looked at the clustering, well, if you remember, the top half is the undifferentiated cells, and this pretty much reflects what's going on in our system. Here you can see that we have epithelial stem cells illustrated in red right here, and this is the transit amplifying cells. So this is representative of the early undifferentiated organoids, and this is exactly what we would expect. Now down below in the differentiated cluster, here again you can see that you have some red cells and transit amplifying cells. This is what we expect, because when we have differentiated cells, it's not just that all of the cells are differentiated. There's always some stem cells present as well in TA cells. So this is like what we would expect. But what I want to draw your attention to is this very prominent cluster here, which we eventually found to be enteroendocrine cells. So this really kind of excited us, because this is a very, very difficult population of cells to. I hope that was the right thing to do. This is really difficult cells to study. So when we looked at closer at all of the clusters that we got, I'm going to draw your attention here. So when we looked at cluster 5, which was representative of this here, right, like so, here we could find this clustered almost perfectly to enteroendocrine cells and was only representative of this. And then we did find 3 clusters for goblet cells, like so. And then what we decided to do then is, because we had a very nice cluster here, I asked the team to go a little bit further and to increase the resolution. And one thing that I want to point out here is that these are all the different, these are the exact cells that were in the system. So if you look, SA here is green, purple, and light blue, and they are all present up here. So you're getting an even distribution of cells throughout. This is a fibroblast that we think occurred from EMT. But the take-home message is, what I want to draw your attention to is that we got a number of goblet cells and enteroendocrine cells. Now we had 2 populations. When we looked at the goblet cell subtyping with their increased resolution, we can see that we had a number of early goblet cells, immature goblet cells, etc. And here you can see these are the clusters associated with it, but you can see that in each of our experimental system, or in each of our adapt-treated cells, we did get a high number of cells in this. We're not just getting 2 or 3 cells, we're actually getting hundreds of cells in our system. The second thing is the enteroendocrine cells. As I mentioned, these are the hormone-producing cells. There's about 20 to 25 of these guys in the intestine, and they all have different jobs. So probably one of the most famous ones you guys hear about is Ozempic. So the L cells are responsive to this. And what we picked out from this is that we got a lot of enterochromaffin cells. So the purpose of these cells is to produce, these cells produce about 95% of the serotonin in our system. And the beauty of taking the multi-omic approach is that just getting enteroendocrine cells isn't enough. We have to know the exact cell type, because we have to know what assays. If we really wanted to study the effects of Ozempic in L cells in our system, we would get utterly nowhere, because that is not the cell types that we have. We have enterochromaffin cells, so the first thing that we're now going to do is start looking at serotonin release from these cells. And then the second cluster wasn't as strong, but this was very, very good for us. And again, we're not just getting 10 or 20 cells, we are getting a lot. So moving on to the ATAC-seq, this really is fresh off the, hot off the press, so to speak. We got a really nice cluster here, cluster 8 and cluster 13. Again, we only just got this. And what I want to draw your attention to is the number of transcription factors associated with this. You can see here, once we looked and checked, we can see optimization of the human intestine at a single cell resolution. Enteroendocrine and enterochromaffin cells, RFX2, 3, and 6 exhibited high expression of accessibility. Again, and we get this in our system. So what this is showing here, without looking for specifically enteroendocrine cells, without like with Paneth cells, we were really trying to find and really trying to like get there, looking for every bit. With the multi-omics, we just said, okay, what is the data showing us? And within 2 seconds of looking up this, it was very clear what we were having. So in conclusion, we started off with DAPT, looking for Paneth cells in our system. We got it. Unfortunately, it didn't work out. But now my lab has an entire new line of inquiry, because when you're looking for apples, you're not looking for oranges. And well, we actually found pineapples in our system. So, but again, I think the power is you can't look for everything in a system. You can't look for, you know, when you're trying so hard to get Paneth cells, you can't be looking at other markers. Otherwise, you're never going to get anywhere. So the fact that this system has very clearly shown us what we have, again, has utterly, you know, changed the trajectory of our lab. So with that, I really would like to thank my lab. Sachi worked incredibly hard. She spent 2 years really developing the system, and Monica came on board and has really assisted her. This is my lab. And again, I would really like to thank the bioinformatics core, Casey, Josie, and Sarah for being really helpful as well. So with that, happy to take any questions. Yeah. Yeah, I think that's exactly what happened. But I mean, you know, one thing that maybe actually that I didn't mean to say in the talk is that we spent about 6 months working with Chinta to optimize this system in terms of trying to get the correct permeability, etc. And you know, we didn't look to see specifically what cells were becoming permeable. So yeah, these cells are larger, and we probably learned the hard way. But I mean, you know, one thing to do this is you need a really high live pure population. So what we did was we dissociated these guys for about 5 to 10 minutes, and then we sorted them for nearly an hour and a half with a ghost dye to ensure that we were getting no dead cells. And I think it was probably just too much for the Paneth cells ultimately. And again, yeah, you know, it's just the way it is. We found enterochromaffin cells, so I mean, it would have been nice if we could have gotten both, but I mean, again, I think I'm here really to speak about the realities of the system. Again, it did take 6 months to optimize this, and it was still, you know, this is a complicated technology. So, I mean, we got it. And I guess one other thing I want to say is some of these cells were immature, but this goes back to the realities of the system. What we're comparing these things to are in vivo, where all of these cell types are seeing the microbiome, they're seeing the mucosal immune system, they're seeing, you know, a whole host of other things. So expecting these to fully recapitulate the human, you know, fully developed, adult, mature in vivo state is probably a little bit unrealistic. But I think, you know, this is just a really good system, and it showed us the one cell type that is probably as close in vivo as you can probably get, I think. So yeah, it was good. Okay, thanks, guys.
The next speaker is David Rinco Fernandez Pacheco from Cedars. presenting multiomic analysis of pediatric brain tumor MADR models uncover a novel treatment approach. Prior, Arcana Beta Dapor from Scale Biosciences will be talking about the technology.
Great. I'll get started. Hi, I'm Archana. I'm a product manager at Scale Biosciences, and happy to tell you a little bit more about Scale and our single-cell methylation product. Methods have been developed in single-cell genomics to assay across the spectrum of biology, from the genome, epigenome, transcriptome, and proteome. But measuring these modalities in small subsets or small sample numbers places a ceiling on the next level of insight for greater impact. Scale's mission is to get that next level of scale for cells and samples across more ohms for deeper insight into biology. Researchers have realized the need for single-cell granularity complemented by high sample throughput and easier sample preparation in a number of research areas to enable longitudinal studies, patient cohort diversity, rare cell population characterization, and training for machine learning models. Scale's massively parallelized barcoding technology allows researchers to get high-quality single-cell data with flexibility in workflow and ability to scale up cells and samples, which is actively being expanded to new modalities. And single-cell methylation is an example of that product innovation. As a recent entrant into the single-cell sequencing space, we are keeping a high pace of innovation. Within the past few years, we have launched multiple products to enable researchers and plan to further expand our portfolio with impactful and differentiated products. In this talk today, we will be discussing single-cell methylation, which you can see on the right here. Now I'll give a little overview of our single-cell methylation kit and workflow. We are excited to enable single-cell DNA methylation as it is a well-studied epigenetic mark, typically done in bulk or arrays today. And this is the first commercially available methylation kit on the market. Our technology enables single-cell methylation by combining bisulfite conversion with Scale Bio's massively parallelized barcoding, which uses the cell as the compartment. Fixation and permeabilization-enabled cells and nuclei are used as the reaction vessel. Sequential addition of short DNA tags to the analyte of interest creates a unique index per cell. Combinations of indices and numbers of cells scale exponentially, removing the limitations of Poisson distribution. This allows us to vastly increase the throughput of single-cell methylation compared to homebrew academic methods. We put a significant amount of development work into making our workflows as streamlined and user-friendly as possible from start to finish. The fixation that enables our massively parallelized barcoding also allows for experimental flexibility, where samples can be stored and shipped on dry ice after locking in your biology. Our workflow is plate-based and reagents are pre-stamped to minimize pipetting. This eliminates the need for specialized instrumentation and enables broader compatibility with automation. We've also incorporated smart plastics for ease of use with the pooling funnel, which removes the need to do pooling steps with a multi-channel pipette. You simply flip over the 96 or 384 well plate into the funnel and give it a gentle spin down in a centrifuge. Prepared libraries are compatible with Illumina sequencers, and all of our workflows have fully supported bioinformatic pipelines to process your sequencing data, leveraging cloud-based platforms and compatibility with advanced downstream analysis tools. As mentioned, our plate-based workflows also lend themselves to being very automation-friendly. Automating our single-cell methylation workflows significantly decreases hands-on time while maintaining consistent, high-quality data, identifying the same cell type heterogeneity across complex cell populations. In addition to the kits themselves, we also provide the option to run samples in our in-house services, which can be proof of principle or even whole projects, depending on the resources available in the lab. And lastly, our website is a great place to go to get more resources. If you have any questions about single-cell methylation, there's more information on our products, technical notes, webinars, and access to product support. Thank you.
Hello. Okay. Should I start from the beginning or just keep going? I mean, I'm just talking about the tumor heterogeneity and how the different tumors are extremely heterogeneous in the brain, and also inside of the same tumor you have extreme heterogeneity. Also, I work specifically in pediatric brain tumors. Until now, they have been always considered as part of the brain tumor. They weren't taking a lot of consideration, but the World Health Organization in the last classification, they finally separate these tumors as pediatric brain tumors in an independent class of tumor, because they are really, really difficult. This is really, really sensitive. I cannot put even my hand inside. So pediatric brain tumors are extremely different to the adult tumors, so we really need to model these things properly. So that's what my lab started doing, well, the lab I work in. For this, we developed this technology, which we call MADER, as mosaic analysis by dual recombinase mediated cassette exchange. And basically what we are doing is taking advantage of a Cree reporter mouse, which is here, you can see here the rosatonesis locus in the MTNG mouse. And the way this mouse works is just a Cree reporter. So what you usually have is you're gonna cross it with a mouse that expresses Cree in any kind of tissue. And when Cree appears here, it's gonna remove the TD tomato expression, so these mouse are, all their cells are red. But when you have a Cree expression, this TD tomato is gonna disappear and they are gonna become green. So the good thing is that usually when you make a mouse, an engineered mouse, you need to purify those cells. And for that, you need another cassette, which usually it has FRT site for the flipase. So if you add not only Cree, but also flipase, and you put another plasmid that have also those two recombination sites, then you can do an actual recombination from a plasmid to your genome in a specific locus. And that's what we do. We just add flipase, Cree recombinase, and we add a plasmid that is going to have anything we want to put inside of those mouse, and they will just swap those cassettes, and you will have cells that have removed the whole Cree reporter system and just now have expression of whatever you want by the endogenous promoter. And this is a really good thing, because then you are not using viruses or transposon, things that are going to give you variable expression and supraphysiological expression. You are actually having one copy of your genes, which is what you usually have in your genome. So using this technology, we have, this is myself, we have generated already several different types of tumors. The first three are the ones that I'm going to be talking mainly, which are pediatric brain tumor, diffuse pediatric brain tumors, gliomas. And they are basically different by a single nucleotide, in fact, single amino acid, which are tumors that have a K27M mutation in the histone tree, a G34 mutation, or they don't have mutation in the histone tree. Working with this is the model that we use. This is our P donor, our plasmid donor of the mutation. So we have this histone tree, then we are adding the two secondary targets that are mainly found in this tumor, which are PDGFRA mutation or amplification and p53 knockout. So using this system, we can just inject in pups, in newborn pups, and electroporate them in the ventricles. And then after some time, they will grow tumor that we have already characterized are extremely similar to the one we find in human. K27M mainly grow in the midbrain and in the striatum. G34R tumors grow in the cortex and the corpus callosum, and wild type show like different a mixture of phenotypes. Survival curves show that they do the same thing that you see in human. K27M are really fast growing tumor that usually reach morbidity in child in 5 years old. G34 are a lot slower, and usually you find this tumor growing in teenagers and even young adults, even if it's a pediatric brain tumor. And wild type are even slower because they don't have the mutation in the histone tree, so they don't have that oncogene. So our idea was, okay, let's look at this tumor and see how they look like a single cell level. So we have used single cell RNA-seq, single nucleus ATAC-seq. We look at the DNA methylation and use all those thousands of different packages that we have now for analyzing. And this is like a first image of how our tumors look like. This is a mix of the K27M, G34, and wild type. And you can see how we have our tumoral mass here, which have typical cyclin cells, oligo-like cells, the OPC-like, which are like the less differentiated cells, astrocytic-like, and then we have all those stromal tumor environmental cell, like the microglia, immune cells, endothelial cells, all these kind of things. And you can see here in all these plots the marker that we tend to use. All the programs are coming from papers that have already set transcriptional profiles, and then we have all the marker cells here. So the good thing with our system is that we are actually labeling the cells and modifying a gene. So we can actually look at that gene in the transcriptional profile, because we are doing single-cell RNA-seq here. This is single-cell RNA-seq short read. So we can actually see what is a tumor and what is not a tumor. We will have the endogenous normal cassette. And what was striking to me is that these three tumors are different. They grow in different regions. They grow at different time points. But at single-cell level, at short-read single-cell level, they look the same, at least at the general view. When you have them, they completely overlap, which is a little bit crazy. So the next thing I did is to, okay, let's look how the different transcriptional program that I can find here. So I just took the tumor. This is only the tumoral mass. You can see how they are overlap. And I use RNA velocity. It's a little bit controversial, the technology right now, to actually say how the transcriptomic profiles are going to change, but you can use it for finding stable transcriptional profiles. And that's what I did, trying to find like different cell types, tumoral cell types that could be inside. So I found these different regions, these different spots that are like highly stable transcriptional profile. I figured out what they are, and now I have the different cell types. I use this to try to figure it out if the different tumors have different percentage of cells, and I found some differences. I found a cluster that is really high in heat shock protein and ribosomal proteins genes, and that seems to be higher in G34R cells as well as myeloid-like. These one are the heat shock protein, and the myeloid-like are here. It's not as high, but it's still different. There are some subtle differences, but not as many as you should expect. So we went to the single nuclei ataxic. Just look at the epigenomics. And at least when we did this, it was even worse because you have a more sparse matrix. We have done some other analysis that I am not adding here because I don't have the time for that. But the general image is like the tumor is just a ball. You don't have a lot of differences at the ATAC level. So we decided to look for methylation because histone 3 is a marker for different kind of epigenetic modulation. So we look at the methylation of the DNA. So the first approach that we did was using bulk. So we used this methylation array that has been already validated, that is for mouse. And for this, what we did is using organoid, because with our technology, you can develop your tumor, then take them and put them in culture as spheroids. And those spheroids we have validated at single cell level that are still matching the primary tumor. So we use this organoid and we can culture them over time. So we are losing the non-tumoral cell because tumoral cells are going to overgrow anything. So you're going to keep your tumoral cell, but you're going to be losing any other cell that could contaminate your bulk RNA, DNA methylation. And with this, what is funny, even if they are single cell RNA-seq level, almost identical, they are really different at the methylation level, at bulk DNA methylation level. And this matches biology because K27M tumor are the most hypomethylated tumors, which is already described, is something that happened, while the G34 are the less methylated tumor at DNA level. So we look at this transcriptional profile. I can extract, these are the probes from the RNA methylation, but I can extract the genes that are being actually affected by this methylation. And I took all the hypomethylated signature, and I created a transcriptional program from those. And when I look at those transcriptional programs, you can see in the dot plot here that they match in the single cell RNA-seq dataset. So I took these transcriptional profiles and go to the single cell, and you can see how they clearly match the tumors, the specific signature. So even if they are overlapping, they have these differences. And how these signatures are in different regions of the single cell. So they are matching different cell types within this tumoral mass. Looking into this signature, I decided to do a pathway analysis, and I found that they have different possible targets. G34, I think, is maybe too small, but G34 shows a significant difference in the arginine and proline metabolism, while the K27M show hyposignaling pathway alter, and the wild type show like JAK stack signaling pathway. I'm working in all these three. But here today I'm going to be talking more about the G34R. So you can see in the dot plot below that there was a high suppression of the genes in this arginine and proline signature pathway, aside of some genes that are highly overexpressed compared to the other, but is clearly perturbed compared to the others. We also validated this model with human data, and this slide I'm going to go fast, but it's basically how our models are overlapping with human data set that are available on the bibliography and how the programs match together. And in human, we find even more suppressed this pathway, the arginine and proline pathway. If you look our tumors compared to the human, they are extremely similar. And I have been adding all the data that we have right now. And you can see on the left how the mother tumors are matching the G34R, K27N, and wild-type tumor. The K27N human tumors are a little bit different because are not specifically the same tumor we are actually modeling. It will be this one here will be the one that we actually modeling, and you can see how it's really more similar in this region here, the GBN high. And as I say, these transcriptional profiles still clearly match what we find in the methylation layer. It really matches the transcriptional profile in the human tumors. So we decided to explore this pathway further, and we found that there is a suppression in two enzymes that are responsible of making arginine. Normally in your body, you don't need to make arginine or your cells. The liver is going to release arginine into the system, and your cells can take it from there, from the blood supply, but they are able to make their own arginine. The thing is that these tumors have suppressed the two enzymes responsible of making arginine, so they need the blood supply of arginine to grow. So we decided to try to make a treatment for this. I think my slides are changed because this order is, well, that doesn't matter. So, and there is actually an arginine dehiminase, which actually what does is you can put it in the bloodstream and it will degrade arginine. So now all your cells need to make their own arginine, and the tumor can't, so it should be doing an effect on them. So I use our model to check this, and you can see how the G34R tumors are really, really dependent on the arginine. You can see how when we add the arginine dehiminase, the cell death really skyrocket compared to the other, which don't change against the control. And the next step was, okay, let's look at the single cell level methylation, because is this methylation general in the cell? We saw that there are some differences in the single cell level at this transcriptional profile, but we want to see if this differential methylation is changing in all the cells in the same way, if it's an homogeneous changing, or is something that is heterogeneous and is going to affect any kind of treatment that we want to do. So for that, we use a scale single cell methylation analysis, and you can see here the preliminary data. We just did a small kit for having a first test of concept. And I just want to show this is how it looked with single cell RNA-seq, how our tumor looked like, and this is how now our tumor look with single cell methylation. So our three tumors are clearly getting separated. So they are not overlapping anymore. There are differences in the coverage distribution, in the global methylation of CpG island. And if we focus in the G34R tumor, which are here in red, and we reanalyze them independently, we looked at these two genes that are responsible of making arginine, and you can see that they are not homogeneously altered in methylation. You can see that there are some clusters like the cluster 6 that is specifically methylating the ASL gene, but doesn't have such a high methylation in the AAS1, which are the two different enzymes that we are talking here. So with all of this, we have started already a preclinical trial to try to test this arginine dehiminase in mice, and I'm just going to show some of the results. This is how we are doing it. We are just electroporating mice to make our models, and when they have started developing the tumor, we will start all this treatment trying to validate the arginine dehiminase as a treatment for pediatric gliomas. So we are doing all of this characterization right now, but I have some data about the survival curve. And you can see that while if you don't add ADPEC20, there are no significant differences. While we are starting, and this is still running. I have like another 80 mice running during this survival course, so we are still finding results, but we are already having a trend in differentiating between the different treatments that we don't find when you don't add adipec-20. And interestingly enough, adipec-20 doesn't do anything by itself, but it improves the effect of other treatment, like the chemotherapeutic temozolomide plus lomustine, or the radiotherapy effect. So we are finding that this arginine starvation is actually an effect that can improve other therapies more than being a therapy by itself. With that, I just want to say what we are going to be doing and why I think it's important to do all these things. You don't want to focus in just one technology. You need to look at all of them together because they are going to give you a fuller picture. And some of the technologies are not going to show you everything. So now we are looking to do single-cell RNA-seq, but we are going to be looking at not only short read, but also long read. We are going to be looking at ATAC-seq, even if for the tumors it's more complicated because tumors are crazy in the genome. For other cell types, it's incredibly interesting to look at them. And finally, single-cell methylation. I think single-cell methylation is one of these things that you really need to do if you work in cancer, because we are seeing more and more that methylation is one of the biggest drivers of cancer progression. And with that, I just want to thank everybody, and I will ask any question. Thank you.
Very nice talk. Just the last part. It was very interesting to see. Hi, I'm Colin. I'm representing Atlas Omics. I lead the product development team. We are a up-and-coming company trying to bring new tools for spatial epigenomics. And so many different technologies have been discussed today, and I'll just jump right into it. We have spatial ATAC-seq and spatial contact to look at open chromatin in tissue, as well as histone modifications and chromatin-associated proteins. So we have the ability to look at the cellular level, doing a 5.5 by 5.5 millimeter region, and we can work with a very wide range of tissues. And we have a genome-wide analysis. For our contact assay, we can look at a wide variety of post-translational modifications, as well as some chromatin-associated proteins such as Pol2 and CTCF to look at 3D chromatin. So we are working constantly to make a wide variety of epigenome tools available for spatial context. We put in a lot of work from the original academic technology, where we had our original Gen 1 chip only looked at a 2.5 by 2.5 millimeter region. And with significant work, we've vastly improved the throughput as well as sensitivity of the platform to look at epigenome changes at the single cell level. We've developed a kit and platform that's set up at Cedars Sinai, as well as a data bioinformatics platform to make it easy to work through very complicated epigenome data sets. We have our process, we take a little bit of different approach to spatial biology. We do our entire work on a tissue section on a standard 1x3 inch slide, where all the chemistry is done in situ. That allows us to do epigenome analysis. Specifically with ATAC-seq, we run the tagmentation reaction within the tissue, keeping the tissue context intact. And then we use 2 microfluidics chips to create a coordinate system within the tissue context. So an X chip comes down that we hybridize spatial barcodes to the tagmented fragments. And then we'll take a second chip to do an orthogonal ligation to create XY barcodes in a coordinate system within the tissue. From there, we'll image the tissue, lyse it, and recreate a spatial map after sequencing. And we've made this quite simple to work with, where all of the chemistry and biology is done in a well over a tissue. So tissue is put onto slide, put it in our apparatus, and a user can just pipette in, add a spatial barcoding, and then do the library preparation as standard single cell workflows. And we're compatible with a wide range of instruments
And like I said, we have a cloud platform that seems like most of the tools discussed today has some version of this and makes it a lot easier to use. We've worked with a very wide range of tissue types, pretty much everything. Where our bread and butter has been cancer, where we've had significant success over pretty much every cancer type, still limited to fresh frozen tissue, but we're able to get very good differentiation and identification of cell types from the epigenome perspective. And we've worked with almost every species and tissue type that you can think of, we've had success with. And to finish this up, and I want to make sure there's time for Dr. Falcone to talk about her data. We have worked with over 70 different labs at this point with a wide range of success in cancer and neurodegeneration, pain pathways, aging, where we've seen how epigenome changes can drive disease and function. And some of these preprints are all coming out right now, but it's pretty exciting stuff. And I'll lead that off to Dr. Falcone. We'll talk about her work looking at prostate cancer, looking at chromatin accessibility. So thank you, and I'll pass it on. Yeah, she's virtual.
Hi, Dr. Falcone, if you can share your screen.
Hello, can you hear me?
Yes, we can hear you.
Okay, and can you see the slides?
Yep.
Okay, perfect. So, hello everyone. First of all, I would like to thank the organizer to give me the opportunity to be here. And today I will go over about what we have been doing at the Lee Ellis lab at CPDR, exploring spatial biology, specifically using the ATAC-seq technology in collaboration with Atlas Exomics. As you have already heard, I will be focused in my talk in prostate cancer. And the reason why we are in the lab focusing in the study of this type of cancer is because this cancer is presents the highest incidence and also is the second cause of death in men. Moreover, if we talk about racial disparities, we can see that Black patients are the ones that present the highest rate, and also upon diagnosis, these patients present really aggressive tumors. And our objective is to know why this is happening. So before to go to the data, I want to introduce one of the concepts that we are going to be discussing over the full presentation, that is phenotypic plasticity. So this is a mechanism that has been included in the hallmark of cancer recently, and it's a therapeutic challenge. So a cancer cell that undergo phenotypic plasticity is changing the phenotype, and this is happening with a modification in the environment. So in the case of prostate, in general, the tissue is formed by different glands. These glands are compounded by epithelial cells that are subclassified in luminal and basal cells. So under development in prostate, a stem cell is going to express AR and will become a luminal cell. But in the case of prostate cancer, these luminal cells will start at the beginning to express high levels of AR. But at some point, these cells will transdifferentiate. They will start to decrease the expression of AR, and also they can present other alterations. And these luminal cells will present a new stage that is a multilineage state. So this phenomenon has become really important in clinic because over the years, we can see that prostate cancer patients, initially the standard of care is based on surgery, radiation, or androgen deprivation therapies, but many of the patients that are undergoing all of these type of therapies can relapse. And patients that present relapse, many of them present characteristics of phenotypic plasticity. So these tumors many times can have low level of expression of AR, but also can present alterations in RB1P53 and also increased expression of epigenetic markers. So in general, the take home of this slide is that with the introduction of new generation androgen deprivation therapies, the percentage of patients that present relapse and present characteristics of phenotypic plasticity has increased in the last years. So considering this situation, there were a lot of different labs that decided to study this phenotypic plasticity situation using different models to mimic the castration-resistant prostate cancer stage. So here you can see some results from Ellie's lab, but also Sawyer's lab that study in detail the epithelial compartment of these in mouse models of these tumors that resemble the castration-resistant prostate cancer situation. Moreover, some data unpublished from our lab show that if we take these tumors and we treat them with an epigenetic drug, in this case an EZH2 inhibitor, we are able to reshape these cells and make these castration-resistant tumors become a more adenocarcinoma state because these cells are able to express again AR. So in summary, with all of this data, what we can say is that when we are in a situation of a castration-resistant prostate cancer, we have tumors that present alterations in PTEN, RB1, and P53. Also, these tumors present an increase of EZH2. If we treat these tumors with an EZH2 inhibitor, we are able to induce a cell transition, and these tumors become more an adenocarcinoma state. What is more important for the talk of today is that there are some high-risk primary prostate cancer that has been reported that also present alterations in PTEN, RB1, and P53. These high-risk tumors also present worse overall survival. And there are, more interestingly, some last publications that they have been using and doing neoadjuvant studies in which patients treated with androgen deprivation therapy were divided into groups, responders and non-responders. And the ones that were not responding to the therapy were the ones that were presenting PTEN and P53 alterations. So this could be associated with the potential of these primary tumors that are undergoing already a process of phenotypic plasticity. Moreover, I just want to highlight that if we compare white patients and black patients with prostate cancer, in black patients, we see that these tumors present higher levels of EZH2. Also are characterized by an immunosuppressive tumoral microenvironment, and as I said before, more aggressive tumors. So considering all of this, we wanted, the main objective of our project was try to have an idea if phenotypic plasticity is present in primary prostate cancer. So we have high-risk primary tumors. These tumors can have high levels of EZH2. So this led us to hypothesize that these tumors can be primed to have an epigenetic reprogramming. And the specific sub-questions that we established were, are black patients' tumors more aggressive due to an increase in chromatin accessibility? Are we able to detect different master regulator transcription factors that can be specific of race? And finally, we wanted also to understand the proximal and distal relationship between the tumor and the tumoral microenvironment. So considering all of these questions, we decided to apply a spatial epigenomic profiling based on the publication in 2022 of the Dr. Ron Fan. So in our case, we decided to select a cohort of patients with 10 patients, 5 black and 5 white. In the slide, you can see the different clinical characteristics of the patients that we included in our study. And in the down part of the slide, the workflow that just Colin described really in detail, I will not go through it, but was performed by Atlas Omics. So we process our samples, and now what? So first of all, we did our quality test to see how our samples perform the experiment. So in this slide, you can see that represented the different fragment counts distribution that we could see in our tissue spatially. But also in the right part, you can see two graphs that represent the fragment size of all the samples that we analyze, and also the distance from the transcription starting site. So these values give us confidence to go deeper and continue with the downstream analysis. So our first question was, are we able to identify phenotypic plasticity at early in disease? So to answer this question, we went to the literature because Dana Pierre recently published a paper based on pancreatic tumors in which she described that the cells that are presenting high plastic states are the ones that also present high chromatin accessibility. So considering this definition, we went back and we saw the total number of peaks that we were able to identify in our two groups of interest, black and white patients. And we saw that black patients were having higher number of peaks. So with this, we decided to continue deeper in the analysis of our samples to see if we were able to see different features in the chromatin landscape between these two groups. So using the chromatin state, we were able to identify different clusters, as you can see in this hue map. In the bar plot, you can see the representation of the percentage of the cell proportion that were associated with each cluster in white and black patients. And also you can see the spatial distribution of all of our clusters. We were really happy to see that spatially we were able to have a high level resolution of the structure of our tissues when we were comparing these maps with the hematoxylin and eosin staining of the samples that we were analyzing by ATAC-seq. So, okay, great. We have all of these clusters, but what are all of these cells? So we needed to perform cell annotation. And to do that, we decided to choose two different strategies. First of all, we use a cell kano that is an algorithm that is doing an supervised analysis using the peaks of the ATAC-seq and using a single cell ATAC-seq database as reference. So using this algorithm, we were able to see that in our samples, we were able to clearly separate the epithelial cells versus the tumoral microenvironment. We try to go deeper in the cell annotation, and we perform more bias analysis. So in this case, we selected different canonical markers that has been associated already with different cell types, as KLK3 for epithelial cells. And we saw how the chromatin accessibility was changing between all the different clusters that we had in our sample. So doing that, we were able to identify stroma cells, epithelial cells, and immune cells. These are really preliminary data and is a work that is ongoing. So right now we are trying to have a better characterization of all of the cell types that are forming our tissues, because we want to be more in detail about which kind of immune cells we have. Also in prostate cancer, there are different epithelial subtypes that has been described, and we want to go more in detail to know what is the composition of our tissues. So I hope that little by little we will improve in our analysis and we will be able to have a deeper annotation soon. So considering this, we were also interested to see chromatin characteristics between the race and the two groups that we were comparing here. We saw that all our samples were presenting cells in all the different clusters that we identify. And when we took the differential accessible regions and we compare black patients versus white patients, we saw that the peaks that were enriching black patients were associated with inflammatory responses and metabolism. This is not new. It has been reported already in previous publications. Black patients present more inflammation and more activation of metabolism. But considering these results, right now, our objective is go deeper on this and try to perform some GSEA analysis to have a better understanding of the different pathways that are activated, the different chromatin state of genes associated with pathways that can be associated with specific clusters like epithelial cells to know how AR signaling or how EMT or inflammation is the chromatin of all of these genes is behaving between black and white patients. Finally, in addition, we wanted to have a better idea of the motif analysis because we wanted to know how short DNA sequences are overrepresented in regions with open chromatin. So what we found when we did a volcano analysis in this volcano plot is that in our white patients, we had a high increase of CTCF. This was really interesting for us because this protein is it's a protein that is associated with the 3D structure of the DNA and the organization, and is also able to regulate gene expression. So in addition to the data that we got with our ATAC-seq, either with the volcano plot or also when we represent it spatially in our tissues, we wanted to validate these results. So we took an additional cohort of patients that is a TMA that is formed by 100 patients, 50 black and 50 white, and we perform a CTCF immunohistochemistry. In this case, we saw that again, CTCF overall was highly expressed in white patients when we compare with black patients. So this is one part of the project that we want to continue digging and having a more functional analysis to see really the relevance of CTCF organizing chromatin in these samples. So in general, what I want to finish my talk with a similar slide than the previous speaker, David, because here I show you some preliminary data related with the spatial ATAC-seq. But the real power of all of this analysis is the integration of this data set with other spatial technologies. So right now, we are at the same time that we did the spatial to know the general chromatin state of our samples, we perform also a spatial histone 3K27 acetylation, because this data integrated with the spatial ATAC-seq will allow us to have a better idea of which chromatin is active, and also select and nominate different enhancers that can be associated with the development of prostate cancer, and also enhancers that can be associated with more aggressive disease and are also specific to race. But not only that, we are also performing in the lab spatial transcriptomic analysis and protein analysis, everything is spatial, because if we take all of these data sets, we pull all the data, we will be able to have a better understanding of why black prostate cancer patients present tumors that are more aggressive than white patients. So with all of this, I would like to thank all of you for your attention, all the ELIS lab members, all of the collaborators are at CPDR, and also collaborate at Omics, and all the collaborators around, and also the patients, because without their samples, we could never perform this type of analysis. And I will be happy to take any question that you may have.
It looks like we don't have any questions in the in-person audience, but thank you very much for your wonderful presentation. And with that having said, this is the end of day one, and see you everyone tomorrow at 9:30. Bye-bye. Thank you. Thank you everyone for participating. Day one symposium. Happy hour and poster session. Yay. Thank you.
Hear you on Zoom. Okay. All right. Good morning, everyone. Welcome to day 2 of this awesome symposium. I unfortunately couldn't be here yesterday. So I'm Michelle. I'm the director of the Genomics Core. I had other meetings on the schedule yesterday, but I'm going to be here for almost the entire day today. So I'm looking forward to learning a lot and meeting a lot of people. So if you have questions, Chinta is a wonderful person to chat to, but if she looks busy, as she almost always is, you can also try and snag me in the crowd and I'll help you as best as I can. So I'm a research faculty in biomedical sciences. I have a lab that we work on the genetics and genomics of ovarian cancer. And then in my spare time, I help out at the Genomics Core a little bit. I'm going to be talking later today about bioinformatics services that are offered by the Genomics Core. I work closely with Casey there, so we can always chat about that if you have questions. But we're going to start the morning with Mitra, who I actually have known Mitra for an incredibly long time. We went to grad school together here at Cedars. We were in the first PhD program. And so we're back. We're still here. They can't get rid of us. So I'm thrilled. They can't, right? Never. So I'm thrilled that Mitra's here speaking today. We get to be on stage still together.
Yeah.
15 years later.
I know.
I shouldn't say that. I'm aging us. But she's going to give us some wonderful information about multi-omics approaches that are really centered in proteomics. And that reflects the layout that we have at the Board of Governors Innovation Center over in the blue building up here at the Pacific Design Center. You know, we're kind of an off-campus campus of our own up here. And Sarah Parker, who's co-director at the Proteomics Core, has her office right next door to mine. And our labs are next to each other, our offices are next door to each other, and it's very representative of a close collaboration between the two core facilities. So we can always pass you back and forth between those two facilities for your research projects, and we work a lot together on academic projects as well as core work. So I'm looking forward to hearing what's cooking up in the Proteomics Core, because they're always out on the cutting edge.
Yeah,
Thank you,
Yeah,
Mitra.
thank you, Michelle. Yeah, hi, everyone. As Michelle said, I'm Mitra, managing Proteomics and Metabolomics Core. I'm going to talk about our core and what we offer, what services and what we do. And thanks for joining us for the second day. I'd like to start with introducing our team. Can move it. Oh, now it moved, I guess. Yeah. I did, but it didn't work the first time. But so I think, you guys know the co-directors, Jenny and Sarah, but we have great, scientists working with us. And I think, a lot of you know them, especially Alex Stodland. Alex Stodland is our metabolomics manager. A lot of consultations happens with Alex. So I think, whoever worked with our core probably already know Alex. And he does also a lot of, data analysis and, like working with people, explaining the data for people. So, but then we also have Justina, who's a faculty member here. She is working with us for post-modification, specifically citronylation. Then we have our newest member of the core, Alex Binek. I'm not going to talk about this later, and I think she has a talk that she's going to go over some of the things that she's going to do. But she just joined our core. She's doing a spatial imaging. And the two advisors that we have are Simeon and also Chris. Simeon, maybe a lot of you already met. He's also at PDC. He does a lot of, method developing for mass spec, a lot of innovation. He's amazing. He can just, put the different parts of mass specs and together and make a new, new thing. So, and Chris is our actually also a mass spec and also, IP expert. So a lot of, people who want to do IP, we make a consultation with Chris. Then the rest of our groups are Christina. Christina is volunteering right now. She's not in the room, but she also does a lot of different things in our group, but specifically single cell assay. She's expert on that, and she does, almost all the single cell runs that we have. So, and then we have Matt, who is sitting here, and he's our connection between our sample prep to mass spec team. He also does many different, tasks in our group, and a lot of our tracking system in terms of design or how it works, all is done by Matt, and fixing it if there is any problem. So, so he is very critical to our group also. And then we have our wonderful sample prep group, who are Liam and Lindsay, and they could not be here today either. But anyway, we appreciate all of them. And now I'm just going to go to, this screen is a little bit, AJ, small. I can't see even what I have. Can I? Yeah, I do, but it doesn't move. Oh, yeah, now it moved. Now I went way ahead. So, okay. So now I'm going to, sorry about that, guys. So now I'm going to talk about first how, so you can go to iLab and request a service, and we get an email, and usually I'm the one who responds to the email. I kind of like, and the first thing we do with that is that we set up a consultation meeting for you, because usually, like, people maybe know what they want to do, but then when they talk to our experts, they just better understand that what they have to do in terms of their sample prep or even what assay would be the best for their project. So we do always have that consultation meeting first. And then when we know what assay you like to run, then you let us know how many samples. And I create this, like a code in our SharePoint and send you a code and sample submission form. And if you approve the code, then we send that, job to our tracking system, which is, this first, page. And in the tracking system, when we kind of mark it as we receive the samples, step by step, where it goes, and then whether we have processed it. And when we have the data and we send you the data, then we call that job complete and we send it to the ready to build category. And then that is where we build, our clients. And usually we ask for a project number for that. We kind of like do that usually at the end of the month. I mean, we ask a project number in the sample submission form, but we send this to the core finance at the end of each month. And then I just like to talk about our services and what we have. So the services that we have, generally for mass spec, I can say we have a discovery and targeted, assays. Discovery is just, if you kind of want to know what's going on between your two cohorts, it's a really good assay to run. And we just have this, I will send a flyer for this. We got Astral, our new mass spec. It's a sensitive, very fast, system. So it's really making our work actually more efficient. So these days, a lot of times we try to run the assays with Astral, and it's really good for bigger cohorts. We do have like Ascend also, another system that we have. The Ascend is mostly used for if you want to know about more of the structural biology or complex, molecular characterization. We do also it's very similar to the Astral, but it's not as fast as Astral. So, but it can go deeper in certain, especially for molecular, structural biology. So, and then in terms of the services here, what else I want to say? And then the targeted, the targeted assay, if you want specifically any specific protein. The only thing with the targeted is that it's going to always take longer because we need to develop the assay for it. And it's just like there's more time needed from any mass spec expert to work on it. So it's going to be maybe even a little bit more expensive. So but we do offer, if you have a specific protein that you don't have, like you cannot do ELISA with it and find out when you need mass spec for it. So so I think in terms of this is another system that we have. This one is also used actually for targeted, but that can do discovery. This was our, we use this a lot and still use it. But now that Astral came, we run most of the assays with Astral. But if we have a pilot study, for example, we might run it with the Explorers. And but if you have like 100 samples, we definitely send it to the Astral. So, but because it's a good system, of course we have it and we do use it, and also you can use it for targeted assays. So, and then for all these things, of course, you need to do sample prep, and we have a lot of automation systems for sample prep. One of the ones that we use generally for discovery work is the i7, BioMag i7. We can just, have plates, for, like large cohorts, easily prep with the i7 system. The other thing, system that we have, this is SEER. So what is about the discovery assay is that a lot of times you run the assay for example, serum and plasma. And in serum and plasma, there are proteins, that abundant proteins, that like albumin, that is just like they're hugely, different from some other low abundant, proteins. And you can't, like detect or measure when you have such high it's hard to detect those low abundant. So we had, and we still have, this depletion, assay, which uses some thermo Fisher beads that we use, and we take away almost like 14 abundant protein from the serum and plasma with that. But what SEER does, SEER has nanoparticles. Those nanoparticles actually gathers, all those like a lower abundant proteins. And then what we do is that then we elute that and then we do digestion. So you have a much better coverage, deeper coverage when you use SEER technology. It's a sample prep system, it's an automated sample prep system, but they have their nanoparticles that it's a kit that we need to get to do SEER. But it's a great, system also if you're looking for, those low abundant proteins. So we encourage people to use that for some of the proteins that they cannot see regularly with just a regular discovery, assay. So the next thing that we offer, which I just mentioned that about Christina and single cell, it's our single cell assay. We sort the cells with this cell-in-one system. It's usually 384 plate that we use and sort like a usual tissues. For this, we need kind of fresh samples. Not sure if we can still do frozen. So if you like to use, the SLN1, definitely we talk to you and how to submit the samples. We are checking whether the frozen samples can be run also, and it would be the comparable. And I think Jesse Meyer's lab actually has done that, and it seems that it is possible to also use frozen samples to do single cell. But for now, we still want to kind of stick to the fresh samples to kind of like validate all that. And if we completely validate that we can also use a frozen sample, then, that would be great because people can actually send us samples from other places and we can do single cell for them. But we have the teams, the SCP mass spec for that, and Simeon mostly responsible for this, and he's the one that run these, on the team, on the SCP. And so this basically about our single cell. I don't think I missed anything about it. So the other thing that we have that I mentioned is Alex Stoughton and metabolomics, but Alex Stoughton is not just metabolomics. She developed assays, for example, hormone assay, bile acid assay, but for the metabolomics, for the targeted metabolomics, we have like 219, metabolite that we can measure. We can send you those if you are interested. I mean, again, when we talk to you and we know what you like to do. So these are his system, and he just mainly, is the one that runs this metabolomics and analyze it and send the data to you. So if you are interested in any of these assays that I have here, please contact us. But also, we have posters here, 2 posters. So it actually shows more details, shows some data actually on it. So please check the posters. They're just right on the right side when you get out at our table. So you can learn more about what we do in more details, and you have the information in terms of like a link and everything that where to go to find us. So the next thing is that I want to show you the sample of the report we actually sent people. So at the end, when we send the data, we have a report that besides sending you the protein and peptide IDs, we have like some statistics, that these are the examples of plots that we have for in that report. And I just want to get, oh, okay, sorry. I think I am going. Okay, so then the next thing that I have to say is our immunoassay. This is our, that was all the mass aspect, of course, but these are the immunoassay that I actually am more responsible for it. So definitely, if you want immunoassay, contact me and talk to me. We have two systems, MSD and Olink. Olink is a newer one that we have. They are both great. They showed actually in a little publication that they are very comparable. The biggest difference I have to say is the volume of the sample. If you're very limited in the volume of sample, or Olink only uses 1 microliter. But otherwise, in terms of the panels, they both have good, nice multiplexes. And we have, I think a lot of people have from Cedars, I've had a lot of requests for MSD already, and they, a lot of people know about MSD, but Olink, again, it's a newer system that we have. So I think actually this is basically what I have. And this is our newest, we haven't announced this yet. We are gonna have a flyer for this sometime in summer. We are still having training for this spatial imaging that Alex Binek is gonna manage, and she's the specialist for it. But it's all just started, so I'm not gonna talk about it a lot. And Alex has a talk, and she's gonna talk about it actually more and go to more details. But this is, I included here, but this is not a service yet. We are probably gonna have it somewhere in midsummer to offer it to everyone. So this is basically, and I want to thank everyone. I want to thank the directors, my co-organizers. They are great. Chinta, Alex, Senate. She's not here, but she did great job. She worked a lot. So, but all of them, And also my team members in the core, I want to thank all of them. One person that I want to mention that I didn't have his picture in the team because he's not officially in the core, but he is very critical and important for our lab and core, and that's Ali Haqqani. He is taking care of almost all the mass aspects, and without him, really, we could not function. So I want to thank him too. So that's it. If you have any questions. Sure.
Okay, thank you. Thank you so much, Mitra, for your talk. Our next speaker of this morning's session is going to be Alex, also known as Lex Hatton, from Jesse Meyer's group. And the talk he will present today is entitled Single Cell Trajectory Interference for the Detection of Transient Gene Expression.
All right. So today we'll be talking about single cell trajectory inference for the detection of transient gene expression, which we are calling SC Transient, put together a small software package. So I'm going to start by telling you things that a lot of you already know, so please bear with me for a minute. So single cell experiments often look at differences between conditions, so with or without treatment, before or after differentiation, or between different clusters. And part of the reason for that kind of grouping is that it is the discrete cellular states is a very appealing model. And part of the reason is that discrete cellular states is a very appealing model. So you have some stem cell which differentiates into a completely different cell type, and so on. And as soon as you have the omics perspective, things become a little muddied. It's not entirely clear at what point you're supposed to have a different state. So if you take a single gene which is differentially expressed, at what point does the transition happen? Is it halfway? Okay, that's fair. But what if you have a dozen genes, or halfway between half the proteins, and that's a mess. Now, so there's kind of a practical aspect of single cell experiments that kind of lend themselves to encouraging the discrete state, and that's At least from what I've been told by biologist colleagues, if you take a cell, rip it open, split its contents and shove it into a bunch of tubes, you don't really get the same cellular function as you did beforehand. And that's a little messy. Now, if we're looking at something like a larger scale analog with mouse experiments where you are sacrificing mice at irregular time points, you have a bit of advantage of, you know exactly when the treatment started, you know what conditions going on. For a single cell, you can know when the treatment starts, but the state that a given cell is in is kind of unknown, and that is very important to how a cell reacts. And so you could do something like synchronizing the cell, the biological process, but that is very difficult to do in vitro and effectively impossible in vivo. But despite all that, we know that there are transition states, so we don't have discrete cellular states. We know that there are important things that happen during these transitions. So think about the cell cycle. You have checkpoints partway through. And one of the questions that we were interested in was how would we detect omic changes during these transition states? So just so we're on the same page, this is kind of what we're looking at. So you have the gene expression curve on the top. You've got some state transition on the bottom where you have some initial baseline for the gene expression. There's some increase partway through and then return to baseline. So if you were looking at just differential expression between the end and start state, you wouldn't see that this gene would not come up. And at the same time, we don't really know what we're looking for here. So if we were just detecting this pattern, that'd be one thing. But what if we have something like this where we would detect the gene in differential expression, but we would miss some of the important dynamics partway through the process? Or what if we have something a bit messier? So we decided to cobble together a bunch of existing computational tools and take a look at this. So the first thing is that we have to take our data and then put it into some function of the biological process. And we do that using an approach called trajectory inference. Now, omics are generally high dimensional. You'll have one cell with hundreds or thousands of genes, but the cells and the transitions only occupy a very small subset of that high dimensional space. So the idea is that you want to have some representation along the axis of that transition in the high dimensional space, and that's what trajectory inference provides. And the rough idea is that you select some starting point, some starting cell, you find its closest cell, and then you repeat until you run out of cells. And once you have the trajectory, you can project the cells onto that trajectory, and then that should give you a rough idea of the gene expression as a function of biological process. Now that projection, that's along an axis that we call pseudotime. And pseudotime is not poorly named, but time is very misleading. The pseudo should be emphasized here. So if I mess up and then say function of time, I do mean a pseudotime. This is not a reflection of time. So what we end up with our data is that we have a bunch of dots that we've lined along some axis. And in order to get that red line, we'll just use something like a very simple windowing. So we move some Gaussian window along the process, and that gives us the red gene signal. Now for the last thing, how do we do transient event detection? And then we have to be specific with this question. What do we mean by transient? So where should the transient change occur? How long should the transient event last? And how do we deal with noisiness? And on that same front, what do we mean by significant? How do we decide which of these genes show better transient changes than others? And wavelets can do a lot of work here. It is one of the tools that's used in signal processing. It is more or less exactly the tool that we need for peak detection, which is the problem that we're looking at. It does localize to different parts of the pseudotime axis, so we don't have to worry about exactly where. So we don't have to bias our analysis by saying, oh, it needs to be exactly halfway through. And the important part is that it is also scalable, so we don't have to specify how long a event should last. So what the wavelet transform will give us was some coefficient matrix, and then we came up with a heuristic based off of that matrix that grades different genes with how present a transient event is. Now, we don't really have a good ground truth for what that's supposed to look like, so we started with synthetic data. Big part of that is that we are able to know the ground truth in synthetic data and then investigate a whole bunch of different properties of the signal on our heuristic. So just so that everyone's on the same page, orange down here is the underlying signal, blue are the samples that we would take along pseudotime. So broad behavior, it behaves kind of as expected. So as we have a higher gene signal, we have a better score. Now, one of the important aspects for single cell experiments is how many cells you have. And so one of the things that we wanted to make sure was that our heuristic makes sense with number of samples. So the blue curve at the very bottom here is the low number of samples. And you can see that it starts off somewhere here and then doesn't improve much and becomes wider and wider. The distribution becomes wider, as opposed to the high sample ones, which steadily increases and doesn't have that increasing spread. And when we contrast that by the same score in noise genes, we see that having more samples reduces the score given to noise genes. And that's important. That means that if you have more samples, you get a better score, a tighter score, and you can differentiate from noise more easily. Yeah, so that increasing spread I mentioned for low sample counts is largely not due to anything due to the wavelets or any of the analyses, and more if you happen to have a sample point which lands near the actual gene transient event, then you detect it and you get a high score. If you don't and you miss it by chance, then you get a low score closer to noise. So that distribution of scores becomes increasingly bimodal. So using this, we've more or less come up with the novel insight that more data is good. But at least our metric is behaving as we would expect. So if we use the rule of you have your noise baseline, and then you want to move 3 sigma above that in order to count as a hit, what we have is for low sample counts, we end up with a asymptotically approaching some threshold which won't be crossed. So if you have low samples, it doesn't matter how high your signal is, you're more likely to miss it, as opposed to high sample ones where you don't need that much of a high signal to detect it. That one I'll skip over for the sake of time. So we looked at 3 data sets. I'm going to skip over 2 of them again for the interest of time, but the punchline is the first 2 didn't work so great. We found some results that looked okay, but that weren't quite what we were looking for. So for hematopoiesis, the bottom right curve, or the bottom right distribution shows the distribution of scores. When we do gene enrichment on the retained ones, we have a bunch of pathways which relate to the red blood cells, and that was kind of what we were looking for, but when you actually look at the gene expression signals, they don't have that transient characteristic that we're looking for. This is more or less a differential expression. So I'll skip over the macrophage. But when we looked at the cell cycle data set, which was acquired as one of the benchmarking data sets for the Astral by a different group, we ended up with some results I didn't quite trust because they were exactly what we were looking for. So what we did for that was that we used something called supervised pseudotime, where we identified the cell cycle phase, we ordered the cells along pseudotime using that information, and then scored all the genes, took the top, I think, 100, clustered those, and then did gene enrichment on each one of those clusters. Now, one of those clusters, cluster 1 at the bottom left, what that shows us is that we have some baseline for those genes, a transient increase, and then back to baseline. And the part that is exceptionally exciting about that was that without us informing it directly, that transition happens exactly in between the G1 S, exactly in the S phase bounded on either side by the other checkpoints. And it's entirely possible that this is entirely accidental. So when we did gene enrichment analysis on that, we found exactly all the pathways you would want to find if you're looking at things which are transiently expressed in the S phase. So DNA replication, hydrolase activity. I'm not a biologist. I'm not going to embarrass myself and say that I know any of these, but I'm told that this is very exciting. And then for the other one, for the other cluster that's displayed here, we have a transient increase right before the S phase. That one didn't scream, this is definitely what's happening. We did get some pathways, but we are looking into that was beyond the scope of the project. So if anybody recognizes these and it makes sense that it would happen right before the S phase, come find me. I'd love to hear about it. Okay, so to summarize SE transient, we used trajectory inference to order cells. We obtained gene expression signals as a function of progress along a biological process. We did wavelet analysis to identify the transient events. We investigated the behavior of our heuristic against synthetic data, found that it made good sense. And then we identified biologically plausible pathways from real data sets. Yeah, I don't think I mentioned this, but yeah, I'm part of the Omics data science group led by Dr. Jesse Meyer. And I will leave the bibliography here in case anybody wants to look at either some of the data sets that we used or some of the methods. If there are any questions, happy to take them. Otherwise, you can probably find me in the hallway cursing my laptop.
Thank you so much, Lex.
Right. So my question is, when you see all this single cell data, you do the pseudotime analysis and the trajectory. Like, let's say the most common phenomena that happens in cancer cells is plasticity. You take a cell, I treat it with a drug. Sometimes the cell responds and later on it doesn't respond because it bypassed the mechanism for the target of the drug, right? What I want to know is, because when I know the wet lab, like this is expected from my wet lab experiments, then it is very easy to define the node in the pseudotime and then see the trajectory. And then I can say it started from here, it went into these states, so the drug is not responding or the tumor is not responding. As a bioinformatician, if I don't have that supporting wet lab and I start from a single cell, how do I define that node? Because to go for the trajectory.
Yeah, so the short answer is you basically can't. A lot of trajectory inference relies on external information to validate it. So one of the ways that you want to link pseudotime to actual time or some progress is by finding some set of markers that are kind of expected. A lot of the times, if you're looking at differentiation or treatment, it'll be exactly the starting state. So you'll know there's a set of samples which have the starting state, and then you go from there. If you don't know how to order along, like where to start, that becomes, I don't want to say impossible, but there'd be some amount of guessing, checking, and hoping. So it's not a satisfying answer, but these methods do require external information. So the way that you're doing it is correct.
Yeah, because what I think is now any single cell paper you take from literature, any system, any cancer, they always show that it starts here, it goes to these multiple lineages. What if actually the multiple lineages are going towards the backward? But
yeah,
that can happen.
Yeah, so a lot of these methods don't. So since pseudotime isn't actually progress, it's just changes along the omic profile, it doesn't actually have any directionality. The directionality comes from external information. So you may know that your cells started on day 1, and then these other samples are at day 2, and then that allows you to say, okay, well, this makes sense because we are seeing that the progression is from day 0 to day 20. If you look at that progress and it turns out that it starts at day 3 and then it goes to day 0 and it goes to day 21, either the ordering is wrong or that tells you that trajectory inference doesn't work here.
Thank you.
Thank you. No? All right.
Is that all? Okay. Thank you so much, Lex. Our next speaker is going to be me. Good morning, everyone. My name is Alexandra Binek. As Mitra already kindly introduced me earlier in her presentation, and as she also said, I recently joined the proteomics and metabolomics core. Before that, I've been a member of Jennifer Van Eyck lab for the past 6 years, doing some cardiovascular proteomics, broadly speaking. And in my presentation today, I will introduce everyone to our newly acquired mass spectrometry imaging fleet, and I will try to answer some of most frequently asked questions about the sample prep, single cell proteomic sample prep, and some just like briefly go over some more unique applications that we've been working over the past like 4.5 years. So very quickly here, 2 months ago, we have installed 2 new spatial mass spectrometers in our lab. First of them, NeoFlex, is a benchtop mass spectrometer, which I would call it more a translational research instrument. It can leverage multiplex approaches that would combine intact labeled protein mapping imaging and with also advanced small molecule lipid, glycans, and other macromolecules imaging. And it all can be measured on the same platform, while our second instrument that we've acquired is called timsTOF Flex. It is more of a discovery instrument, which offers high resolution. It's more powerful, more sensitive for more advanced omics researchers who would like to perform deep discovery projects. It has a dual source with a passive driven identification, so that allows us to separate isobaric or isomeric ions. And when I talk to our prospective users about those two instruments, I often get asked those same questions. What slides do we work with on those instruments? What's the resolution, et cetera, et cetera. What is the biggest difference? NeoFlex works with conductive slides, such as ITO-coated slides and IntelliSlides, slides, the vendor slides. And TimSoftFlex works with also conductive slides and glass slides. And the main difference between the two instruments is the resolution and speed. NeoFlex can work with 20 micron resolution, while our current TimSoftFlex works with 10 micron resolution. It can be in the future upgraded to 5 micron. One of them, NeoFlex, is more, as I said, throughput, translational, faster instrument. The other one would be more a discovery driven projects and higher resolution. In this slide here, I wanted to quickly overview main differences for the mass spectrometry imaging sample processing. So we use fresh frozen and formalin fixed paraffin embedded tissue that is obviously sectioned and mounted onto the conductive slides, such as ITO-coated slides. Then in case of paraffin embedded tissue, it needs to be dewaxed. It can be then subjected to antigen retrieval followed by the enzymatic digestion if we want to prepare it for peptides or glycans mass spectrometry imaging. While fresh frozen prep for MS imaging is much simpler, it requires thorough drying and can also involve that enzymatic treatment if we are interested in those analytes. And then a very important step that involves a spraying instrument that can be used to spray the enzymatic different enzymes for the specific analytical preps, but mostly is used to apply matrix. And that matrix allows the mass spectra to be acquired across the tissue section, and then the data can be analyzed using mass spectrometry imaging software. And then what is this matrix? Here are a few things about MALDI matrix. So it is this small molecule that absorbs UV light of the laser and analytes, different analytes of interest. Their molecules will get embedded in the matrix through the process of coating with our sprayer instrument. And inside the mass spectrometer, the mixture of our analytes embedded in matrix will be ablated by the laser, and that would lead to the desorption and ionization of those analytes molecules, which then be acquired by the mass spectrometer analyzer. As Mitra has kindly already mentioned, we yet are not offering mass spectrometry imaging services openly to the research community users. We will soon. However, right now we are focusing on a few pilot collaborative projects with few members of Cedars research community, and we are focusing on establishing our workflows so that we can run smoothly once we are open. And now I would like to shift gears a little bit into the challenges of working with single-cell proteomics. Most of those challenges are inherent to cellular morphology, unfortunately. some cells are very large, some cells are very small, and both of the cases is problematic. Another level of challenges is existing sorting technologies that decrease cellular viability due to the stress of the hydraulic system of the software sorter. Another important issue is the sample volume. Nanoliters, let alone microliters of buffers added to one single cell is like an ocean. Finally, there is a problem of proteome dynamic range, especially for those highly specialized differentiated cells. Yes, I will show you our the multiple different systems that we have worked with and developed protocols for high throughput and gentle isolation of cells ranging from 10 to 200 micron in length. And our approach leverages instrumentations that dispenses sub-microliter volumes of reagents and also has very sensitive, highly sensitive mass spectrometry instruments. So the sample processing step by step is shown here on this slide. Tissues are collected prior to cell isolation, and they are treated with enzymatic cocktails such as collagenases. The resulting cell suspension would be then instantly transferred onto our gentle sorting cell-in-one system that isolates those single cells into 384 well plate. Cell-in-one interface can capture every single image of the sorted cell, so we have that additional level of quality control. And if you want to use that imaging of the cells for some other bioinformatic normalizations, this is available. After the cell lysis and tryptic digestion, label-free analysis is carried out on our mass spectrometer instrument from Bruker using DIA, data independent acquisition. A little after 2 years of our experience with, after we purchased Cell-in-One and timsTOF SCP instrument, we ended up locking our main single cell label-free workflow. We published that workflow in 2023. It basically ventures this very nice LC configuration of parallelized nanoflow dual trap single analytical column approach, and it operates at 10 minutes per one cell, and which brings us to a turnover of over 100 cells per day, which is a very often asked question that I get. That comes with a follow-up questions: how many cells do we need for a single cell proteomics project? So our capability and the setup, the technology setup allows for a lower thousands per project. Even with that seemingly low number in comparison to genomics, we are still publishing the largest single cell proteomics datasets in the community. So it is still a lot for proteomics. All that we saw until this slide was our standard workflow that we use for majority of the cells that we do proteomics for. But now I will walk us through some our non-standard single cell protocols. And one of our very, very first protocol was optimized and involved very large, very fragile, and very weirdly shaped cells, which are cardiomyocytes, due to this sample type morphology. And it's very low viability. The cell death rate is so bad in some preps with cardiomyocytes. It requires gentle handling large nozzle sizes, and lots of love. We were able to successfully isolate cardiomyocytes from both perfused entire hearts or chopped up tissue chunks, enzymatically treated. You can also visit our app note we did with Cellin1 to get more details about the protocols. And they were developed with our collaborators from Cedars, Dr. Marban's lab. A huge shout out to Tassio. We also developed with Leffer's lab, and our spearheading lab member, Blandine, who is already doing her postdoc in France. But these people were the main drivers of this specific protocol. And then next, we shifted to something completely opposite. So we started to attempt at the very, very small things, that is single mitochondria. This approach involved appropriately sized tiny nozzles, and also a software plug that allowed for the detection of subcellular particles. The mitochondrial purification protocol itself, it undergone multiple iterations of optimizations led by Dr. Alex Stotland, whom you heard about today already, and ended up leveraging the nitrogen cavitation protocol, followed by nickel magnetic beads HISTAC pull down. This ended up working and producing the clearest mitochondrial fraction. Further analytical optimizations led by our dear member, MS team member Ali Hagani, also mentioned here today before, further improved the sensitivity by applying longer analytical chromatographic columns with a smaller internal diameter, and also longer LC gradients. Then in my last example of today, I will show you today the project involving the isolation of pacemaker cells. So the sinoatrial node is this organ, primary pacemaker of the heart, that is an oval-shaped region in the right atrium that consists of the cluster of those specialized pacemaker cells. And it controls the normal heart sinus rhythm. In this particular study, our collaborators, Doctors Goldhaber and Ross, they wanted to determine what's the proteomic heterogeneity and composition and interplay of pacemakers and all the surrounding nodal adjacent cardiomyocytes isolated from control mice. And the problem with these cells, with the pacemaker cells, is that they are very, very low abundant. This is a very tiny organ, and these cells are not sortable in a normal sorter. There's just not enough of them. I hear from Doctors Goldhaber's lab that usually they would yield 100, 200 pacemaker cells from one mouse heart. So how do you sort that with conventional system? So we used those GFP nuclei labeled mice. The HCN4 channels were GFP labeled in those pacemaker cells. These channels are essential for generating pacemaker activity. And in our approach, we combined fluorescence selection and bright field selection criteria for the pacemaker, successful pacemaker cells isolation. Again, we utilized our gentle cell sorting Cellin1 platform, and we positively selected for GFP positive cells, and morphologically further selected them for their size, their length, We tested that methodology, single-cell proteomics methodology feasibility, performing measurements in this pilot, mass spectrometry measurements in this pilot experiment. You can appreciate here in this slide the first results of our pilot experiment. We used UMAP projections where we identified two main cardiomyocyte clusters, the orange one and blue one, that we isolated from the cardiac cells, right atrial cardiac cells. The first cluster, the orange one, is enriched in right atrium cardiac marker isoforms, which you can appreciate here. I don't know if you can see my marker on the right-hand side of the slide, in the top of that UMAP. And then very, very interestingly, that very orange cluster contained almost most population of pacemaker cells. So the Leiden cluster 0, the blue one, appeared to be less enriched in metabolic proteins and cardiac myocyte proteins, sarcolemma content. I'm not showing all those proteins here just for the time's sake, but this suggested the existence of two distinct cardiac populations, which only one of them was closely interacting with pacemaker cells. In the last step, we decided to focus on the differential proteomics analysis of just that Leiden cluster 1, where all the pacemaker cells were located. And in the center of the slide, we can appreciate the volcano plot that shows differences, differentially expressed proteins in pacemakers with respect to other right atrium cardiomyocytes. And interestingly, not only the calcium handling proteins were upregulated in pacemakers, but also phospholamine, which is one of the top upregulated proteins in those pacemaker cells, which is also reflected in that bottom dot plot of top five expressed proteins. Here I'm going to just conclude that last pacemaker project, and we presented that it is possible to do single-cell proteomics from the sinoatrial node pacemaker cells. We clarified some differences in the cell populations surrounding the pacemaking cells. We identified 3 clusters of cells with 2 main cardiomyocyte clusters, one of them interacting tightly with pacemaker cells, and 2 broadly different, metabolically different cardiac cells in right atrium. Here, I would like to just thank all of our teams in ACBRI laboratories. Big thanks to Jennifer Van Eyck, obviously, who established all those collaborators for all those fun projects for us to do and learn. And thanks to our collaborators from within Cedars, Sinai. If it wasn't for you, we wouldn't be able to do any of that fun stuff. And again, thanks again. Big shout out to our team. Thank you.
It's time for one quick question.
If not, I will be at the poster session, and I can chat with you one-on-one. And our next speaker, are you going to, thanks. Our next speaker is going to be Haley Norwood from Spatial Molecular Profiling Shared Resource Core. The title of her talk will be the SMP Core Services. Big shout out. Warm welcome. Zoom is working. Please unmute and say that everything is okay.
Everything's working.
Perfect.
Awesome. Oh, I don't have to press anything. Cool. All right, great. Thank you, everybody, for allowing me the opportunity to speak about the spatial molecular profiling core. Today, my organization of our talk is we're just going to briefly go over the core, our mission, the personnel. We'll then transition into the type of technology that we're offering, which includes the workflow and frequently asked questions. Oh, gosh, what do I do? Oh, okay. So, so sorry. So as I mentioned, our introduction, this is the outline of my discussion today. We'll talk about just the introduction of the core, the available technologies, which includes the workflow and frequently asked questions, and then I'll conclude. So our mission as the Spatial Molecular Profiling Core is to provide advanced technologies and our expertise to analyze the molecular makeup of biological samples. And both with proteomic and transcriptomic methods available, the researchers can gain insight into the complex biological processes. You can identify biomarkers, study the intricate cellular interactions within the tissue microenvironment, and finally, ultimately, as we all know, we want just to achieve a more comprehensive understanding of disease mechanisms. So here's our personnel involved in the core. We have Dr. Keel Merchant, who's our scientific director. This is myself. I am basically the manager of the core. I work with all the consultations in terms of bringing together what type of technologies we can offer you. And then I also work with the finance as well. We have our two operators for the core. We have Ying Li. She primarily works with the Omics digital spatial profile, which I'll talk about shortly. As well as she works with the Hyperion XTI, so that's imaging mass cytometry. We have Anton, who is also our operator. He primarily works with the Luna-4 Comet. He's been our biggest user for that, as well as he's really optimized the programming for it. And he also works with the Hyperion XTI. And then finally, we have Simeon Mayhoff, and he's our senior bioinformatician. All right, so our instrumentation that we have at the core is going from the left to the right. We have the Hyperion XTI imaging system. This is actually the upgrade to the original Hyperion. We then have our newest platform that we've launched, which is the Comet by Luna-4. And then finally, we have a proteomic transcriptomic piece of technology called the Omics digital spatial profiler. So the Hyperion, it is a multiplex tissue imager that uses metal tagged antibodies for detection, and it can detect up to 40+ biomarkers within a given tissue sample. It clearly can define any tissue type without background autofluorescence and spectral overlap. So let's talk about kind of like what the workflow will do. So you'll, as a user, you'll come to us providing your sample slides, and following a consultation, we'll provide you with the panel of biomarkers that you're interested in investigating. The slides will be stained with the said biomarker panel. The Hyperion will then acquire the image. We'll provide you the images that you wish, and then working downstream, we can work with you in terms of analysis or however you would like to proceed. Here is a representation of images taken from the Hyperion XTI, and what you see is a comparison between normal and cancer-related breast tissue, and you can definitely see the differences in coloring patterns between the two tissue types. What do I do? Oh, okay. Excuse me. So what are some frequently asked questions that we get from users is what type of samples can be run on the Hyperion? And that includes the FFPE, formalin fixed paraffin embedded tissues. We can also utilize frozen sections, And then finally, we can also run cytospin. So that's the cellular pellet of your sample. How many biomarkers that can be detected with the Hyperion is 42. And the type of antibodies that is utilized for this piece of technology is metal tagged. The viewing imaging area is 1.0 millimeter by 1.0 millimeter ROI, or regions of interest. And the resolution of the images is 1 micron. In comparison to the Hyperion, we're also offering the Comet by Lunaphore. So this is a multiplex sequential immunofluorescence system that uses fluorophores instead for detection via secondary antibody or a conjugated fluorophore primary. This will honestly open up for users to use a wide variety of antibodies. So I think that's a very nice benefit to Comet. Finally, referenced by Lunaphore, they say that we can detect 40 markers, but in our case, we can actually exceed that and go to about 80 markers in a sample. And finally, this is a fully automated process. So you can basically program it into the machine and you walk away. So that's definitely a great plus. Here is just a breakdown or the walkthrough of what will happen during the cycle in the machine. So we acquire your samples, and after we discuss the panel development and so forth, we will then apply that antibody cocktail to our samples, and through a cycle of staining and elutions of the samples, the images can be then acquired by the microscope of the machine, and then we'll provide you the necessary data, and we'll proceed with whatever downstream applications you wish to do. So next slide here is I'm just showing a representation of images taken following Comet. We have on the left a human tonsil via the germinal center. And then on the right is an example of staining from a non-small cell lung cancer sample. Frequently asked questions. Again, what type of samples and tissue types you can run. Similar to the Hyperion XTI, you can run paraffin embedded. We have frozen and again, cytospin. A big difference between Hyperion and Comet now is the number of markers that can be used to detect. So for Hyperion, as I said, it's about 42 markers. For Comet, it can be 42, but we've gone above that and we can actually do 80. So we've been really pushing that forward. So what kind of antibodies can be used? There's two different types of antibodies that we can utilize for the instrument. One is a primary antibody that's been conjugated with a fluorophore, or you can just use a secondary antibody that's conjugated to a fluorophore. So the benefit of this is it's just you can use any antibody that comes from a vendor, compared to IMC where it has to be conjugated to a metal that can kind of make it a little bit more complex in terms of study design. Finally, the viewing imaging area is 12.5 millimeter by 12.5 millimeter, and the resolution is very great, very good. I mean, it's very small. So we have a 0.2 micron to 0.3 micron. All right, so the final piece of technology that we offer at the core is the GeoMx Digital Spatial Profile, or GeoMx DSP. So this is from NanoString, and it is a flexible and robust spatial omic platform that is used to analyze FFPE and frozen sections. And what this guy particularly does is he non-destructively profiles the expression of RNA and protein from distinct tissue compartments and cell populations. This can also provide whole transcriptome and 570 plus protein targets separately or simultaneously. So this is a kind of a breakdown of what happens during the workflow following our acquisition of the slides. We will work with you in terms of like what kind of cleavable oligos and protein antibodies that will be used. And the significance of the protein fluorescent style protein antibodies is that this will allow you to actually figure out the region of interest. So that's why those are there. We will then just place your samples into the machine. The machine will then acquire the data. We work alongside the applied genomic and computational core to get the sequencing data. We acquire it back, and then we'll present it to you researchers. So this is a representative image taken following using the geomics. We have on the left an example of a sample of a Hodgkin lymphoma. And you can see staining specifically for DNA, which is going to be blue. We have then 4 other staining, CD4, CD30, and CD68. And then on the right, we have just a sample of a human kidney sample. And you can definitely see the white is the selection of region of interest. So some frequently asked questions. I have a lot of these. Okay, anywho, so how many human or mouse whole transcriptome atlases are provided in this RNA assay? For human, it's about 18,000, and for mouse, it's 1,800. How many morphology markers can be used in a single experiment? It's 4. And then these 4 fluorescent channels include DAPI, or FITSI, excuse me, so that's for your DNA. We have Sci-Fi, Texas Red, and Sci-3. Okay. And why are these? The question would be, why are we using these markers? And they actually, as I mentioned previously, it allows us to visualize tissue components and also to guide your selection of the region of interest. Okay. All right. So I appreciate everybody listening to my discussion and talk. If you guys have any questions, please find me by our poster during the poster session from 2 to 5. I want to especially thank you, the Merchant Lab, as well as all members of the Spatial Molecular Core for providing their input. And if you have any questions, please don't hesitate to email us, as well as there's a beautiful QR code that you can take a look at, and you'll see additional information about the core. Thank you, guys.
Thank you so much, Haley. Are there any questions?
No? Oh, yeah. Okay, great. Thank you, guys.
Welcome back after the break. Our next speaker of the day is Simeon Mahoff from Spatial Molecular Profiling Shared Resources Core. And his talk today is entitled Navigating Spatial Experiments from Pixel to Niche. Warm welcome.
All right. Hi, everyone. Let's see if we can get this thing up a little bit higher. Hello. All right. Can everybody hear me? Yes? Okay. All right. So today's talk from the Spatial Molecular Profiling Core, we're going to be, the theme here is more on, like some emphasis, some of the emphasis here is on exploratory studies, which have proven to have kind of both shared and some unique challenges in comparison to biomarker-driven analyses, which Dr. Merchant was talking about yesterday. So in contrast to biomarker-focused studies where you have a clinical endpoint, for example, you have survival data, or you have some other quantity that you're measuring in patients, from clinical information, some of the exploratory studies are more focused on, how can we break down the tumor microenvironment? Is there, some differential expression here that is notable? How do we run statistics on this, or like what kind of conclusions are worth reporting? And when you're in that space versus the biomarker space, things can get a little complicated because it's or that space has its unique challenges in the sense that you need to make sure that you're normalizing the data and that you're trusting, whatever it is that you're going to use, whether it's like whether you're doing phenotype exploration or whether you're trying to break down the architecture of the tissue and then run various comparisons. So what I'm showing you here is just some raw marker images. On the bottom, you see histograms of the raw isotope count. So this is from IMC, but much of what I'm going to discuss is actually platform agnostic, more or less, because we're going to be talking about raw expression, at the pixel level and cell expression and what you can, the things that you have to be aware of when you're working with these data sets from a bioinformatics point of view. What you see on the bottom is two histograms. So the top is the raw isotope count for IMC. The bottom is a log transformation of that. As you can see, the distributions can look quite different. And it's not necessarily straightforward to decide, where is a marker positive, right? And that can vary across tissues. So, if you're trying to, because all of these platforms, whether it be Lunaphore, whether it be IMC or some other platform, they have some noise built in the measurements, right? So some of the kind of like important, starting points in the analysis of these data sets is to, kind of keep that in mind that, in across different samples, what represents a positive pixel or positive or signal versus noise can vary, right? In many of these analyses, what we do is we try to get a certain sense of kind of what's happening in the data. So what is the geography of these tissues, more or less? To that end, we can have, again, different normalizations of the tissue, right? Like you can apply, what I'm trying to illustrate here is that you can kind of see how, you can bring out like the lower portion of signal or emphasize like the higher portion of signal depending on what you're after and, what is the information that you're seeking from the markers in the panel. In terms of normalizing this data, so and again, this is an example with IMC, but with Lunaphore, some of these same challenges exist as well as with other spatial experiments. So on the left here, you have the raw signal, you have log transform signal in the middle, and then in terms of trying to deal with the noise that is present in these images, some of the techniques are coming from the classical image processing world, right? So we're talking about stuff like smoothing, or like morphological filters, Fourier-based filters, which we readily use, especially in IMC, but also in Lunaphore, because they help to remove some of the snow-like noise that you have in these images and focus on, signal where you have contiguous pixels being positive for a given marker. This is important because obviously, noise can accumulate, especially if if you switch to the single cell side of things, and then the more that you have noise at this early kind of stage in the analysis where you're doing the preliminary cleanup, of course, the more you're or the less of a signal to noise ratio you're going to get, and that has an impact further on clustering, which is the basis for further analysis like phenotype interactions, like differential expression and so on. So what I'm showing you on the right is some of the filtering that we use currently, and even at a visual level, you can see how it really brings out the signal. This example is with CK19 from a pancreatic tissue. So another interesting thing we can do at the pixel level, right? It's important that we consider how much information we can get even before we run segmentation, because segmentation in the single cell side of things has its own set of challenges. Obviously, it's not perfect, and you know, the resolution at the end of the day, as good as it is with modern spatial platforms, is still, you know, not at a level where, I mean, it's still at a level rather where you're going to have one single pixel represent, you know, multiple cell boundaries potentially and cell leaking. So in you know, in tune with the title of the talk, you know, can we get to studying the spatial architecture of tissues directly from pixel level data without even using segmentation? One way to do that is by using model-driven downplexing. So, you know, we can take a panel, regardless of how big it is. It could be transcriptomics, it could be proteomics, and basically use the same dimensional reduction techniques that we use in when, you know, with your typical UMAPs, you know, like UMAP or leverage linear transforms, stuff like non-negative matrix factorization or PCA, to get a colorization, sort of like, you know, when I say downplexing, what I mean is that you reduce information from, say, a 40 marker panel down to 3 dimensions, you know, as opposed to, you know, you can analogously to how you do that with with cells and a UMAP, and you reduce it to 2 dimensions to visualize the phenotypic space. Only here, what you're doing is you're reducing to 3 channels and using that to get, you know, like a visual of the ROIs. So on the left here is an example with using PCA, just on a lineage subset of an IMC panel from a pancreatic experiment. And on the right is a non-negative matrix factorization, which again downplexes the tissue using a functional subset of the panel. So the two are mutually exclusive, the lineage and the functional subset of the panel. Both are more than 10 markers, and their information is now compressed in these images. What you can see is that both sets of panels, when you leverage the information from all of the markers in the given subset, you more or less can recover. Interestingly, you can recover sort of like the compartmentalization of the tissue, right? Like you can see, for example, that, you know, these are islets, right? So you can see islets, you can see like a tumor cell compartment, and you can spot that in both cases. Another example is from a lung experiment, again using mutually exclusive subsets of markers, and you can see that once again you can kind of spot compartments in the tissue, and this is without even this is basically a direct visualization of what's happening in pixel space, right? No segmentation, no no clustering, no nothing, right? This is just a transformation from the information in expression, in high dimensional expression, into visual space. We can leverage that bioinformatically, and I'm going to talk about that in a little bit. I just wanted to discuss first another strategy for looking at compartments in the tissues. So we can also use masking. This is useful, or can be useful, certainly for very large images, such as whole slides, what we can image with Lunaphore, where we have many samples, and of course with smaller ROIs like cores on a TMA. The reason why you know, this is important is because in some cases we want to again compartmentalize specific areas, or get isolate specific compartments in the tissue that are of interest, like, for example, islets in this pancreatic data set. So here, what we use is just two specific markers, like if you have a marker that you expect to be specific to a compartment in an experiment, we can run image filtering together with thresholding and image smoothing to obtain masking for compartments of interest with that are parameterized, basically. So we can make those as tight or as expansive as we want, which means that if we were looking at these islets, for example, there are parameters in this process where we can control how tightly the mask lassos the compartment. And what that allows you to do, just like the spatial motifs, which I'm going to talk about later, is you can do sort of like set arithmetic with this kind of masking. What that means is that you can take, more expansive masks around the compartment of interest and tighter masks, and then subtract the difference so that that would allow you to look at the periphery, of a given compartment, which can be important for some study aims. And then back to the model-driven downplexing. So like I said, we can leverage these visualizations to start talking about compartments and structure in the tissues even before we've done anything that's single cell, even before we've introduced any bias from segmentation into the data. And the way that this is done is, we take the representation of pixels in this downplexed space, this visual space, and then we can apply smoothing plus downsampling of the images and use clustering much in the same way that we apply clustering to single cell data in order to obtain spatially contiguous sort of like subsections of the tissue, which will have a mixture, like inside the compartment, you will get a sort of a homogeneous mixture of phenotypes, because the overall expression in those areas of the tissue is of a particular type, right? So this allows us to go from pixel to niche in much the same way as we do with single cells, but for exploratory studies, that can be important because it is a dual complementary way of looking at your data, and in exploratory studies where you're not, just looking at biomarkers, where you're not just using some sort of like clinical endpoint and throwing a myriad of features at it and then running some modeling and looking at what shakes out, right? When you're doing exploratory studies, you kind of want them to be more interpretable, right? And you want to know what you're looking at in terms of biological interpretability, because you're trying to understand, what are reasonable ways in which you can stratify the tissue, or you can stratify patients, or, according to some exploratory aims that you may have. So this kind of approach allows us to, look at to remove some of the analysis bias that is introduced by methods when we want to talk about the architecture of tissues and breakdowns of data sets into microenvironment types. Now on to the single cell side of things. So models that we currently use, Mesmer, this is a Caltech developed model from the Van Valen lab. There's InstanceEgg, which Joe from our group has made to work quite well with Lunaphore. And we also have Stardust and a couple of other methods that we've tried. So far, Mesmer works pretty well, especially when you have high packing of cells, so for lymphoma samples, for example. And then I have to mention, right, what I was talking about earlier in terms of like bias, in terms of potential error that you're introducing, even under perfect segmentation. So it works well on average, and when we're talking about spatial biology. But even when segmentation is perfect, hypothetically, if it were perfect, you would still you you would still get, you know, some interference between cells. You would get some basically like the stacking of cells. You know, it depends on the cuts. It depends on the tissue quality. So and because the resolution, right, is still limited, like you're not looking at molecules themselves, you will get sort of overlap of cell boundaries where you're going to get cell mixing. So the implications of that are that when you get the single cell data, of course, you're going to have some sort of signal buildup, and you're going to have some outliers. You're going to have potentially some of that kind of inhibit you from necessarily being able to get to the rarest phenotypes in the tissue, right? And it can introduce some fuzziness or quite a bit of fuzziness in the data. But the procedure goes like, right, we have the segmentations, we have the raw data, we pass basically the segmentation through the image stack, kind of like a cookie cutter, right? And then you average the signal. And an important step here is how do you normalize the data, right, such that the markers will have equivalent weight. That is important because when you want to run something unsupervised, you know, like clustering, for example, you want to make sure that the clustering is kind of aware of the differences in cell fates, you know, or basically lineages, and it's aware across the panel equally, right? That's easier said than done. I mean, it's basic common sense, but also how do you do that? How do you make sure that your markers have equal impact, you know, and that the clustering can stratify the data on CD15, you know, like myeloid markers is as much as it can on like CD3 and CD4 and FOXP3 and so on, right? So if you were to just divide by the max in the expression, what's going to end up happening is that if you have many outliers in a data set, or if you have really high outliers, let's put it that way, it's going to depress a lot of the signal actually into sort of, it's going to compress signal and noise because your outliers are so high. So that can be a problem. To address that, we use something called elbow detection. So, or, you know, that helps us to estimate sort of like upper thresholds or like effective max in the data. And because this is controlled by a parameter, we can bias how close the elbow detection is to the max. This is also important because it has implications for sensitivity. I'm not trying to like make you here like make you an expert into what data normalization and data scaling involves, just to be aware that these things can potentially affect your sensitivity to phenotypes, and they can have an impact on the clustering and how you the kind of picture that you get from the experiment. All right. Now, another important thing to consider, which we've thought a lot about at the SMP, is how to mitigate batch effects, right? Like we see that in many experiments, people bring TMAs and they bring, you know, like they bring samples from different conditions, sometimes even different diseases, and comparing running differential analyses, when you're doing that, it's key, of course, that you make sure that you're comparing apples to apples, right? And also, especially as concerns differential expression, you want to make sure that you're working, you know, with like a shared basis across samples, right? So that can be a tricky balance because on one side, you have technical variance, right? Which is you get a TMA from Nebraska, you get a TMA from Washington, you get like you get different tissues. They could have been prepared very differently. And so the consequence of that, and also of machine performance, which, as we know, it's just entropy, right? It's like machine performance can differ as well. So all of that introduces technical variance, but it's a fuzzy threshold, from going from technical, removing technical variance and going into removing biological variability. So that can be a potential issue, and some very smart people have come up with interesting ways of mitigating that. On the left here is a method that we've used quite a bit. It's PsyCombine. It came out maybe 2 or 3 years ago. These are just examples. There are other methods out there and in development, and as a matter of fact, we're developing one on our own. But this one just uses the Earth mover distance to basically equalize expression distributions between batches. The Harmony paper that came out is the Harmony method is using clustering like PsyCombine does, but then after that it equalizes, it makes sure that clusters that appear across batches also have a similar distribution, but in latent space, so in PCA space. Now, a key thing in Harmony is that it uses non-autologous clustering. So what that means is that when you're looking at when you're trying to get clusters of cells that behave similarly across batches, what you do is that you're only looking for any given cell, you're only looking at its neighbors in other batches, right? So what that does is that it prevents cells from the same batch from like aggregating together, and then you don't actually get clusters that are shared across batches in order for you to do normalization across batches, right? That is a key concept, and that is a concept that can be used in the general, approach to single cell clustering, as I'm going to show you later on. But just last thing to mention here is both of these methods, almost all current batch correction methods, they rely on some sort of clustering, right? So you're clustering your cells, and then you're normalizing within clusters, because you don't want to normalize B cells with tumor cells, for example, or T cells with tumor cells in a lung cancer experiment, obviously, because you're going to erase a lot of the biological variability. So you do apply clustering. The thing with clustering is that it discretizes the batch effect, and then that can potentially introduce some topological irregularities in the data. So there are some ongoing efforts that some approaches that I'm working on to try to address this, and maybe by the time of the next symposium, I can give you an interesting talk on that. So on the left here, you see that this is an example clustering from a lung cancer data set, and you can see that if you don't apply any sort of like batch mitigation work, basically, you get in some cases you get kind of like mixed phenotypes or a little bit dirtier expression, and a lot of the phenotypes are more like heterogeneous bags rather than like a neater looking heat map. That's important for exploratory studies, because it's you're kind of relying on getting a clear image of what's in the data in order for you to decide what is important and what isn't. And also, while this may work in a biomarker study, because machine learning doesn't care whether you can interpret it or not, it's going to find something, and then you can come back and you can drill into that something and talk about what it actually is. But in exploratory studies, you're not using that as an anchor, right? So you want your phenotyping, you need it to be a little bit clearer. So on the right here this is a clustering looking much neater, same data set, but using the non-autologous clustering that I talked about. So what is it? It's restricting neighbors to cells and nodes coming from other samples only when you're constructing the graphs for clustering, right? So these approaches kind of enable us to improve on how recognizable the clusters are. But of course, a potential consequence of that is that they can bias discovery, because obviously the more recognizable you want things to be, the more you're biasing the outcomes to be what you expect them to be. So in terms of considerations for experiments where clustering is a foundational step for later analysis, there's this kind of balance between like trust and scope, right? So on one hand, you want sort of more confidence in single cell data normalization lets you take a more exploratory approach to like lineage clustering and the phenotyping, and maybe gives you a little bit more fidelity. But of course, you also have to balance that against scope, which is you want to limit basically how you want to constrain the resulting phenotype diversity, right? And that can help you with downstream analyses and promote some shared niches, when you're looking at architecture later on. So wrapping up here, I have very little time left, but the last part, like how we get to niching in the cell space, is we can switch, when we're looking at cells, we can switch, from expression space, we can switch to phenotypic space. What does that mean? So each cell has a vicinity, which is the value of spatial biology, right? We can define a radius, which determines the tissue scale that we profile. So that can be 20 microns, 50, 100, right? And that has an impact on like the structures that you're going to recover. We can quantify phenotype enrichment in the cell vicinity. We can do that in a discrete or continuous way, which means like factoring in how close the phenotype is to the cell, or just if or not it is within a given radius. So that gives us counts versus a continuous value. We can cluster cells on their milieus, which is analogous to how we cluster them on their behavior to get the phenotypes, right? And then we can obtain spatial motifs, which are also called niches and some other terms that you're going to encounter in spatial biology. And finally get spatial motifs, excuse me, finally get the heat map of the motifs where we can kind of see what are the modes of operation in the tissue, or like the modes of mixing of phenotypes. And that can further allow us to filter cells again on interactions, perform set arithmetic like we do with masking, like I mentioned before, and also run collocation scoring, which basically is, that can look like allowing us to filter on cells that are like around the tumor or versus inside the tumor, for example, and run pairwise phenocorrelations using the motif enrichment as paired samples, and then talk about, like whether within an ROI or within a group of ROIs or within a condition, we see more spatial collocation between phenotypes versus less, and then do differential analysis on that as a third layer above differential abundance, differential expression, and kind of wrap up these studies like that, and also break down the TMEs using these quantities as well. And that concludes my talk. Thank you very much. If you have any questions?
Thank you so much, Dr. Simeon. And are there any questions?
Very nice talk. being not a bioinformatician, the talk was very smooth to understand what you're doing and adding the layers just to keep it normal. But I wanted a simple answer to my question. For example, when you explain the normalization, right? You tried to take these pixels, like the images, and then try to do a normalization, and you especially mentioned about the outliers. Let's say if I have something, if I give it a drug, there may be many because usually the cancer cells are very heterogeneous population, that's a problem, right? So there may be very less cells that may have the expression of very, like let's say the immune markers come up so that they can respond very well to immunotherapy. But if I consider them only a small subset, let's say less than 2% of the cells that, as per you, if I consider them as an outlier and try to remove them during my normalization, then I may lose some important data, right? How you set up that threshold when you say that this population of cells may be considered as an outlier using normalization?
Wonderful question. And this is actually something that we've thought a lot about looking at this data for these exact reasons, right? Like you don't want to actually cut out or numb your data set to really important but rare values. So what this does, elbow detection, it doesn't take out cells in any way. What it does is that it's going to, the approach you want to take here is that you want to limit how high of an expression you can get. Like, what is the highest high expression that you can get? The reason why you're doing that has to do with the data structure in high dimensional space and basically the distance distributions that then can impact clustering. So while you want to use that for clustering, you don't have to use normalized data when you're after that doing differential expression, right? So the key thing is here is that you can decouple the kind of normalization you're doing for clustering, but you don't have to necessarily stick with it because you can have copies of the data set and work with that as well. But to answer your question succinctly, you're not taking out any pixels, you're not removing any cells, right? You're just working on the ranges there for the purposes of making sure that distance distributions between cells or pixels in the data set are within reasonable bounds for further machine learning applications, and procedures. You got it.
Thank you. Are there any last questions? If not, thank you so much, Dr. Simeon. And now we will have a very short technical break, just like 2 minutes, 3 minutes, to split the auditorium into 2 rooms. Now we will have parallel sessions, the workshop, the Visium workshop on this side of the room and the main sessions on this side. Our next speaker is going to be Chinta from the Genomics Core, and her presentation today is entitled AGCT Core Single Cell and Spatial Omics Laboratory Services. Warm welcome.
Thank you, everybody. Thank you, Alex. So my name is Jinda, and I am the manager of laboratory services for the AGCT Core. Just want to make sure. Yeah. Okay. So thank you, everybody, for joining us. As you know, there is a workshop going on in the next room. If you did not have the opportunity to sign up for it and you're interested in doing something like that, we are going to be having similar sessions in the near future. So there is a single cell one scheduled for June, and then we might also try to do some more spatial ones as well too, since it seems like there is a pretty big interest in it. Okay, so I wanted to make, I just wanted to thank everybody for being here, and it's been wonderful seeing everybody participating in the event, from everyone from the attendees to the speakers and to the vendors. And before I get started talking about the single cell and spatial services that's offered by the AGCT core, I actually wanted to take the time to say to say a great big thank you. Thank you to those who provided the support to make this symposium possible. It was a great pleasure to work with everyone on the symposium organizing committee in putting everything together. There were a lot of moving parts, but we all came together to work collaboratively. So from the organizing committee, we have Sinai and Gangothri from Cancer Shared Resources. We have Alex and Mitra from Proteomics and Metabolomics Core, and then we have Charlene and myself from the AGCT Core. And special thanks also to Dr. Michelle Jones and Dr. Sarah Parker, who were the ones who supported the idea of having this, this symposium. And one of the goals of the symposium was for the 4 core facilities to listed here the 4 cores listed here to come together and be able to advertise their services. And these 4 cores were chosen specifically due to the complementary services that they provide. It was great collaborating with them on the spatial comparison table poster that we have outside, this great big poster, and also on the symposium. And I hope that we were able to carry this collaboration momentum forward, not only into future ventures, but also in supporting the Cedars research community. And financial support came from Cedars, Cancer Shared Resources, the Board of Governors Innovation Center, which is a part of Academic Affairs, and also Dr. Jonathan Kay. They provided financial support as well as the capital equipment that allow us to, or the course, to bring to all of our users all the cool and leading edge technologies that we provide. I'd also like to thank the single cell and spatial partners that are here with us, that have been here with us for the past 2 days, and also the speakers who have been kind enough to share their work with us, some of which is unpublished, and as well all the volunteers who helped sort of tie the loose ends together these past few days. So thank you so much for to everybody. Okay, so the AGCT core is a 3,100 square foot high throughput genomics facility equipped with the latest next generation sequencing technologies, specializing in single cell and spatial omics. We are part of the Board of Governors Innovation Center, and we are located on the North Campus at PDC Blue Building B130.17. We have 18 main pieces of equipment, which we use to provide over 85 laboratory services and over 30 bioinformatics services. We service, over time, we have serviced over 20 institutions and over 100 Cedars Sinai investigators, and we process over 300 requests annually. Since 2016, we have co-authored 58 manuscripts, although the number of services that we, or number of manuscripts in which we provided a service is much higher. The AGCT core is the core facility for which the most CTSI vouchers have been awarded here at Cedars. 47 have been awarded since 2016, totaling more than $400,000 together. So the team member, or our team, consists of 12 different members, and it is led by Dr. Michelle Jones, who is the interim director, and Simon Knott, who is the co-director. We have 2 main teams that work closely together. We have the laboratory sciences team, which is managed by myself, and we also have Charlene as the lab manager. And we have 2 team leads, one for bulk cell and long read sequencing, as well as another team lead for single cell sequencing. And then we have the 4 biomedical technicians who basically make all the magic happen every day with the complex NGS workflows that they perform. And then the other main team that we have is the computational sciences team. Here we have a team lead as well, and he is supported by 3 research bioinformaticians. The mission of the AGCT core is to gather genomic, transcriptomic, epigenomic, and metagenomic information for investigators. and to make these technologies accessible and biologically interpretable for basic and clinical research scientists. And we do so by developing complete laboratory and bioinformatics analysis pipelines for bulk cell, single cell, and spatial omics. We provide end-to-end services from sample preparation to manuscript-ready figures. And all this work is made possible by the leading-edge equipment that the AGCT core has. At the heart of everything that we do are these next-generation sequencers. So we have the NovaSeq X Plus, which is Illumina's fastest and most powerful sequencer. We use the sequencer mainly for single-cell and spatial sequencing, just due to the number of reads that we need per sample. But we also use it for both bulk cell sequencing as well. On the long read side, we have the ONT Prometheon 24 that allows us to run 24 flow cells simultaneously. Long read sequencing is a newer technology to us, so we are still adding on new services as investigators approach us about new ways that they want to use long read sequencing. Now, that said, we do have several established services already, including single-cell isoform sequencing, which we heard about yesterday. And then we have the Singular Genomics G4X spatial sequencer, which we have just purchased. Rather than sequencing NGS libraries on this sequencer, though, which it can also do, the main use for this sequencer will be for spatial analysis, in particular due to its high throughput capabilities with an emphasis on clinical samples. And then, of course, we have our single cell and spatial equipment. So on the single cell side, we have two pieces of equipment, the 10x Genomics Chromium X and Mission Bio Tapestry. We offer, however, a total of 5 different platforms. It's just that some of these platforms actually don't require any equipment or require a simple equipment like a very specific vortexer, but still just a vortexer nonetheless. And then these single cell platforms can be divided into 2 main categories. So we have our oil encapsulation platforms, and then we have our combinatorial indexing platforms. In the oil encapsulation platforms, we separate the cells into individual samples or individual droplets where reverse transcription will take place. For combinatorial indexing, the cell itself is actually used as a reaction chamber as they move through a series of plates where reactions take place and to uniquely label cells using a combination of barcodes. And another distinction that we can make between these different platforms is that these 2 systems here have use microfluidic channels in a chip. And I'm pointing this out specifically just because it causes a bit of a limitation in terms of the cell size that we can put into the system. So using these 5 platforms, we offer a total of 13 main assays, not including product extensions such as protein detection. So on the next couple of slides, I list each assay that we offer for the platforms, but I won't go over them in detail, but rather I wanted to talk about just some general advantages and disadvantages of the systems. So first, we'll start with the oil encapsulation platforms. So as the trend in single cell is to increase the number of cells profiled, it's important to keep in mind that not everyone has a lot of cells, to be able to put into the system. But we do have platforms where we can start with a low number of cells. So that would be, for example, the Chromium system or the Tapestry system. So in this case, a low cell number is actually something that's actually good. Another advantage of these systems is the possibility of doing multi-omics, such as looking at extracellular and intracellular proteins. So yesterday we had a talk on looking at intracellular proteins. The disadvantages of these oil encapsulation systems are the limitations on cell size for the microfluidic-based systems on the left, as well as higher reagent costs. But to that end, 10x does have an assay that addresses this issue. So for the combinatorial index systems, an advantage is that they allow for massively high throughput single-cell sequencing up to 5 million samples for the parse kit. In general, cell size is not a concern from parse, so large cells such as our cardiomyocytes would work. Now, both platforms, parse and scale, require cells or nuclei to be fixed, and as such, this allows for the preservation of gene expression profiles, as well as for sample batching. These systems are also more tolerable of debris because of the multiple rounds of pooling and washing that we have to do as we move cells from plate to plate. And so there's also, because there are no microfluidic channels that we're working with, there is no possibility of debris clogging a channel. So some of the disadvantages of the two systems is that combinatorial indexing is just a longer workflow because we're just doing so much pipetting going from plate to plate. And then another disadvantage is that multi-omics is not possible. So in the next few slides, I have details about various gene expression assays, and I wanted to highlight important features about them. So in this case, I wanted to highlight this multiplex assay. This is their 10x Genomics' on-chip multiplexing assay. So it's a cheap, easy, fast way to capture multiple samples. The caveat, though, is that you can only capture 5,000 cells per sample. But this assay does use their 3' chemistry. So it's a great way to just run a pilot study before spending a lot of money on capturing a larger number of cells, like 20,000 cells. Another way to multiplex samples is to use the 10x Genomics Flex assay. So in this case, we are capturing transcripts using sets of probes that have different barcodes on them. So these little green probes here, different color probes there. So we add barcode 1 to sample 1, barcode 2 to sample 2, and so on. And then we combine everything together into one tube so that we have one big pool. And then we sequence the samples. And when we do, we will be able to figure out which transcript has which barcode, and therefore we're able to determine which sample a particular transcript came from. Now, if using the maximum number of barcodes available and capturing the maximum number of cells, the assay actually becomes relatively cheap, almost on par with the combinatorial indexing assays. For the parse Evercode system, something notable about them is that they offer a range of cell inputs. So anywhere from 10,000 cells up to 5 million cells. The system, as I mentioned before, is actually good for large cells such as cardiomyocytes. Similarly for scale, we can input a large number of, or a large range of cells also from 84,000 to 4 million. What is unique about this system, though, is that this ScalePlex multiplexing barcode fixation plate. So if we do standard fixation, then we actually start the barcoding step at here at the reverse transcription step. But if we want to increase the number of samples that we can process, we can actually start barcoding at this step here when we do the fixation. And when we do that, we can actually process 9,216 samples in total. So a great number. Now, everything we just talked about was single-cell RNA-seq. However, there are also epigenetic assays as well. 10x Genomics offers a standalone ATAC product as well as a multi-ohm product. For multi-ohm, what's interesting is that we can actually interrogate both open chromatin and gene expression in the exact same cell. So with these different platforms and different assays, it's hard to know what to use for your studies, right? There's just so many different options. So there are things that one would want to consider. So platform capabilities, the number of cells that it can accommodate, or the number of samples it can accommodate, for example. Something to look at also is sample requirements, sample type, or perhaps sample viability. And other things is cost as well. So there's a lot to think about, right? A lot of information. So what we've done, and I know you can't read this, but what we've done is we have consolidated all this information for you into this great big table with all of the different assays that we offer. So we do have a printout of this if you are interested in taking a look at it, and then we can also send it to you as well if you would like. Okay, so next I wanted to talk about our spatial omics platforms, of which we have 6 altogether. So these systems can be divided into 2 main categories, which is our sequencing-based ones and then our imaging-based ones. And then they can also be separated by whether they use whole transcriptomic or epigenomic or a targeted approach. So of the sequencing-based options, we have 2 platforms that interrogate the whole transcriptome. So this allows one to do unbiased exploration of their tissue. However, these assays are costly as it is, and then also on top of that, we actually have to sequence as well too. So that's an additional cost. And another disadvantage is that throughput is low, so you can only process 1 to 2 samples at a time, and also the capture areas are small, like 6.5 by 6.5 millimeters or 10 by 10. The G4X is also a sequencing-based system, but it's different than the other 2 that we just talked about in that this one is a targeted approach. Now, although the capture areas for this one is small, there is actually a large flow cell that can accommodate multiple capture areas. So in a sense, it's almost like a TMA of sorts. What we are, the reason why we're interested in this system is because you can make like a TMA type of thing without actually having to go and get your samples and punch out the cores. You know, TMAs, what we have found for these spatial systems, can be difficult to work with. We get detachment of the cores from the slide or from the paraffin, and so you lose those samples. Now, an advantage of the G4X is that, another advantage is that it provides single cell and subcellular resolution. However, being a new system, the number of tissue-specific panels currently available is limited, and there is no cell segmentation option currently. I do know that they are working on additional panels. The imaging-based spatial platforms are similar to G4X, are similar to G4X in that they offer single cell and subcellular resolution, and they use a targeted approach. But they differ in that they can actually do cell segmentation. So the Xenium and Cosmics have a larger imaging area in which a single piece of tissue can be placed, or if you have a skilled sectioner, then you can actually place multiple pieces of tissue. These 2 systems, however, suffer from having long run times. And depending on the plexity of your panel, it can take up to a full week, sometimes maybe even more. And because of this, it is considered low throughput. Now, to go into some more detail about gene expression spatial platforms that we offer, what I'm showing here is Visium HD. This figure, though, is for Visium HD for FFPE, so their current product that they offer. They just came out with a new product called Visium 3' HD, which captures transcripts with a captures the poly-A tail of transcripts. So this makes it similar to other systems that were species agnostic. However, this one currently only accepts fresh frozen tissue and not FFPE. Another species agnostic system is the Curios Seeker platform. So in this case, we have a capture area of 10 by 10, so it's a little bit bigger than the 10x Genomics Visium platform. And again, this can only be used on fresh frozen tissue. And then Curio also has their Trekker platform that is interesting in that you first tag nuclei with a barcode, and then you isolate the nuclei from the tissue, and then you take that nuclei suspension and you put it into a single cell system. That makes it costly because you're now spending money on two different set of reagents that themselves are actually sort of costly. So. And then we have the imaging-based platforms, the first of which is the Xenium Analyzer, and the second of which is Cosmics. Now, both systems are similar in that they approach the detection or imaging of transcripts, but then they detect the detection or imaging of the transcripts in different ways. So essentially, what they do is they have iterative cycles of fluorescent probes or reporter hybridization, imaging, and then removing of those probes, and then just repeating the process. So something to note about the Xenium system in particular is that after slide processing, the tissue is actually still intact. So we can take that slide and then do additional assays after that. So for example, spatial proteomics, and there's one system that the SMP core has that works well with this. Now, a notable difference between Xenium and Cosmics is that Cosmic has protein panels, which Xenium does not have currently. And then also Cosmic, they just released a whole transcriptome panel. Okay, and then another, something else that I wanted to point out is the Atlas Exomics spatial epigenomics assays. So in this case, what we are doing is we are actually interrogating the epigenome rather than the transcriptome. And this is one of the only available commercially available products that actually does this. And this is something that we are actually working on right now to develop in the core. So again, we have the same question, how do you know which platform to choose? There's so many to choose from. You have to think about resolution and sample requirements and everything like that. So again, we have a table for you to consult, and we do have a poster of this outside, and we have flyers as well, and we can also send it to you if you would like. So these are all of the spatial platforms that we in the core provide, and we also actually collaborated with the SMP core and also proteomics core to add their information as well too. So you know about all the different spatial platforms that are available at Cedars, and you know all the information about them. Okay. Something else I wanted to point out is that the genomics core or the AGCT core offers end-to-end solutions from sample prep to analysis. So if needed, we can actually do the sample prep for you for single cell analysis. And for spatial, we can actually do the sectioning for you. In terms of the sectioning, we will work closely with you to determine like the ROI, how deeply we need to section the samples and things like that. So this is something we do like to be sure that we can provide to everyone. Okay. And lastly, I wanted to say that Cedars had a campaign at one point called Here For You. I think it was like during COVID or something like that. I sort of like this motto, and I like this motto for the core because one of my goals for the core is to make sure that that the core is a resource for Cedars investigators and clinicians. I want it to be a place where people can come to us and we can provide answers to them. So to this extent, we provide guidance and consultations. We will facilitate meetings with vendors so we can all talk together about different, about your project. And then, as I mentioned, we will do detailed discussions regarding sectioning for spatial projects. And we do this at no cost to investigators, which is in contrast to other institutions. We also offer monthly seminar series with vendor talks, and we are going to be working on incorporating user information or user talks as well. And we also offer grant programs for pilot studies using new technologies. So we've done this with ONT, with PARSE, and with 10x Genomics. And these are grant programs that are worth like at least $10,000, if not more. You can run a couple of samples absolutely for free, and we do everything from you, including data analysis as well too. And then finally, we also put on annual events such as this. We've done also other smaller ones, and then we've done open houses as well too. So with that, I wanted to say thank you to everyone. If you wanted to reach out to us, you can reach us at genomics@cshs.org. We are located in Blue Building, B230.17. We have sample drop-off Monday through Friday from 8:30 to 4 p.m. Most people are on the main campus, right? So we have sample drop-off hours there for at Davis 5009 on Tuesdays and Thursdays from 9 to 11 a.m. And thank you to everyone. Thank you also to the AGCT core staff, and these are just pictures of past and present team members. Thank you, everybody.
Are there any questions?
Question.
For example, startup companies to utilize core services for extra fees. Do you have that capability?
Yes, we do. We do. We have worked with several companies in the past, and we also work with other institutions like UCLA, Cedars, or CHLA. Yes, we do.
Great, thanks.
Thank you, everybody.
Our next speaker is going to be Michelle Jones, the director of the Genomics Core, and her presentation today is Bioinformatics Services at the AGCT and summary of how the AGCT can support your genomic projects with bioinformatics services. Warm welcome, and we will start briefly after loading the presentation.
Great, thank you. Thank you, Chinta. That was a really helpful presentation. Those tables that Chinta put up look incredibly overwhelming when you see them up there, but actually a huge amount of time and energy from multiple staff went into making those tables, where we collated the metrics that help you on sort of a choose your own adventure type journey through figuring out, based on your sample type and your budget and your experimental outcomes that you need to help you kind of guide through some of those decisions. It can be very difficult, particularly interacting with vendors, particularly. You know, we have no stake in the game for any particular vendor. Our only desired outcome is a sort of happy user experience as a core. So you can find those tables and lots of information about the core services, including the template text you need for your grant applications in the Lab Archive notebook. And I don't know how many people here use Lab Archives, but I strongly encourage you to use it. Dr. K has put a lot of effort and resources into getting that set up for us. And it includes huge volumes of information about the genomics core. It's much more up to date because we have the ability to update the notebook ourselves internally and not our website. The website gets updated by the media and website group at Cedars, and if any of you have tried to update a website here, you can imagine how that goes. But if you want something that's new and up to date, just log into Lab Archives. You can automatically on the left-hand panel navigate to the AGCT Genomics Core, and you can get all of that information, including those tables on there as well. I know I had sat down, I did some spatial experiments, and I sat down with those tables, and it really helped guide me as to what I should be using for my academic lab too. So I started helping at the Genomics Core about 2 years ago. I've worked at Cedars for a very long time, and I had been a Genomics Core customer for a long time. And I started out helping as a co-director in the bioinformatics group. And so we spent really probably a good year fleshing out the hardware and resources, personnel, and workflows and pipelines to try and sort of catch up a little bit with the investment that had happened in the sort of what we call the wet lab or the bench side of the sciences. You know, mainly because anyone who's doing genomics work, you're probably excruciatingly aware that the data analysis is the beginning of the hardest part of a genomics project. The bench science team and the bioinformatics team work really close together, hand in hand. But a 6-week sample prep and sequencing run experiment is going to be the start of a 6 to 18-month analytic journey for people who are generating data. And there's many more bench scientists than there are bioinformaticians available. So we really acknowledge that it's a huge pressure point for many of our customers, users at the core, both internal and external. And so we try very hard to staff the bioinformatics team in a way that we can provide really standardized services to suit as many investigators as possible. And the talk that's going to come after this one is going to be much more talking about customized analytics support that's available through the BDSSR. So I'm going to talk about what we can offer you as a core ourselves, but I just want to make sure everyone sort of knows that we go back and forth between the core groups. We all talk to each other. We all meet on a regular basis. And so if we know that we can't give you what you need, we will refer you to someone that can help you out and give you the support that you do need. Almost every application that you send into the genomics core is going to pass through a bioinformatician's hand. We have 4 bioinformaticians in the team, and they're all incredibly skilled, but with diverse areas of specialty. Casey Hansen is the team lead for bioinformatics. And then we have areas of expertise in single cell, general bulk sequencing, long read sequencing, and we have a new team member coming very shortly who's going to bring some spatial experience, and she'll be the lead for the spatial projects. We have core bioinformatics services available in 2 tiers. One is a tier, what we refer to as tier 1, which is a very simplistic data delivery option. And this is really for core users who have a bioinformatician of their own. So if you're a lab where I have an academic lab as well as working at the core. In my academic lab, we have all bioinformaticians. I have just a bioinformatics lab. So I only use tier 1 services from the genomics core. So that's going to be essentially data delivery. We use as a sequencing service like you might somewhere like Novogene or an external service like that. But you'd have a happier experience if you came to the genomics core than Novogene, as someone who's used both, I have to say. So we have basic data processing and data delivery. It's really cheap, it's really fast, and I'll talk about why in a little bit. So some of our users, that's absolutely the right fit for them, but that's really the minority of people who are coming through the core. The vast majority of people need some support in terms of bioinformatics and data handling. So that's going to be basic data processing with some kind of standardized analysis pipeline. And as you could see from Chinta's slides, we have this enormous array of equipment, and it's the breadth of services that we deliver in our genomics core at Cedars is far greater than any of the genomics cores at any of the other academic institutions that have decently sized genomics cores. And so it's a challenge to provide standardized pipelines to fit all of the projects we take, especially given some of the new platforms. The ONT Prometheon has given us a real run for our money because they can literally sequence anything, which means we have to figure out how we're going to analyze absolutely anything as well. But that's kind of what we're focusing on at the moment. Tier 3 is a service that used to exist if you've been a long-term user of the core, which was customized and analytics support for individual projects for investigators. And that tier of service is no longer available from the genomics core. If we have time, we can help a little bit in where you can hire a bioinformatician by the hour to give you some level of support. But what used to be tier 3 service, if you've been our customer for a while, that level of service is moving over to Nick Tatnetti's group in the BDSSR. So the subsequent talk after this is going to really address those. So for about a year, we spent some time really doing a stock take and thinking about what our bioinformatic resources were like at the genomics core. And it turned out that we kept buying newer and bigger sequences and never buying newer or bigger computers. And so that was kind of holding us back a little bit. So when we brought in the NovaSeq X Plus, which has an onboard Dragon server, we also brought on an offboard Dragon server that runs independently with a 250 terabyte external data storage server. So we can run everything locally within house on a 10 gig network without streaming things to the data center down at El Segundo for analysis. And that has sped things up enormously for us. The offboard Dragon server has all the standardized Dragon pipelines on it that Illumina is responsible for the version control, which takes a lot of the version control work out of our hands. But it means that if we have customers who are submitting batched samples over significant periods of time, that we're going, that's just a very loud cart in the hallway, right? Okay. I don't know if I should be alarmed at that noise or not. Where they have batched samples being submitted over significant periods of time, we have really good ability for version control over those different runs, where we're not going to be updating pipelines because dependencies broke and having problems with batching samples over time. So that's something that really helps customers a lot in terms of having these large, very high volume projects go through the Dragon pipeline. We are able to offer internal data transfer, whether it's tier 1 or tier 2 to you. It used to be via box, so if you've been a person who's been using the core for a while, we used to be able to deliver via box and the genomics cloud. We've all transitioned off of, but we've almost all transitioned off of box, and so now we're working through the process of having SharePoint be the place for you to pick up data instead. Or if you have bioinformaticians in your lab and they're already proficient with using the high-performance computing cluster, HPCC or Isilon, and if you don't know what they are, then you probably are not that person, so it doesn't matter. But if you have that capability, then we can just transfer it internally into your workspace on the Isilon, and then you can just pick your files up and kind of move along as you need to. We do have the ability to deliver data on encrypted USB drives. EIS is not wild about this as an option, and for understandable reasons because of data security. This is just research data, so we can offer it to you if we need to, and our external customers, who often tend to be pharmaceutical companies, private biotech companies, tend to select this as an option because they want encrypted delivery rather than, we can't give them access internally at Cedars. So Chinta talked sort of generally about sort of general sequencing services and then single cell and spatial services. So I'm going to divide mine into sort of two sections that follow that kind of idea. And I always love it when I see these figures, particularly when wet lab, when sort of bench scientists prepare them, where they have this excruciatingly detailed workflow to get from having a sample to sequencing, and then the final step is just a screen with bioinformatics. And so, where it's like the first 6 steps there take like 8 days, and then the bioinformatics part is like a 4-year PhD for someone probably. So it's important that we acknowledge, number one, it's going to be really hard, and it's going to take a lot of time, and that's okay because we know it's going to be. Don't look at these figures and think, oh, this is going to be fine. It's probably going to be tough. So it's going to be hard. So I'm going to talk about what happens after the word bioinformatics, which is kind of the most challenging part. So we split our services into sort of short read and long read sequencing. We have one team in the lab that drives those services forward, run by Ben, and then we have our single cell and spatial team, and they drive those services forward. So I'm going to talk about short read sequencing first. So that's our classic Illumina sequencing, I think is what everyone calls it now, right? Where we have an Illumina NovaSeq X Plus with 2 independent flow cells, and we can run 1.5 billion up to 25 billion flow cells independently of one another. So some of the waiting and turnaround times that used to happen with the NovaSeq 6000, where you were waiting for a flow cell to fill before we can send a run, are really largely alleviated now because we can run those 2 flow cells whenever we need to. So that's helped return times and delivery times a little bit, but it has meant that we make more data. So we run more runs now, and they need to go through that Dragon server. But because it's local, we can just speed data through. So if you're a tier 1 customer and you're just submitting samples for sequencing and you want data delivery, we can really send it out to you pretty quick. But we will still put hands on the data a little bit for you. We're still going to run some pretty standard QC, so give you some comments about the number of reads. All of these are automated pipelines that the very talented bioinformatics staff have built and maintained over the years. So one of our primary services, probably bread and butter still of the genomics core, is bulk RNA-seq. We still do a lot of that. So when we think about bulk RNA-seq, some of you would like to submit samples that are tissues, some of you would like to submit RNA, some of you would like to submit libraries, and we can take the samples at any of those 3 steps of the experiment. And then we're going to, plan our run out, think about the barcoding structure, generate the reads that you need. We run 2 by 150 as a standardized kind of format across the flow cells as much as possible, understanding that some customers are completing experiments that might have been run in 1 by 100, 1 by 75. I will always advise you to no longer do that. If anyone is still running anything in any format that's not 2 by 150, please be prepared over the next few months to year to let go of that as a format. The pricing will be cheaper if you move to 2 by 150 because we run those flow cells much more often, and you'll get your data back much more quickly. So once we get the sequencing data off, we're going to run very standardized pipeline for you through Dragon, and it's going to be read alignment. We're going to do feature counting or gene quantification, gene identification and quantification. We can then deliver to you as tier 1. We can move forward and start doing some tier 2 for you. So we're going to be running principal components analysis, generate clustered heat maps for you, do differential expression analysis and give you volcano plots. And these were publication quality figures. There is some cap and some limit on how much we can go back and forth and redo things for you. So we can do differential expression analysis with a set or limited number of groups. If you would like 18 pairwise comparisons, that's not going to be within our standardized package. And I see you laughing. That is literally a request we got last year. It's not something that we can offer within a standardized package because it's we're rerunning so many analyses. It's just not tenable. But we can always work it out with you, and when we have time, we can run as much as we possibly can. That will always be our long-term goal. We also offer bulk attack and chip-seq, where we're going to perform the standard read alignment, some QC for you, some peak calling. And we're using the Anshul Kundaya's pipeline from Stanford up north, differential peak analysis, so you can find the areas that are differentially bound in your genome or differentially open in the genome, and then do some motif and enrichment discovery for you, so you can think about what are the motifs under your transcription factor of interest or under your active enhancers or promoters in whatever sample type that you've chosen to work in. And you'll have the same set of figures being delivered that are going to be volcano plots, those nice PWM, position weight motif plots for you, and all those kind of standard metric figures. The other main short read sequencing product we offer is just going to be standardized bulk whole genome, whole exome sequencing. So we run a decent amount of this in the core. I run a lot of my own samples for my own sort of germline studies in the core using this as a product. It's going to be the standardized Dragon pipeline for variant calling, where we are calling SNPs and indels as small variants and then structural variants, with the Dragon pipeline, which has been built off the back of the GRIDS pipeline, which is phenomenal structural variant calling pipeline. So if you're interested in looking at complex genomic rearrangements in either germline or somatic samples, the Dragon pipeline performs very, very well. We did a nice benchmarking project, 2 years ago, as we were getting ready to purchase the server, and found really, really strong performance from it. And then we have variant annotation options. Some customers or users will have a specific variant annotation pipeline they would like, or a specific variant annotation database that they would like. And, you know, we can interact with you in some ways to customize what we do a little bit. I personally have a set of tools that I like and use in my projects. I'm always happy to recommend them, but we'll accommodate your requests as much as we possibly can. And then the Oxford Prometheon P24 device, when it came into the lab, kind of shook things up a little bit, because every time we want to run a project, it feels like it's the first time we've ever done that type of sample. So basically, if a protocol exists for the ONT Prometheon, Ben can seemingly get it to work. So because the Prometheon is direct molecule sequencing, if you've got some sort of nucleic acid, we can throw it on and sequence it. The library prep is very, very quick, and the data turnaround time is quite fast, but the analytic timeline is very long. You generate incredibly long contiguous sequencing reads, which it's difficult bioinformatically to handle this data. So I would just give you a little word of warning. My lab is doing a lot of this data, and it's quite challenging to work with. The amount of data that comes out from these types of projects is also very overwhelming. So you have to be ready with lots of data storage and lots of external hard drives and strong computational resources to work on it. We've been doing long reads for bulk germline and somatic whole genome sequencing with 30x coverage in the germlines and 60x to 100x coverage in the tumors, both in methylation-aware mode and not in methylation-aware mode. So you can get the whole methylome off of your whole genome while you're doing your sequencing. It comes in a single contiguous read, and they're adding additional base modifications all of the time, apparently. So we're picking up new things. We also can offer the long read sequencing as ultra long read, depending on how your sample prep is done, and we can help guide you through that sample prep process. Ben is quite good at interpreting a lot of the femtopulse reads and all that kind of stuff, so we can make sure you get good quality samples in first. And then we have some experience in annotation of the structural variants and trying to get some interpretation going for things like that. The tier 3 level work with the ONT is really not available at the moment through our genomics core. It's an incredibly laborious type of data to work with. But if you have some expertise yourself, it is incredibly fun data to work with, especially if you do any kind of structural variant work, which we do. It's super fun, and you're going to find very novel things that are really exciting. So we're doing DNA sequencing, RNA sequencing. We're also able to offer long reads. So use the ONT to sequence your single cell RNA-seq libraries, so you can get transcriptional isoform level data now out of your 10x single-cell RNA-seq libraries per cell. So you can look for transcriptional isoform variation between individual cell types. So that's a really interesting type of analysis to do, particularly if you have very heterogeneous tissues. And you're having difficulty subsetting out clusters of cells. The isoform level analysis will help you be able to do that. So I recommend giving this a try if you're a single cell RNA-seq person. We have also been doing some what would be called amplicon-based sequencing as a very broad term, really targeted sequencing. So there's an amplicon-based method on the ONT Prometheon, and we can offer that service to you. There's also a CRISPR depletion method or an enrichment method that's available on the ONT, and so we've delivered this as a service actually to my academic lab. We've received this as a service, and it's been quite successful. And then we've been working on metagenomics projects with Suzanne Devcoder and people down in the Microbiome Institute, and so we've been able to help with some of the analysis, getting some of that going for the long reads as well. So we have quite a lot of products available in the standard sequencing sort of arena, and the bioinformaticians fortunately have really been able to deliver analytic services for all of those as well. And then single cell and spatial are really the other two main areas that we run a lot of samples in. And this is the single cell workflow, which has a similar theme to it, where there's about 20 steps in the wet lab and then just demultiplexing and data interpretation for the bioinformatics. But the bioinformatics for single cell is incredibly complicated. So if you've already been a customer and you've done single cell with us, you will see what the workflow looks like. We have a wonderful tier 1 option where we will just send you all the files. And you and your graduate students and postdocs are going to have a great time learning how to do single cell analysis. But we can also help you through that process if you don't have that. And you would have heard from a speaker, Rob Barrett, yesterday, where he was a customer that received a little bit of more support and some tier 2 support from us in order to get those results that he was presenting for you yesterday to help give him some clarity on those iPSC-derived gut organoids. So we're going to run standardized analysis like feature alignment, read alignment, feature counting, and then help you cluster the cells. This is a much more iterative process than standard bulk sequencing. Because we're not experts in cardiovascular disease or iPSC-derived gut organoids or breast cancer, whatever your cell type is, identifying the cell type clusters and the genes that should be defining those cell type clusters is not really in our wheelhouse. We're going to need you to help us. Send us a reference paper. Send us the list of genes that define that cell type. Send us a publication that's shown us that previously so that we can help apply those to our own analysis. We can't go to the breast cancer literature and understand that this lab is the top of the field and they have the best gene list. We need you guys to tell us that stuff. So we'll have a little bit more iterative analysis within the tier 2 for single cell so that we can help get you down into cell quantification, differential expression analysis, if you're interested in biomarker discovery, and some of the pathway analysis, which is so important for interpretation and biological insights. So there's these quite, there's really a vast array of figures that we're going to generate for you as part of our standardized workflow. If you select tier 2, these will just get generated and sent to you. But as we look at them and work through them with you, meet to discuss the results, start to deliver the results to you, we can go back and more thoughtfully work on these together. The trajectory analysis and the pseudotime analysis can help with lineage tracing and identifying developmental pathways for cell types depending on the structure of your experiment. So then the last slide I have is to talk about the spatial profiling methodologies. So that's been the newest set of services that have come online, and we have those 3 devices that Chinta spoke about, Cosmic, Xenium, and Visium. And now we're going to have this brand new device coming in from Singular. So I'm not even going to address that because we don't have a bioinformatics solution for that because we don't have the device or the data yet, but we know that's coming. So that's going to be our summer project to start thinking about how to handle some of that data. We have a bioinformatician coming in June. She's going to join the team, bringing some spatial expertise with her, but we already have some because we've already been generating this data and practicing on pilot projects and the first few customer projects that have been coming through. So the sequencing-based methodologies like Visium, for example, we're going to perform basic tier 1 type analysis where it's essentially same concept as single cell, where it's read alignment, it's feature counting. We're going to identify the boundaries of each cell, which genes are being expressed within that cell, and what the gene expression level is going to be. And we're going to share all of that information with you. We can work on batch effect normalization and correction with you if your project is being done over a period of time, which of course we don't want that to happen, but we acknowledge clinical samples, that's how the collection works. And then the standardized downstream kind of analysis is going to include things like Leiden clustering and identifying cell types, quantifying cell types. But because this is spatial work now, we're going to help you identify relevant niches, cell neighborhoods, cell-cell interactions, and genes that might play critical roles in the cells that are uniquely interacting together as a sort of neighborhood or a cohort of cells that are co-located. So this is what we are able to offer. There's so much more analysis that happens though after we finish this set. You know, we're sending you tables, we're sending you figures, and then really the customer goes away and does all of this deep diving for interpretation and thoughtful analysis and designing what the next experiment is going to be. So after we finish this type of analysis and we start handing it off, it's going to go off to really probably BDSSR and Nick Tadnetti's group, especially if you need lots of iterative analysis back and forth, we are, there's an analogy that I heard someone use once where we're trying to take a sip of water from a fire hydrant in terms of trying to get data pushed out of the genomics core. So that's why tier 3 services are no longer available. We would need probably 12 bioinformaticians, honestly, if we were going to continue to offer that as a service. So in trying to delicately sip from our fire hydrant of data, this is the point at which our analytic services typically end for most customers. And it would be Nick Tadnetti's group that would take on the challenge after this point. The team is led by Casey, and then we have Sarah and Josie also in the team, and Sarah's been with us for several years, and Josie I think for 2 years now, so many of you probably already know them, and they're incredibly helpful, very kind, very patient people. I really encourage you to chat with them and get their input, particularly at the point of experimental design, that often having a bioinformatician be part of the design of your experiment will make your experiment better, because they will know, they're used to fixing the problems at the end of an experiment, and so it means they're really, really good at spotting the problems before they've happened and preventing them from ever being introduced. And then we'll have a new team member come on board in just a couple of weeks as well, so there'll be a new name on the slide next time. That's it for bioinformatics services. Anyone have any questions? It's based on projects. So we do not have a hire a bioinformatician by the hour service available at the core, but the next group who speak, BDSSR, they can offer that as a service. Yeah, we are just so overwhelmed, honestly, with data delivery. We just don't have time. It would be that we would be doing hourly bioinformatics consults and not delivering people sequencing data, which we for sure cannot get into that position. But the next group can offer that, so they're going to explain what that looks like for you.
Our next presenter is going to be Hongyu Liu from BDSSR, and the title of her talk today is going to be Introduction to BDSSR Services and Opportunities Applications in Single Cell Analysis. Warm welcome.
Hi, yeah, thank you for introducing the BDSSR. So we're pretty collaborate with AGCT course and also the Spatial, thank you, and for the Spatial core together. So we want to introduce the Biomedical Data Science Share Resource, short for BDSSR. And there's a QR code you can scan to learn more about our core systems. And we kind of like focused on tier 3 analyzed. We are working on the large scale data projects at tier 3 is separate with the tier 1 and tier 2, which more focused on basic analyzed, basic nesting sequencing analyzed, and provide the data delivery. We providing the bioinformatics and machine learning and some AI servers for customer analyzed. And we aim to provide both advanced research support and robust training and education in the form of seminar and workshop. And our mission is to provide a comprehensive suite of service design to support the whole Cedars community members in navigate those complex landscape of multi-omics technique and AI in cancer biology research. So we focus on storing, retrieving, analyzed, and organizing those biomedical data, including genomics data, EHR data, and so on. Here is our team. We're kind of like a small team right now since we are new build. We're only like 2 years old right now. And there's a Nicholas Kennedy is renowned for his contributions to drug safety, drug discovery. And we just released one data set called OneSet from his lab. It's a database relative to data set effect. And he also authored over 180 articles of working for the integrate of the real world data set and high dimension biology data, scanning medicine, system biology, machine learning, and bioinformatics. He's leading and oversee all those BDSSR projects. And here is me, Hongyu Liu. My whole career path is focused on the cutting edge next generation sequence data analyzed, and especially focused on single cells RNA-seq level. And also have some expertise in spatials and ataxic dataset analyzed, and try to get involved in diverse multi-omics data. And here's my collaborator, my colleague Kevin Zhang. He's specialized in developing pipeline for next generation sequencing and SMP genotype data analyzed. His expertise extend to the applications of machine learning and deep learning techniques in various forms of biology data. You will hear the talk from him about LLMs, the large language models in clinical data set later. We want to highlight the service we provide for the whole community, including the customer analyze consultations and support for potential collaborator. Then we can assist you with experiment design to generate and interpret the preliminary data for the paper and the grant support, and contribute to your paper method, create those high resolutions figures for your proposals and papers publication. We also have abilities to download those Omics data sets from public repository, loading and query the data from database systems, and transform the data to different necessary format. In deploying those AI clinical methods like SCBIRD, DNABIRD, and others, cancer-relative AI models to your study and application. We have the internal data set available, like molecular twin data set, Tempest Lens data, and also have exercise for OncoBiobank data sets. And for the external data set, we also get exercise for the All of Us research data, the TCGA with all know public cancer data set, and other highly accessed shared database. We have our own HPC, high performance workstations to storage data, and also using Amazon AWS instance for web-based needs. We also provide the version controls code to repository from like GitHub if you need that one's for publications, and we share the results through emails and your like preferred file exchange method. And right now, we don't using Box, we're using OneDrive for the larger file transfers inside and outside. For here, I just want to give you an examples like our probabilities in single RNA-seq level. So this is the collaborators with Dr. Authorities about understand the function of the ELP1 genes, since ELP1 could cause sympathetic neuron death and sympathetic nervous dysfunctions. Then for here, we apply the single RNA-seq data analyzed to try to understand the function of the from like ELP1 knockout samples was the wild type samples. So for here's the basic probabilities we can have is to help you build up the pipeline to analyze the data things from like the raw data set we have for single cells, generates the FASTQ files, then do basically like quality controls to mapping to the reference genomes. If you have your preferred like reference genome, we can do also generate some like new target reference and then mapping to the particular areas you like, and do some expression qualifications. Then we also have ability to do downstream analyze, including filter the low quality cells, consider the mitochondrial genes, cell cycles effects on your data. That will be case by case filter method, and also will help you using some like machine learning method to identify the doublet cells to make your cell type annotations more clearly and learn the pattern of your each cell type more clearly. Then we also will apply different methods to normalizations and scale your data set. Then do the same kind of like dimension reduction, clustering analyze, do some like cell type DEG, the differential expression analyze to find the biomarker, pathway function analyze, do cell interactions. We also would have the abilities to do like cell drug discoveries using like single cells RNC data set. For this particular analyze, we also provide a Shiny R, which is highly recommended by our collaborators since it's easily for the people's not in the bioinformatics areas to see how their data will looks like, especially help them to learn the biology meanings under their data set, under each cell type. Then you can see from here, you can select your data set and select the cell type you have in your data. And for here, so you can enter the features, which means genes right now. And then you can highlight those purple dots in the graph to see how there's dot expressions levels in each position or each cell type, which will help you to see your data easily, by the way. So that's one of our probabilities, and just show you a case. And we provide FTE resource to support your funded research project. Please contact my personal emails or scan this QR code to learn more about our course. Yeah. Yeah. Thank you.
Are there any questions? If not, we will proceed with our next presenter today, Kevin Song, and his talk today is entitled "Serving Large Language Models for ICDO Site Classification from Unstructured Pathology Text." No idea what that means, but I'm about to find out.
Okay, can everybody hear me? Okay, my name is Kevin, and as Michelle has explained, we are doing the back and forth with collaborators and using kind of like the tier 3, kind of more customized data analysis for any grants and paper publication. So today I want to highlight one of the projects that I've been working on to fruition. Hopefully gets published in a conference coming up in August. But it's the use of LLMs to do ICDO site classification from unstructured pathology text, which is free text. So cancer remains a leading cause of death worldwide, and an accurate cancer diagnosis is very foundational to doing any cancer treatment, research, clinical trial enrollment. However, this is sometimes a bottleneck in research and care because in pathology reports, there are a lot of, it contains cancer diagnosis information. A lot of times it's in free text by different pathologists who were trained maybe in different expertise or schools that writes things a different way, or maybe embed a lot of references to their path reports. The ICDO third edition is established by World Health Organization. This is not to be confused with ICD 9s and 10s. Those are, like obviously we, as probably most of us understand, for billing purposes, for diagnosis. The oncology edition is a key framework as a subset that categorizes cancer diagnosis. So here I provided a couple example. So it talks about the site histology and behavior. On the left, C34.1 is a site code, which indicates upper lobe lung. And it's important to note that the 0.1 usually is a more specific region of that site. So there will be, for example, in breast, it will be C50.1 through I think 9, and it will indicate different quadrant of the breast, the upper or inner quadrant of the breast as well. And then the 8071/3 is the histology/behavior code. Here is the squamous cell carcinoma keratinizing, which is the 8071. And then if you take off just to look at the 807, it's just any non-otherwise specified squamous cell carcinoma. And then here I also provided a quick code for the behavior just to see the different behavior of this tumor, whether it's benign or it's malignant or it's like carcinoma in situ in different states. So ideally, categorizing a cancer diagnosis into the site and histology code. So the main problem here is that the manual ICDO coding is costly and laborious. Currently, the cancer registry does this, and a lot of the times it takes over 6+ months. You see for cancer diagnosis procedures, and that leads to kind of missing this window of clinical trial enrollment, and also kind of like diminishes the data for any research. However, as you know, AI is the buzzword. We don't want to like promote just like AI replacing our job, so we carefully put a person next to a computer to show that it's AI assisted, and like in some ways training these transformer models, and now even leads to bigger models like foundation models to help coding these ICDO cancer diagnosis that leads to higher enrollment and not missing this window, but also getting more data and more real-time support. So as the buzz is, all these GPT-4, Llama 3, DeepSeek, what have you, we want to understand whether LLMs can solve this problem. So I can break it down into 2 questions. One's easy, one's hard. Can LLMs effectively distinguish these path reports, whether it has a positive or a negative diagnosis? And a harder question is, can they actually give those site and histology behavior code and actually give you and classify the codes? So in this, I guess, paper and this project, this is kind of like the methods overview. We have collected from Cedars 58,000 pathology reports that has a ICDO classification from the cancer registry. Then we kind of doing 2 big experiments. One is prompting, one's fine-tuning. Depending on kind of your knowledge in this, I'm gonna go just explain briefly what that means. Prompting, just for example, is kind of like you logging into ChatGPT and asking a question and kind of sort of interact with the model itself, in which 0 shot is basically you're kind of just relying on what was the pre-trained materials in the model and kind of converse with the AI, so to speak. And then few shot prompting is you can then give some examples. So for example, if you ask a question and you wanted to format something specifically how you wanted to format, then you would give an example. In our case, that we give an example of the pathology report diagnosis, and then with a ground truth of, say, C50 as a breast cancer, then they'll know, it will have an example to help guide the model to produce, I guess, unseen pathology report classification. And fine-tuning is a method, if you're familiar with machine learning, that essentially now it works in this multi-class classification because we have actually a lot of sites. I didn't include the bar chart here, but there's actually 273 sites in the 58,000 pathology report. So we're trying to distill all of these multiple sites into around 65 general site categories. And with fine tuning, you're giving it some training data essentially with what the actual diagnoses are, the ICDO code, and you train the model, and then now it will kind of learn. It kind of adjusts the weights of the neural network, and also then we'll be able to kind of categorize ICDO code for unseen pathology reports. So that's kind of the overview of what we did. And this is an example of a system prompt. So like it will be very laborious to like just chat with I guess ChatGPT and keep asking questions. So like we can embed to the system prompt a prompt for zero-shot prompting. So you first kind of give it a role. So here the AI is the cancer registry that would kind of categorize and give a summary of all these treatment disease status. And then given the pathology report, which will be embed to the prompt, please determine the ICDO site histology behavior code. And then a couple of things that you can kind of this is kind of where the art of prompt engineering comes in. You could, you realize I used the word please for when I didn't actually put it there. But you could be nice, quote unquote, nice to the AI, or you could have it think step by step and then try to justify answer so that it would be easier for us to see what the LLM is thinking. So another cool thing we did is that you could tell it to format the response as a JSON object. And then in that case, because of the generative nature of LLMs, sometimes it kind of can spits out a bunch of paragraph and without really an easily kind of structure manner. So here, I was able to use JSON as an object to denote the site histology behavior, negative finding, just so that it's a Boolean to see if it's true or false. And rationale is kind of like for us to take a look at the response to see why the LLM thinks that this diagnosis is fits that code. And then confidence is kind of just like for us to evaluate what it thinks and how confident it is in the response. So the performance for the easy questions, which is just to see if the pathology report has a positive or negative diagnosis. GPT-4 and Llama 3 are the two models that we used. So here is the results for the zero-shot and few-shot experiments. And as you see that for few-shot, which is, again, giving some examples for GPT-4, it actually didn't perform as well in terms of accuracy. And for Llama 3, actually, for zero-shot and few-shot, performed the same and pretty well in terms of like being able to tell whether if you give it a pathology report, can it spot a positive or negative cancer diagnosis. As for the hard question, we looked at comparing the prompting methods and also the fine-tune methods. And so for GPT-4 and Llama 3, the way that this is evaluated is to kind of extract the LLM response to do kind of an exact match to the site, but also what I will use the term group term, which is earlier when I gave an example, you can shorten the code and that shows a general area of the site instead of a very specific site. And so for that, we used that as a comparison and looked at the accuracies for all of them. So GPT-4 and Llama 3 didn't perform as well as the fine-tune methods. There's a reason why, because fine-tune, obviously, because we have labeled data, as you guys all know, like data is king. Like if your data is good and you have good ground truth and gold standard data, the model will learn and will be able to do well in these multi-class classification. So the best model is a fine-tuned clinical Big Bird model. And you see that Llama 3, even though it was fine-tuned, did not perform as well. So just to summarize the key findings, the easy part, LLMs can definitely spot out positive or negative findings. And then for the hard question, we see that Llama 3 kind of excelled at the binary classification, but then kind of lacked some of the granularity when kind of giving a detailed site coding. GPT-4 did moderate well for both tasks, but it was lower for a few shot prompting. One kind of I guess one theory that we have is that if you give it, so for the prompt, we would embed the entire pathology report, and that could kind of include a lot of noise. Sometimes some pathologists would give a whole reference list of why some of the genes are important to look at. But that could be very veering far off from what the actual site classification is. So there might be a lot of noise, and that could kind of explain why actually few shot giving some examples didn't perform as well. And then as you can see that the detail codes, the performance kind of like varied, and CBB, the clinical Big Bird model, achieved the best accuracy. Some of the limitation, as I kind of alluded earlier, is the prompt engineering. It just requires a lot of iterative process. And for here, we use a same prompt for GPT-4 and Llama 3, but honestly, you could, if you really want to, do a prompt for each of the model if you know it's going to perform well for that specific model. And for evaluation consistency's sake, we use the same regular expression strategy in Python to extract those responses. And then in general, like Llama 3 loves to kind of generate a longer generative kind of response. So it was a little harder to normalize that. And then we also looked at the AUROC, as some of you may know for AI, that's the model, one of the key metrics to see how the model is performing. But only the CBB model has a per class probability output. So we weren't able to use that to compare for every model. And then, yeah, so that's why we use accuracy only. And then mainly is there's fundamentally, I guess, hard to compare apples to oranges with prompting and fine-tuned classification. But we kind of do our best in this paper to look at the accuracy. And lastly, just some future directions, and LLMs can be hopefully you use this model to integrate to cancer data workflows, especially point of care, with real-time or near real-time ICDO code generation that will help clinical workflows and clinicians to make the best decision. And then these structure outputs from LLMs can be shared with the different database and clinical decision support tools. And then to tie it back to single cell, because obviously this is why we're here, is that actually our next speaker, we just, KJ, we had a quick conversation yesterday about foundation models and all the hype. So there's a lot of like single cell foundation models out there that essentially claims to do really good job at cell type annotation, things like that. But I think from this project, we can see that fine tuning and having the right data is actually very important. So our team here at BDSSR is happy to help, collaborate, and also just to try the different new models that are out there and fine tune to do classification, kind of these projects with you guys. So yeah, this is my talk, and thank you for all the organization from the AGCT core. And yeah, just thanks Nick and also my team here, Hong Yu and Jose and a couple of people, and also all the collaborators. I see Ty here, just to keep the lights on for us. And thank you so much.
Don't need this. So you're trying to do some pathology reports classification, right?
Yes.
But from my understanding, for pathology diagnosis, why not trying to use vision LLM instead of, because like for the like golden standard is like from the the pathology images, right? So it's like if you can link with the images, classification or detection,
Yeah.
you don't really need the reports, right? The reports is not a real. Sometimes like the
Processed
clinicians.
by a pathologist. Yeah. Yeah. So that's definitely great. One big reason is that definitely takes a lot of computational resources to process images. But actually, one of our collaborators here, Ty, he's requesting a lot of the MRI images, and we're actually are in projects to build like vision language models using like CLIP or different modalities to do that. But I think this specific project, we're just trying to see if we can, like use the least amount of computational resource to get to a pretty high accuracy in doing ICDO site coding.
Thank you so
Thank
much.
you.
We will move on to our last speaker of the day, and I would like to invite KJ Won with the talk entitled Deciphering the Mechanisms of Cellular Communication and Fate Determination.
Thank you, and hello everybody. This is the last talk before lunch, so I try to make it brief, if possible. Whenever I have data, I guess you have interest in single cell or spatial transdynamics data at least. Whenever I have data, I feel happiness if I treat it in a different way. If I treat it in a normal way, I feel a little bit boring. Okay, so when it comes to cell-cell interactions, well, for single-cell data, a typical way to treat it is using dictionary, which is ligand receptor. Of course, it works, but when it comes to cell-cell interactions, ligand receptor mediate pathway is just one of the ways cells use for communication. There are other ways, for instance, cell contact, but cell-cell communication for cell contact is missing for single-cell RNA-seq because the concept is to separate it. How can this communicate? So single-cell RNA-seq, ligand receptor signaling, possibly yes, even though it's not accurate, I may say. But for cell contact or extracellular matrix, you cannot detect it using single-cell RNA-seq. Well, then can I tweak it a little bit to study it? Well, we thought about doublet, not single cell, it's a doublet or physically interacting cells, so I may say. Then we may be able to study cell contact mediated gene expression for ligand receptor, possibly. So we did it in collaboration with Joshua Brinkman in Copenhagen University, and he is interested in embryo development. Okay, why not applying our approaches for embryogenesis? And in mouse embryo, we obtained a single cell and doublet-seq, or people called it physically interacting cells. It's obtained from mild dissociation way. So to make the long story short, okay, these are single cells. So neuroprogenitor cells express neuroprogenitor markers, definitive endodome cells. Do you see my cursor? No. Definitive endodome cells express some definitive endodome markers. And interestingly, these doublet or physically interacting cells showed definitely endoderm markers and neuroprogenitor markers. At the same time, they showed some different genes which are not observed in each cell type, but only in these doublet. That includes GCX, NKX 2.1, and LHX5. And we try to validate it in many ways. One of the ways to validate it is just co-culture of these two cell types, and we found that these cells, these genes were induced in co-culture condition, meaning that they are induced due to cell-cell communication. But one thing I want to stress is that you cannot find it using ligand receptor dictionary. Now, this is a time for spatial transcriptomics, and what about just using the similar concept for this one? Because we have many doublets, but instead of using doublet, we can use a two-dimensional information. So we have, can we study cell-cell interaction, or in this case, cell contact induced genes? Answer is yes. So this is the case of the gut tube cells. And the foreground is gut tube cells surrounded by at least one urocrist cells. And as a background, we checked the gut tube cells surrounded by other gut tube cells. And we found that genes such as PITX1 is highly expressed for gut tube cells when they are sitting next to urocrist. And this is a visualization. So these are gut tube cells, which is marked with red circle, and the blue circle means gut tube cells, but not with the neurocrest sitting next to it. And inside the circle, I colored it based on PITX1 expression that tells that gut tube cells sitting next to neurocrest express PITX1. And again, it has nothing to do with ligand receptor expression, and they are induced due to cell-cell communication. Another message is that depending on the neighboring cells, so these guys, these are gut tube cells next to cranial mesoderm expressed TBX1. Gut tube cells sitting next to urocrest, as I said, PITX1. Gut tube cells sitting next to splenic mesoderm expressed FOXF1. So depending on who you are, who is the neighbors, they express different gene expression profiles. These approaches can be applied to many spatial transcriptomics data, including molefish, sickfish, and genome cosmics. But there are other types of spatial transcriptomics data, including Visium here and SlideSeq. So let me start with SlideSeq, which has a resolution of 10 micrometer diameter. And I try to draw a cartoon. In a cartoon with that resolution, we may say that there are one or two cell type in a spot. So this is called a spot. In a spot, on the second row, you can have the blue color spot and yellow color spot. And also you have the transcriptome, which has the mixture of blue color and yellow color. And now it's similar to the doublet case. This is your foreground, and we call it heterotypic bed. And these homotypic beds could be your background. And by performing some statistical test, you can obtain the CCI-related genes, which has nothing to do with the ligand receptor, and this is one of the case. And also, another message is that, so BTI1A is one of the genes that we identified in the heterotypic base of the monocyte and hepatocyte. By using the cell type portion and performing some regression analysis, we can say that BTI1A is induced by cell-cell communication. However, they are from monocyte because the monocyte, as the monocyte portion increased, BTI1A expression is increased. So in other words, BTI1A is expressed in monocyte when they are sitting next to hepatocyte. So this is one of the examples from mouse liver cancer slice data, and there are two reasons, R1 and R2. And interestingly, when we apply the cell level X software to this data, there are some heterotypic pairs of the tumor cells and monocyte. And in this case, we found that monocyte expressed F13A1. And when we map it back to the spatial transcriptomics data, we found that, yes, well, these are the red circles are heterotypic pairs of the tumor and monocyte, and they are only observed in this tumor region, but not in this tumor R1 region. And F13A1 is expressed only when they are monocytes are sitting inside the tumor region, but not in the periphery. So this kind of analysis can tell you about the tumor microenvironment and how cells change their expression depending on their neighbors. Again, we try to check if they are associated with ligand receptor pathway or not. Some of them, yes. However, the genes that we identified using our approach is quite different from the genes you can obtain by running other approaches based on ligand receptor. That was the project that we finished it a couple of years ago. But what about Visium? And that is a headache, because if it is a Visium, you cannot detect the cells sitting next to each other because of the resolution. That is 55 micrometer resolution. And there could be, well, depending on cell size, there could be 1 to, well, dozens of cells inside one spot. So we try to show, cartoon as well. There could be several cell type in one spot. Then how can it determine which cells are sitting next to each other? But our purpose is to detect cells interaction, not identify cells inside. Luckily, if we have the cell type portion and gene expression from single-cell RNA-seq data, we can calculate the gene expression, and I can call it as expected value. So we can have expected gene expression. And we can also have the observed value that can be directly from Visium data. And we can see the differences between observed value and expected value. And they are, I hypothesize that they are from cell-cell interactions. And also, another important question is, okay, if we have cell-cell interaction induced genes, are they coming from A cell type or B cell type? How can we determine it? So we just extended the regression model that we had in slice data to this, to Visium data. And in this case, we use the differences, which is Y, between expected value and observed value. And we use the extended version of the regression model. And in this case, X1, X2 are cell type, and beta 1, beta 2 are coefficient. So using this model, we try to identify cell-cell interaction induced genes, and this is it. This is one example. So this is a slice data. So we kind of generated pseudo Visium data by merging several slice data into one, so that this is pseudo Visium data. You can see the cell type. For instance, this reddish one is the pseudo Visium cell type that has majority of the interneuron and causal vertical cells, and blue is the interneuron plus ependymal cells. Interestingly, we observed that genes such as, well, APBA1 is highly expressed for the Visium spot with interneuron plus causal retro cells, and genes such as FGF1 is highly expressed in this blue spot, which is based, where interneuron and ependymal cells occupy the majority. So that tells that by only using Visium data, we can kind of infer the cell-cell interaction related genes. And this is another example of the Cosmics data. So there are Cedars section of the Cosmics data and Visium data. So we just use the Visium data to predict the cell-cell interaction related genes. And in the paper, original paper of the ovarian cancer data, they already reported some ligand receptor related genes. And we just try to assess that we can recapitulate the genes related with ligand receptor, yes. So without relying on ligand receptor library, we identified the genes which is associated with cell-cell interactions by only using Visium data. And this is another example. So we obtained the lymph node Visium data, and these are the after infection with microbiome, and this is infection with control, which is called PBS. And when it comes to single-cell RNA-seq data, you may want to apply some DEG analysis, differential gene expression analysis. And we try to compare DEG analysis from single-cell RNA-seq data against our own approach to study cell-cell interaction-related genes. And when we just use this cell neighbor EX analysis from Visium, we identified pretty good number of cell-cell interaction-associated genes which are differentially observed. But we could not detect it easily using single-cell RNA-seq data. And one of the examples is the gene such as IL-3, which is observed in the Visium spot where B cell and monocytes are sitting together. And you can see these are monocyte and B cell, and these are gene expression. As you can see that these genes is highly expressed only in the region where those two cell types are enriched. However, when we look at the single-cell RNA-seq data, we did not see that. It's because single-cell RNA-seq data includes all the B cells or all the monocytes, and it's not easy to capture those kind of cell-cell interaction-related genes. Okay, this is the cell-cell interaction part. Well, and I would like to switch gear to single-cell RNA-seq analysis. When it comes to single-cell RNA-seq, and especially for those who are interested in development, what do you want to study? And there are so many good tools, including RNA velocity, which tells that, okay, this is pancreas development, by the way. So these are endocrine progenitor cells, and it has a nice visualization of the cell transition that tells that endocrine progenitor will be matured to alpha cell and beta cell and epsilon cell and delta cell. However, you want to pinpoint that, okay, this cell, does it have a potential to become an alpha cell or beta cell, or we don't know. But there are certain efforts to utilize this RNA velocity to predict cell fate. However, it doesn't work well because RNA velocity is a good visualization tool. But it does not tell about its fate. You cannot follow these arrows and tell that, okay, this is going to be beta cell or alpha cell, which is not possible. So I turned to a network system because it's easy to convert single cell RNA-seq into network. So this is just a Bayesian network, left side, one-dimensional network and two-dimensional network. In the network, the analysis is not easy, but you can imagine some diffusion processes. So there's a one peak here, one signal here, and it diffuses along this one-dimensional path and also two-dimensional path. The question is, at this point, if the signal is diffused, it will gain some signal, so a little bit low in the beginning, but it will reach it to the peak time, peak, then it will diffuse away. So you can obtain the diffusion peak time. you can obtain diffusion peak time in one-dimensional space and as well as two-dimensional space. Question is, which one is faster? Two-dimensional space is faster because there are many paths to go there. So this is just a natural network system. Can we apply it to single-cell RNA-seq data? So basically, by using diffusion, we can analyze the dimensional information. That is the message here. So from single-cell RNA-seq data, we can easily obtain the network, but it's not easy to analyze the network as it is. But instead of using dimensional information directly, we can use diffusion information. So from this source, we can simulate a diffusion to here, to here, to here, to here, which is the blue cell part, and as well as the yellow cell part. By comparing it, we can calculate the relative dimension. And as you see here, lower dimension means a little bit straight information, but high dimension means it's a little bit, the information is a little bit not clear. So basically, lower dimension is associated with longer peak time and direct and also associated relationship. So we try to use this kind of concept. It's a little bit complicated. And apply it to a known biological system, in this case, rewind data set. So basically, what they did was they prepared fibroblasts and performed reprogramming, but before they performed reprogramming, they prepared twin copy. After before that, they applied DNA barcode. So they provide DNA barcode and prepare twin copy, and one set of copy is reprogrammed. Now, by checking the reprogrammed iPS cells and check the barcode, you will know which cell can be easily reprogrammed or not. And these are single-cell RNA-seq obtained from those two copies, and these blue dots are the one that has higher reprogramming potential. And we tried to develop a software called self-fate based on relative dimension that I explained before. And these blue cells are the ones that we predicted to have higher potential to become iPS cells. And we're kind of interested to find that the list of the genes that we identified by only using single-cell RNA-seq data is very similar to the transcriptome that they identified using rewind system, which includes lots of sophisticated algorithms. And majority of them are associated with a cell cycle, and we also do find that they are there are pretty good overlap between the two systems. And another test that we can identify is the cell tagging data. So cell tagging data is a sequential barcode. And because of that, we know they will become, in this case, dead end cells or reprogrammed cells to IEF. And it is not rewind system where we can pinpoint the each single cell. In this case, they barcode to the clones. So we can get the percentage of the reprogrammed portion. And anyway, when we just use the single-cell RNA-seq data using this cell tagging system, we found a pretty good correlation between the expected ratio for reprogramming compared to observed reprogramming ratio, which is directly from the cell tagging information. So basically, this tells that using single-cell RNA-seq data, we can predict the cell fate without any sophisticated experimental setup. To conclude, we have the cell neighbor X, which is to study cell-cell communication using any type of spatial transcriptomics data, I may say, because by applying cell neighbor X2, we can still study cell-cell interaction induced genes from Visium data. And we have Vesalius, which is to detect the tissue territory and cell mapping, which is not included in our talk. And we have another approach which is called cell fate, which is to predict the fate of each single cells by only looking at transcriptome of a cell. And I found it will be very interesting to apply it to many developmental systems. And let me finish by appreciating all the people who are involved in this project, especially Patrick and Hyobin, and also Jeonghyun and my collaborator in Sungsil University. And I'm happy to take any question. Thanks.
Hey, so I was curious, it seems like the iteration between Cell Neighbor version 1 to version 2 was mainly out of the necessity for the bigger micron resolution. Is that correct?
Yes.
What does that look like? So if you were going down, so Cell Neighbor 1 works at 10, right? Or what micron resolution were you doing Cell Neighbor 1 at? And then how did that translate over at the higher 55 micron? Do you still see like a good correlation or you know, how dependent is that? Like, let's say if you're even working at like 20 micron or so forth, like how does it scale?
So Cell Label X version number 1 is for cellular resolution or 10 micrometer resolution data. And if we consider the genes that we detected using Cell Label X and the gold standard, we can still find that those genes in Cell Label X version number 2 when it comes to a visual resolution. Of course, there are certain false positive and false negative because the information that we can get directly from Visium is kind of noised data. But still, we do see good correlation. I do not show the statistics clearly.
Yeah, that's what I was curious. Like, how does that scale?
There are
Yeah.
good correlation between the two.
Okay.
I just like curious, like other great talk is really I have a lot of insight from this talk. I just like, do we identify the cell like interaction genes based on the spatial segmentations or the cell type annotation? Like, do we need to base on that, or we can identify the cell interaction genes and using those genes as the biomarkers to identify the boundaries of each cell type?
So we need to have the cell type information, and based on that, we detect what genes are changed due to the neighbors. And also, we can use those genes or niche-specific niche genes to predict the neighborhood as well. It's possible.
Yeah, because they really have a technique issues to do segmentations for the cell type boundaries, right? So it's a really good ways to see and verify the cell type annotations from the each segmentations method.
But if the cell segmentation is not accurate, then it will add
A little bit.
noise. Yeah.
Yeah, but there's a both way to verify each other's.
Could be. Yeah.
Yeah, good. Yeah, thank you for the talk.
Thank you.
I would like to thank everyone for participating in the Symposium Day 2. And I want to kindly invite you to join us for the lunch break. This is the end of the symposium. It was a really great experience to have you here. And last one favor, if you could take off next to your name at the registration desk that you did participate today in person, that would be wonderful. And that's all, folks. Thank you so much. Thank you, Zoom audience, for your participation too. And you will hear back from us about the resources post the symposium and about the questionnaires. Thank you. Goodbye.